world. So no, we're now recording. So anyway, thank you for joining us in Fallon. We want to make sure that you also get the most out of having the experience here. So again, we're going to hold questions till after all the presentations during each of the panel discussions. We're going to have a mic in the room so that everybody in Zoom land can hear us, and we'll do in-person questions and then Zoom questions. And again, if we don't get to all the in-person questions, we'll do the same thing. We'll take your name and information and make sure that you're connected with whoever needs to answer that question so that we make sure everybody gets what they need. I want to give a little bit of background on the Nevada Native Seed Partnership. It's a partnership of state, federal, and NGO agencies that share the same vision to, quote, keep Nevada's lands diverse and functioning using the right seed at the right place at the right time. And we've been working on this partnership for several years, and I'm really excited about continuing this forum and that people continue to find value in discussing these things. So we're excited to announce that the Nevada Native Seed Partnership is launching our own website so we can have information readily available to producers and those interested. To find our website, you can just Google Nevada Native Seed Partnership. The website is hosted by partners in the SAGE, and we want to thank them for not only being the technical experts, but the hosting for free our website. So please engage with that website. Again, you'll find information and resources as well as recordings of these. The goal of this forum is to connect growers, share information about production, collection, and selling of native seed, and we're excited to see everyone here today, both virtually and in person. On the back table, Stephen, you did print out the poll questions, correct? Okay, so in the back table, there should be poll questions for those that are here in Fallon to be able to answer the questions, and then there'll be those same questions will be available for the virtual attendees. So if you can please fill those out, that helps us get an idea on some of the specifics that we have going out through the forum. I'm going to do introductions a little bit differently today so that we don't have to do the whole mute, unmute process and pass a mic around in the room. So hopefully this will work. I think it's really important to understand who's in the room so that we know during breaks who to connect with and kind of who's hearing your information and that sort of stuff. So I'm going to go through kind of by registration. So you're going to hear a lot of my voice at the beginning, which I apologize for, but I think it's important. We are also interested in sharing the contact list that we have with all of the participants. If you are not interested in having your email and name and organization shared with those that attended the forum, please email Stephen, who you emailed to register here, and we'll make sure that your name is removed before we share that contact list. You could get that to him by the end of the week, by the 25th. We would appreciate that. So I'm going to go through and let everybody know who's here. So from the private sector in Nevada, we have Comstock Seed, Dayton Valley Turf, Fallon Sports, Geomorph Information Systems, Granite Seed, Nectar Nevada Plants, Nevada Environmental Consulting, Owl Creek Ranch, The Progressive Rancher, Rail City Garden Center, and Winnemucca Farms. And we have an international component, Torto. Nonprofit organizations in Nevada is Get Outdoors Nevada, Help Save the Bees Foundation, Reno Food Systems, the Greenhouse Project, Truckee Meadow Park Foundation, Walker Basin Conservancy, the Nature Conservancy, Eastern Nevada Landscape Coalition, and Great Basin Institute. Nationally, we have Great Basin Fire Science Exchange and the Institute for Applied Technology. We have tribal components from Pyramid Lake Paiute Tribe and Walker River Paiute Tribe. We have members from the University of Nevada, Reno in the Biology Department, Great Basin Landscape Ecology Lab, Co-op, Extension, and Desert Farming. From local government, we have Carson City Parks and Recreation, Open Space, Clark County Wetlands, Washoe Environmental Protection Department, and the New York City Parks. From state government, we have the Department of Conservation and Natural Resources, Division of Forestry, Conservation Districts, and the Lahontan Lahontan Conservation District specifically. From Nevada Department of Agriculture, we have somebody from Administrative Services, the Nevada Native Seed Program, and the Noxious Weed Program. From Nevada Department of Wildlife, we have representatives from their Habitat Division. We have somebody representing the Sagebrush Ecosystem Program from their technical team. And from the federal government, we have the Bureau of Land Management, Eagle Lake Field Office, California State Office, and Nevada State Office. From USDA, we have representatives from the Agricultural Research Station and NRCS State Office. From the U.S. Forest Service, we have the Santa Rosa District, Spring Mountains Natural Recreation Area, the HT Sierra Zone and the HT Northeast Zone and the Rocky Mountain Research Station. And from the U.S. Fish and Wildlife Service, we have partners for Fish and Wildlife, Reno Fish and Wildlife Office, and the Farm Conservation Program. So again, I know that that's extensive, but I think it's really important for everybody to know and understand who's in the room. And with that, we're going to kick it off to our first speaker, unless there's any questions about process that we have. And let me pull up the chat so I can see. Does anybody have any process questions? All right, so we're going to start. Our first presenter is a virtual presenter, and he's going to be, or they are going to be, excuse me, talking about the topic of seed production. So I'm going to give a little bit of introduction, and then Jess, if you could unmute Scott and Melissa, and we can get their presentation up. Scott is a botanist in the Rocky Mountain Research Station in Provo, Utah, where he works on evaluating, developing, and testing native seed species for restoration. And Melissa Landine is a research project leader for the Utah Division of Wildlife Resources Great Basin Research Center in Ephraim. She earned her bachelor's degree from BYU and has been working at the GBRC since 2017. Her research centers around big game habitat improvements and plant material development. She's also assisted with management at Utah Division of Water, I don't know, DWR seed warehouse. So I'm going to turn it over to Scott and Melissa.
Good morning. Can you hear me? Sounds like you can.
Yes, we can.
Wonderful. Hey, I'm Scott Jensen. I work at the Rocky Mountain Research Station based out of Provo, Utah. And Melissa Landine works for the Utah Division of Wildlife Resources. She's not actually presenting today, but she provided a lot of the slides that are involved in this. The Forest Service and the previous Division of Wildlife have a long history of working on plant material development together that precedes both of our careers. So that work's been going on for a long time. So today, I think what I'm going to talk about will be familiar to everyone. And that's basically our approach that we've taken to prioritize and evaluate plant material needs for the Great Basin region. Next slide, please. So as we approach this idea of how do you chew off this big elephant of developing plant materials for an entire region, we kind of partition it down into these three guiding elements. First, we needed to select a plant material development model. Next, we needed to understand the regional restoration demand and prioritize our restoration needs. And then finally, we needed to select and prioritize species. So we're going to talk about how we've gone through that structure as part of this presentation. Next slide, please. So in 2014, Andy Bauer came out with these generalized provisional seed zones for the Great Basin. And I'm assuming most folks are familiar with this, but maybe just a quick review. These seed zones are built on a couple of parameters. The first parameter being winter minimum temperature, and that combined with an aridity index, or a annual heat to moisture ratio. And by combining those two, we can identify areas on the greater landscape that have a similar climate envelope. And the idea being that if we move plant materials within the same color on the map, we're likely moving materials to areas that they're well adapted. It doesn't mean that they're the best performing sources, but at least they shouldn't be maladapted to those areas. So Andy started working on this concept back in 2009 and 2010, and he's provided us an early version of this paper back in those days. And we thought it had a lot of merit. So we began developing plant materials back in 2010 based on this concept. And since then, there are a lot of other tools that have come online that are also very valuable and very useful in knowing how to use plant materials across the landscape. We feel that this one it's a reasonable compromise. It's certainly not everything that we might want, but it's good enough for a lot of the uses that we want. Next slide, please. There we go. So I want to walk through an example of how we would use that using basin wild rye. This is a native grass, quite common in the western United States and up in Canada. Next slide, please. So if we look at this map, this comes from the Biological Information Serving Our Nation, or the USGS Bison website. And you could type in a species name and get a point map like this of populations that have been observations or specimens collected. And the utility of this is it gives us a lot more refined look at where these populations occur. So I want to talk through this map for a minute. So let's start down in Southern California by San Diego, San Jose. So we've got populations of basin wild rye that occur right along the coastline. Balmy 70 degrees most of the year. These populations probably never freeze. We move further north, there's a cluster of populations around the Tahoe Basin. Of course, we know the winter snow loads that area gets. And then the eastern slope of the Sierras, there's a nice belt of populations as we drop down in elevation along the bitterbush zone from north to south along California there. Moving north into Oregon, there's populations on the toe slopes of the Cascades and the Ponderosa zone. Those areas receive 25, 35 inches of precipitation. And then moving out into the high desert country near Burns and Bend, sagebrush community types, kind of 8 to 10 inch precipitation zones. Along the Columbia River, there's a number of populations. Those are very low elevation, a couple hundred feet. Up into Washington, that cluster of white points represents the Palouse country or Great Wheat country. Limited summer precipitation, but excellent water holding ability in those soils. Moving out well into Wyoming, there's some populations that haven't been grazed out in the greater Yellowstone system, it looks like. And along the Windy Ridges, well out into central and eastern Wyoming. Moving south into Colorado, all over the Rockies in Colorado. And then let's go over an end in Nevada. So in Nevada, basin wild rye occurs from the valley floors that are about 4,500 to 5,000 feet, all the way up over 10,000 feet in elevation. And it occurs on a lot of different soil types. Up in the Orovada area, there's some lake bed sediments that are high in salt content, and there's populations that grow in there. So with that little discussion, certainly there are a lot of different ecological climes that occur across that landscape that would allow for local adaptation to occur. And we might expect that a source that was good and well adapted to one area may not necessarily be the best adapted for the entire region. Let's move your attention to the right, the map on the right. The two yellow stars indicate where Magnar and Trailhead originated from. And then the yellow circles are areas where they're recommended for use. So the new approach to plant materials development is much more refined than the older approach. And the idea being that we can capitalize on the known diversity, the known differences in climate across the landscape to more appropriately pair materials to restoration sites. Next slide, please. Okay, let's spend a few minutes now talking about the regional restoration demand, understanding that, and how we went about prioritizing our restoration needs. Next slide, please. So in the Gray Basin, fire is certainly the largest disturbance that requires restoration. So we initially looked at the Western Fire Map, and we overlaid the Western Fire Map on provisional seed zones and built some tables out of that. Then we decided to get a little bit more precise with it, and we went to the Land Treatment Digital Library Seedings Overlay. This is a repository of federal projects that have happened on typically federal lands, and it tells us more precisely where restoration efforts are occurring. Next slide, please. So with that overlay, we were able to build a table similar to this. So on the left-hand side, we've got all the seed zones listed. Then across the top, you can see that we've looked at the native seed demand in 3-year increments. And you can see this table is quite aged. And it's been updated. Several folks have updated this in recent years, but the trend is still the same. And in large part, that's because the area of these larger seed zones is so much drastically larger than the small seed zones that they're always going to be the main restoration demand. So the numbers on the graphic aren't of particular use. Basically, it allows us just to color code the graph to give us an idea of where the highest demand is. So I like to look at this as a stoplight, and the red areas indicate areas where there's little recurring demand or not enough recurring demand to be a high priority. The yellow areas are more moderate demand, but the green ones is where there's recurring, consistent restoration demand. These are where the fires are occurring, other restoration projects are occurring. So there's 5 zones there that have fairly consistent recurring demand. And that's where we focused our restoration efforts. So you'll notice right in the center of that, on the left-hand side, zone 15 to 23 to 6 and 15 to 26 to 12 are 2 of the highest demand zones for us. So next slide, please. Okay, so let's talk about selecting and prioritizing species now. Next slide. So we use kind of a tiered approach on this as well. At the outset of the Great Basin Native Plant Project, a lot of surveys went out to folks. And this was like 20 years ago. And these surveys continue to go out, and they are of great use and interest. Initially, there were about 80 species that were listed that folks thought they would like to see for restoration uses. So our initial efforts built off of selecting plant materials from those and collecting wildland, making wildland collections and starting to evaluate those species. We also look at the BLM seed buys. And the reason we do that is it takes a while to get a new species in production. And then once it's in production, it's a bit challenging to move a new species into the seed market, even into the federal seed market. Folks that aren't familiar with the species, it's tough to buy that and stick it in a mix if you're not real sure about where to put it, how it's going to perform things. So we like to look at the seed buys to try to inform what species have some history in the market and are likely to be adapted pretty quickly. That's not to say that we're not working with species that are outside of the market, because certainly we have to bring new species there. But we're a little reluctant to give species to a grower without a contract if it's something that hasn't had much of a history in the market. Next, we feel that farm screening is high value. So we grow and experiment with a lot of different species every year to see which ones have agronomic potential and what challenges that some of these other species might have. At least that way, if we provide seed to a producer, we can give them some starting points and cautionary notes about things we observe. And then recently we started working with annuals, not smaller diminutive annuals, but large robust annuals like bee plants, yellow and purple bee plant, and sunflower. Next slide, please. So let's walk through how we've gone about that with thickleaf penstemon as an example. So we identified 76 populations of thickleaf penstemon in the field, and then we plotted those across a seed zone map. And on the right side of the map, you can see that there's a number of zones with relatively poor representation. But the larger distribution, or the taller peaks on the histogram there, come from that 15 to 23 to 6 and that 15 to 26 to 12, which were our like number 1 and 2 zones. And the 5 to 10 is actually mislabeled. That should be a 10 to 15, 6 to 12, which is in the top 5 as well. So the distribution of this species matches quite well with the restoration demand in the Central Basin Range. Next slide, please. So as we go about developing stock seed supplies, our objective is to conserve the range of genetic variation. And Jensen et al. Came out with this paper in 2010 that talked about the approach that they felt was valuable to do that. And they suggested that the seed, you should pool seed from sources that come from the same ecosystem. And in this case, the ecosystem that we're characterizing is a single seed zone. In this case, that would be the 15 to 23 to 6, and then within a level 3 ecoregion, so the Central Basin Range. There should be more than 50 parental plants that come together to create that pooled source, and at least 5 locations or populations. So this is an increased field at Fountain Green, and you can see there's 3 rows per bed, and each bed is a separate source. So we're growing these individual source in an individual bed, but they fully hybridize with the adjacent beds. Next slide, please. So here's an example of, look at the top line there, thickleaf penstemon. For that particular source, seed zone source, 15 to 23 to 6, we happen to have 18 different populations. You can look down through that list of other species that we've been working with and the number of populations that we've been able to locate for the different seed zones of those. Next slide, please. And more recently, this is a slide Melissa put together. I think the remainder of these slides are ones that Melissa put together for me. Partway down the list, I guess it's closer to the bottom, you can see Penstemon pachyphilus is represented again. In this case, there are 7 accessions. So we moved on to the next priority seed zone for us. This will be 15 to 26 to 12 this time. And we only had 7 populations that fell within that seed zone. And looking at this list, you can see that in many cases, there aren't 5. Those tend to be new species that we're working with. We like to experiment with 1 or 2 species, or pardon me, 1 or 2 or 3 sources to make sure we don't pick a bad one when we're evaluating a new species. We don't want to drop something from the list just because the source we started working with didn't have a lot of potential. So in this case, you can see there's quite a few species that are kind of in our experimental program. And towards the bottom, Palmer Penstemon or Penstemon palmeri, we had 18 sources of that was in production that year. There across the top, you can see that in 2020, we had 15 species on the farm and 94 accessions across those 15 species. Then in 2021, there were 24 species on the farm with 80 accessions that were growing. Next slide, please. Okay, so how do we go about doing selection within our sources? Typically, we're not trying to remove materials based on performance characteristics at this point. We're trying to create a broad genetic base still. We do remove sources that are incorrect cytotypes. So if most of if all the populations are, say, diploid, we happen to have a tetraploid population, we don't want to include that, because the hybridized seed will be non-fertile. So we check the ploidy or the cytotype of the different populations before we plant them out to verify that they're compatible. We have removed populations where the ripening phenology doesn't match. If it's a species that has to be harvested when that seed is ripe. And otherwise it's going to shatter. There's no sense in having sources included with a bigger pool that are going to shatter the seed early or late or not be ripe at the time. They won't end up in the larger pool of seed anyway, so we pull those out and separate them. We have removed sources that are poor seed producers as well. Sometimes a particular source just doesn't have a lot of potential, so we have removed sources that are particularly poor seed producers. And there have been some sources of particularly flax that had persistent disease issues that we removed. And then we do a lot of screening, as I mentioned, and we like to find species that yield better than 100 pounds per acre. It's kind of a threshold level for us. There are many plants that won't do that. We feel that 100 pounds per acre threshold is it's nice to be able to produce that much. Keeps the cost down on this seed when you purchase it, if you can get at least that kind of a yield. Next slide, please. So this is just a seed mix that went out on the Hamlin Valley project a number of years ago. I just wanted to highlight, so here we got Nevada Showy Goldeneye. There's 500 pounds of that we collected from the wild out in southeastern Nevada, grew up at our farm at Fountain Green, provided to a grower. These were grown by Jerry Airstrom in Vail, Oregon. And then these seed from that was purchased back by the Utah Division of Wildlife and used on a project by the BLM on the Cedar City district. So both thickleaf Penstemon and Showy Goldeneye made the full cycle there. So it's exciting to see those materials back on the landscape. Next slide, please. Oh, I'm going to have you go back up just one more for a second there. And I just want to point out here, so if we're looking down that list, Lewis flax, the maple grove, is another material that was released by our lab a number of years ago. The scarlet globe mallow and the Palmer Penstemon were wildland collections. Arrowleaf balsam root is a wildland collection. Monroe globe mallow is a wildland collection. Gooseberry leaf and yellow bee plant are also wildland collections. You can see there's quite a bit of volume of seed that's available from some of those wildland collections. But I would also suggest that those are species that we could move into agronomic production as well and increase their reliability in the market and probably decrease their price a bit as well. Okay, next slide, please. So this is a diagram out of a paper that was published this year. This is a study that was done with thickleaf Penstemon. And so on the map itself, you can see a lot of pie charts. And each of those pies ties back to a small blue circle. So the small blue circles represent the location of Penstemon, thickleaf Penstemon population. And these all originate from that 15 to 23 to 6 seed zone. And what we found was that within mountain ranges, there was a lot of genetic exchange. So looking at the left side of the map, there's quite a bit of pink in those WPs. So that's white pine range, 1, 2, 3s, and 4s. You see a lot of pink color that's similar across those. As we move to the right, the SCRs, the Snell Creek range, there's a lot of orange that's represented in those, quite a bit of green as well. Moving down and to the right, the Needles range, NR populations in purple, and then the WWs or the Wawa Mountain populations in blue. So this is a bee pollinated species, and in large part, the bees are able to move within the mountain range, but not across the valley bottoms. So there's not much genetic exchange east to west across the basin, but quite a bit of genetic exchange north to south. Interestingly, if you look at the very top of the map there, ER1, and then kind of down right in the middle of that map, ER2, those are on the same mountain range. They're quite distant on this map, but there are other populations of thickleaf Penstemon that occur in different seed zones in between that country and between those 2 points that allows for genetic exchange. So now I'll direct your attention to the larger pie chart on the top right. So we've made wildland collections from all these populations on the map on the left, and we've pooled seed together in a stock seed supply. And the composition based on weight is represented by the top pie chart. That seed mix was then distributed to Jerry Airstrom, and he grew that out in Vail, Oregon. And the large pie chart right below that shows the genetic composition of his seed production field. And you can see that there are some contractions in some populations, some expansions in other populations, but everything is still fairly well represented. That seed production field was, of course, high quality soils, higher precipitation zone than what a lot of the native populations would have occurred in, and a nicer growing environment. But nonetheless, there was still some adaptation that occurred at that seed production field. If we look at the 10 smaller pie charts across the bottom, these represent different treatments in the Hamblin Valley. So we took that same initial seed mix and planted it out in different locations in the Hamblin Valley. And in some cases, it went into the same seed zone. In some cases, it went into a warmer, drier zone or a cooler, wetter zone. Some of these went into chaining treatments, others went into mastication treatments. And I'll just ask you to look across those pie charts and say, what trends do you see? And to me, the trend is that the value in having a broad, diverse mix was that there's no consistent winner. Every year, every different combination, something else is more successful. What was good in one year or one treatment may not have been the winner in the next year or the next treatment. So in this case, there was a lot of value in having a broad genetic mix so that something made it every year. Next slide, please. Okay, so we do a lot of work with the Great Basin Research Center in Ephraim. They've got nice facilities to work in. Next slide. A number of different farms that we have access to at Snowfield Station in Ephraim, at Fountain Green. And over the years, the project's been able to get a bunch of specialized equipment that really facilitates our work in this regard. Next slide, please. So when we're increasing seeds, our preference is to direct seed the beds. So this is the images on the right. We like to use a precision seeder that allows us to meter the seed very precisely down the rows, so we can control how much seed we're putting out. We almost always cover that seed with insulate fabric. Our experience has been that we get better emergence and establishment if we cover those plots with insulate fabric. And then on the right, bottom right, you can see that we also get a lot of weeds that come up on that fabric. It kind of creates a mini greenhouse that benefits germination of everything that's in the bed. So we've gone to solarizing those beds the summer before we plant them now. Solarization involves laying down a thin plastic sheet across the bed, tucking it in just like we do the insulate fabric, leaving it on for the hottest 6 weeks of the summer. That denatures a lot of the weeds, weed seed that's in there, and also reduces the nematodes and plant pathogens that are in that soil. That's helped quite a bit in the amount of effort it takes to weed those beds. Where we don't have adequate seed to do that, we grow plugs in the greenhouse, and then we have a transplanter that allows us to drive down the bed and put those plugs in the ground. Next slide, please. On the right, you can see the drip subsurface irrigation system that's been put in place. We find that quite useful. The less water you can put on the surface, the fewer weed seeds that germinate. We'll often surface irrigate with little rainbirds when we're transplanting plants to ensure that they've got moisture to establish. But as soon as those roots get down 6 or 8 inches, we transition away from the surface irrigation and rely exclusively on subsurface irrigation. The bottom left picture is a little tool called an Eco Weeder. It's got 2 round discs that are controlled by handles and driven by the PTO shaft on the tractor, and they rotate, and the idea there, you just drive down the row and you can get really tight to the plants. That's really useful early in the spring when the soils are moist and the weeds are small. And if you miss that early opportunity and the weeds get bigger, the soils firm up, that tool doesn't have a lot of utility. We use a propane flamer for residue management, and then spend a lot of time hand weeding. Next slide, please. We use a variety of different methods to harvest seed. The combine is, of course, the favorite tool. Whenever you've got a crop that you can direct combine, that's by far the easiest. But we also do a lot of hand collection, whether that's with a badminton racket and a hopper or with vacuums. The silver cart in the middle has got a cutter bar on the bottom of that, and we just push that through a crop. So we'd use that on a species like lupin, where we have to kind of harvest the whole plant, and then you need to let that whole plant dry out. Next slide, please. And then there's a lot of effort that goes into drying out that seed and cleaning it. And all that work is done down at Ephraim by the Great Basin Research, their staff. And we're just really appreciative of the time and effort they put in. Next slide, please. So Melissa put together this little slide that shows the amount of time that they spend in various aspects of the farming. I like to think of a farmer as out there driving the tractor, but time spent planting and seed harvesting represents about 12% of the total time that they feel they spend on the farm. The vast majority of it is spent with weed control. And at Ephraim, they do a lot of hand weed control. Most herbicides aren't labeled for use on the farm for native species, so they try to abide by those rules and control weeds with solarization and hand weeding after that. Next slide, please. So with that, I just want to thank our partners, the Great Basin Native Plant Project, funded by the BLM, Forest Service, and where the rubber hits the road has always been with Utah Division of Wildlife Resources. They've been great partners to have all along. So I don't really have this tied up in a nice little tiny bow with, hey, we'd love to see you grow native plants. It's not because I don't think there's value in it. There's huge value in it. There's a lot of room in the market, I think, but it is very challenging. And I'm always looking for new folks who are interested in growing things, and we try to mentor them along, but it's a big challenge. Thanks for the chance to visit with you today.
Thanks, Scott. I can get you a mute. Great, perfect. Thanks, Scott. Really appreciate it. So if people in the room or in the Zoom world can put any questions into the chat. Again, we're planning on doing questions and answers during the panel section. So appreciate that, Scott. I thought that was really great. We're going to move into our next presentation. Jackson, if you want to come up and we'll get your presentation pulled up. No, she's going to change your slides for you, unfortunately, but I'll have it up here so you can look at it.
Awesome.
So you'll have to say next, unfortunately.
Yeah, that's fine.
I will download these in the future so that we don't have to have this awkward pause. All right, well, that loads up. I'm going to introduce you. So you're not going to be able to see yourself because this is up,
That's just
but this is what people are going to see,
okay,
so just
great.
so you know. All right. So thanks for being here. Jackson is a graduate from Utah Valley University and is a premier seed sales manager, and just celebrated his 8th year with the company. They are currently growing out 40,000 custom plants for the Utah Division of Wildlife Resources, and has collected numerous obscure species for customers, including work in national monuments.
Hey, thank you so much for the introduction. I'm Jackson Eady with Premier Seed Company. Today I'm going to be talking about wildland collected seed, kind of the other part of the native seed procurement to production seed. Really quickly, I want to thank Scott Jensen for his presentation. I've worked and corresponded with Scott Jensen quite a bit over the past 8 years, and he a lot of what he says honestly goes over my head, but whenever I have a very specific question, he can either answer it or he goes in and does a lot of research and answers the question for me really thoroughly. And he's even gotten us out of a couple jams when we had questions or concerns with customers and we needed an expert opinion. So I'd just like to thank him for the contribution he's made to this industry. He's a good guy and he's a really good worker. I think I have a lot more time to kill, so I've got you guys trapped. A lot of people have asked me what I do for a living, and I tell them that I sell seeds, and they're either completely disinterested when I say that, or I have to explain to them for about 15 to 20 minutes what I do. And that's because there's a lot that goes into wildland collecting seeds. Scott brought up provisional zones and ecoregions, and wildland collected seed used to be sold by county and state, which was a lot easier. You just mention what county and state it comes from and slap a tag on the bag and it goes out the door, and anything from that county and state will grow way up on the top of the mountain or down in the valley. And then people start asking questions about why their projects and jobs aren't turning out. And I really do believe in the provisional zone and ecoregion way of doing things, but it definitely adds a challenge to the job, a challenge in scouting, but also at the same time, it's a real challenge for cultivated seed and production seed. If they want something from if they need a lot of things from the market needs a lot of things from like very specific provisional zone one year. It's not like they all of a sudden in retrospect, they can just vamp up cultivation. And so it's kind of the wildland seed collectors who need to come in and fill those gaps. We're a very impromptu industry. Generally, they want the stuff that we got that year, the government, as well as private procurers. So I just wanted to explain to the people who procure and purchase seed some of the cost drivers that are involved in collecting wildland seed. Next slide really quick.
So you won't see it, unfortunately.
Oh, okay. No, you're good. So what? Let's go back just one really quick. What we do. So I work for Premier Seed Company, and we mostly do wildland collecting of seeds. We do that across the Great Basin, a little bit in the Colorado Plateau, as well as down in the Mojave. And we focus mostly on shrubs and then also forbs and grasses that seem to be inconsistent when it comes to cultivation. And like I said earlier, it's a way to fill in the gaps of cultivation and production where they can't meet demand every single year. And a lot of the shrubs can't really be cultivated easily anyway. Generally, when I'm going around trying to get permission to collect sagebrush or something like that, when I do see somebody who has a big private piece of land full of sagebrush and I ask them if I can go get the sagebrush off of it, they ask me if I'm going to mow it all up for them and get it out of there. So generally, when somebody has a bunch of shrubs on their land that are natives, they want to get rid of it. So it's a little bit different than the cultivation side. A lot of these plants that are natives are considered pests in large quantities for people who are trying to manage their land for something other than growing. And so it's kind of a win-win situation when we go out into the field. Next slide. Here's some pictures of kind of what it looks like to collect the seed. We use machines to machine up some of the seed, especially the grasses. But when it comes to collecting shrubs and forbs, there's a lot of racketing, or sometimes just picking flowers, and that can be an all-day job. When it comes to the racketing, I've got a visual for the people who are here in person. I don't know if you'll be able to see it on the in the virtual. So this is Justin.
Sorry. Yeah. So Valda Lockie, she made this hopper. Okay. She's the botanist in the Eagle Lake field office. But yeah, these are the hoppers that when they're custom built like this, you know, they're a lot better than using garbage cans. They're reusable. They stack well, and they're quite durable. She's made them. There's been a lot of trial and error that, you know, these are made out of Cordura nylon. A lot of them are made out of like canvas and stuff like this. The ones that Valda makes out of Cordura, they last many seasons, you know, so they might be a little expensive on the front end, but it's way better than hauling around a bunch of broken garbage cans and what have you. So anyway, just a plug for those.
Thanks, Justin. So yeah, when it has to be racketed off, then it has to lay out, it has to dry. You can't just throw it in a bag and put it in a storage unit somewhere. It'll get wet and it'll start to mold. So it needs to be laid out flat. Can I get the next slide? We've also been doing some plant grow outs for the DWR. We're doing 40,000 plants right now, as mentioned in the introduction. Next slide. We've gotten a little bit into showing milkweed production, and we've been trying to do more things with milkweed, such as butterfly milkweed and small milkweed. Next slide. So our customers are generally the Bureau of Land Management, DWRs, Forest Service, wind farms, mining projects, solar farms, Department of Transportations in different states. All these different sectors all need both cultivated seed and wildland seed in order to fill the entire environmental, like each piece of the environmental niche of any given ecosystem. Next slide. All right, so now for the cost drivers. There's a lot of different challenges that we face in the industry. There's just regular variable and fixed expenses, some of which are obvious and some of which are you maybe wouldn't consider that the company would be facing. There's risks and variance. There's a lot of waste, and there's also crop failures, which isn't just a cultivation problem. It's just as big of a problem when we go back to try and check on a place that's been given and given to us, and then all of a sudden it stops. So next slide. All right, so some of the main expenses of wildland seed collectors are tagging fees. We have to pay people like the Nevada Department of Agriculture and the Utah Crop Improvement Association, not just to tag our products, but they go and follow us around on our scouting trips, maybe a week or a month behind us, and go out and make sure that things are coming from where we say they're coming from, and that the plants are where they say they are. We have to pay for garbage service, which you wouldn't think would be that much, but we fill up this 30-foot by 8-foot garbage bin probably a couple times a week during harvest season. We have to do a lot of seed purchases from contractors, especially during harvest time. You can't keep a full payroll of that many people all year round, and so you have to bring on a lot of people from the sidelines when it's time to harvest shrubs specifically. Scouting is another big expense, and it also takes up a lot of time. And even more so, you can't just send out somebody who's been working for the company for 2 months to go and scout out seeds. You have to have somebody who understands about the shrub and what a good stand looks like and how far off the seed's going to be and how windy it is and how much rain that's been getting there recently and what that's going to do. And if the seed looks rotten or if there's bugs in the area, there's a lot of things that need to be looked at. And if you just send any random person out there to go look at it, they have no idea what they're looking for and what to not look for. And then lastly, insurance and performance bonds. That's been something new and tricky that I've had to navigate recently is the performance bonds, getting bonded and making sure that if we fail to meet job requirements, then we're on the hook. So doing all the paperwork for that, as well as putting up a certain amount of money for what the project is worth, also adds to our underlying cost and can be another risk that adds variance. Next slide. So labor is a huge expense. Cleaning seed and harvesting seed isn't a very easy job. You can't just pay somebody minimum wage to do it. You have to have people who are willing to go and travel around the United States for weeks, if not even up to like a month at a time, and live in motel rooms and eat fast food and hang out in the motel out in someplace like Fallon, Nevada for a whole month and eat KFC and Taco Bell and just be without their family for a long period of time. So you have to pay somebody not just for their labor, but also for the opportunity cost of being at home with their family, just working a regular 9 to 5. Also testing fees, they really stack up. When you're selling seed to the government, they want it to be what you say it is by the time it hits their by the time that it hits their shelf. So like we have to test things over and over and over again until we can kind of whittle out the outliers and figure out that we've got a consistent product. And so we spend about $25,000 a year on testing. Next slide. Another big expense for us is buildings. We've got 7 greenhouses, one of which is like about the size of a small airplane hangar. It's 30 feet up. And that's so that we can fit these big pieces of machinery, these shakers and sifters that allow us to clean seed quickly and efficiently. It used to just be an industry of guys standing on 2 ends of screens, shaking it back and forth while either the product or all the chaff fell through, one or the other. Now the industry has been mechanizing across the board. It has been for cultivated seed for a long time, but wildland seed, we've started doing that a lot on our end too. And so there's the cost of the equipment, there's the cost of the buildings, and it's thousands upon thousands of dollars, not just to purchase, but also to maintain every single year. Next slide. So the buildings are expensive themselves. There's some pictures of the biggest greenhouse that we have. It's like 6,000 square feet, not to mention how much volume there is up on the inside too. But we also have to keep those plants warm. Those 40,000 plants that we're growing out right now have to be heated the whole time. And it costs about $2,000 a month to keep those greenhouses warm. So that has to be factored into the price of everything as well. Yeah, like it's even more expensive in the wintertime. It gets super cold where we're at. We're based out of Spanish Fork, Utah, and it gets to be 100 degrees there for like 2 months out of the year, but then in the wintertime it's freezing for like 4 months straight. And so we have to keep a thinly filmed greenhouse at above room temperature and moist for all those months. And there's layer of snow this thick around the outside of it. So it can be kind of challenging to do that. And it's very difficult to look at the gas bill that comes in every single month in the wintertime. Next slide. So as I stated before, there's crop failures and they're not insignificant. Some products that are asked for on every single year, on every single job, especially for jobs in the Mojave, but also some in the Great Basin, are reliant on these California super blooms, where there needs to be like specific rains right at the right time in the winter and early spring in California. And if that amount of rain doesn't come, you can see from the pictures the difference between a good year and a bad year. It's like being on the moon versus being in like one of the most gorgeous places you've ever seen in your life. Like the biodiversity of the forbs at that time of year is like, it's breathtaking. Like it's like you can't take enough pictures. It's truly beautiful. It's like a kid in a candy store. You just don't even know what to pick. Like what am I gonna get this year? Like trying to figure out how you're gonna get more guys. Like they're only gonna be out for so long, and you got to get it while the getting is good, because then, as we've been facing right now for the next 3 years, there is nothing. And government and private purchasers don't stop asking for it. And if somebody else has it and you don't, the price goes way up and they can make all the money. So, and you don't get jobs. Like there's a few species where like, that's what they're looking for. And if you don't have it, you're not getting the job. So it's really important to collect all that you can. And the price does go up when the seed basically becomes rare. That's been happening in the cultivated grass area recently. There's been a couple bad years in a row, and it seems that all the native grasses are becoming more and more expensive. Indian rice grass in particular is one that I noticed just recently has doubled, maybe tripled in price in the past few years. And that's something that we're likely to see with a number of species in the wildland seed collecting side of things. Next slide. Yeah, so it's feast or famine. This is globe mallow, and globe mallow is one of those forbs that's very finicky, unreliable. I don't know anybody who grows it. If you do, keep it to yourself, because I don't think anybody's got a good repetitive yield on globe mallow if they're trying to grow it, but it's worth a fortune. And when you find a good field of it, it brings like a tear to your eye. You're all like, it's like someone's playing a trick on you. You can't believe it. But most years it looks really sparse. It's hard to get a good yield off of it. And it's been getting worse and worse as time has gone on. There's some spots that used to have a reliable crop every other year, every 3 years, that haven't produced anything since a decade ago. And that's not primary source information, but it's one of the complaints that I hear throughout some of the Utah wildlife seed collectors, and they're back and forth about how things used to be so easy. You could go out to this one certain basin and you could pick for weeks, and now that place just never has any seed anymore. So next slide. Yeah, insect damage can be just as costly as poor rains or just higher heat, and anything that there's so many variables that can make what used to be a good stand turn into a terrible stand. And we've seen so many areas, whether they get eaten away or burned away, just turn into an area that can't produce good seed anymore. And it's more common that, and it's probably due to environmental shifting, that like crops are getting more difficult to get yields off of as opposed to the other way around. Like it's not like there's spots that used to be terrible to pick at that are becoming good. It's always moving that one direction. Like sagebrush is getting harder and harder to get from southern regions, and it just feels like a line just shifting from south to north as it gets worse and worse. Next slide. Difficult year-over-year collection. This is Mexican bladder sage. It's not only is this seed difficult to collect because it rarely produces seed, but when it does, it's got this paper thin shell. And so you have to be super duper careful collecting it with these little tiny seeds on the inside. It's really hard to get a yield off of it. And so people will ask about this seed, and we won't even really touch that one anymore. It's too much of a hassle to get. So a lot of these seeds that people want, it's not economically feasible to get it for them, and we offer substitutions because it's just not reasonable. Next slide. So collecting seed is really hard work. You can see some of the safeguards and some of the times that the safeguards didn't work out of this picture. If you have any type of seasonal allergy at all, it's not something to get into. I generally have to take a non-drowsy allergy pill every time I go down to the warehouse, and walking into the sagebrush cooler, it's like getting hit in the face with a baseball bat. It just changes your whole day. It's hard to drive home. It's really difficult work. There's tons of times, like it's almost like chemists or something will go down there, and everybody's in these overalls with big shirts pulled over it, wearing full gas masks while they're in there with the shakers running, and there's dust kicking up all over the place. Our neighbors, because our commercial property is right next to a residential area, this guy would complain about the dust all the time because we started paying for his car to be washed at the car wash, because he would come out every morning and his car would be coated in dust. And so we got him one of those car wash passes, and he kept complaining. So we eventually just bought his property from him and he moved away. But which was nice. We've been expanding onto it, and it was a good purchase, but we needed that guy to stop complaining, but there was nothing that we could do to keep the dust down. And so working right up with your face, and that you definitely need a gas mask on, otherwise we'll be looking like Justin here in this picture.
But it paid for itself though. So this guy would talk to our employees all day long. So like, safe by not having him there. You guys are working, it's worth buying stuff.
Yeah, he was like that guy on the Tim Allen show where you could just see his eyes peeking over the top of the fence, hassling you. Okay, next slide. Another cost driver that is one of the most difficult is variance. This is an example of sending the same seed to 3 different labs. Most seed is sold on a pure live seed basis as opposed to bulk pounds, which means that based on the results of your test, you could, like, the price is generally the price, and it's generally agreed upon before the seed test comes in. So if it comes in at a 7%, 7.5%, as opposed to a 24%, and you were expecting a 24%, then you just sold your seed at 1/3 the price that you were expecting to get for it. Which, if you have to do that sometimes, that means that those types of situations, it always gets passed on to the customer in any industry when there's variance. So it's already factored into the price that every once in a while, we expect to give you seed at a certain rate. We go and we get it tested at the place that you want it tested at, or at this, or maybe it comes in lower, and all of a sudden we have to sell it at a huge discount, generally at a loss. But even more importantly at this slide, there's been an issue in our industry that we've been trying to iron out since I've been around, and I just really don't know how it's going to be addressed, but like the variance is astounding. Like, of course, when it comes to stuff that's less clean, like sagebrush, versus these really, really clean grasses, there's going to be a higher variance for sure. But this is an actual example of when we took three samples of the exact same out of the exact same bag from the exact same spot and sent it to 3 different labs, and we got that type of a variance between 7%, 18%, 25% respectively. Which one's right? Should we take a huge cost reduction, or should the customer pay way more? And like, who's responsible for this? Is it us? Is it the customer? Or is it the variance between the labs? Next slide, please. Here's the actual seed test, just to show you. We've had problems too, where if you send it back to the same lab with the same lot number, you get the same result that you got last time, whether or not you cleaned it up. Almost like maybe the guy who's got the least motivation in there saw the same lab number on there and decided to bump the TZ up or down by 1 point and just send you back the test, even though you just reconditioned the lot. If you send it back with a different lot number on it, you might get entirely different results because somebody else at a different time of day retested it, and what is viable and what is not viable. Like, you had a good lunch, he's in a better mood now, and all of a sudden you got a higher seed test. It's more of a art than a science, the seed testing, which has been a major problem for us on our side, because you do everything you can, and then it's all left up to this test that has so much variability in it. It drives the price of the seed up. And when you get good seed, you're paying a premium for it because of this issue. Next slide. So another risk is that we're driving all across the country all the time. We've got one worker right now where if he gets in another wreck, we're just going to have to take it out of pocket, because he probably drives like 50,000 miles a year, and he doesn't pay the greatest attention, and he backs up into light posts here and there. And I mean, when you're driving, we drive these trucks into the ground, like brand new trucks, practically totaled in 4 or 5 years because the engine's toast. Like, we're not just driving with them, but we're towing big long trailers. We're towing them all across the country. They're fully loaded on the way back from wherever we're coming from. And sometimes trucks roll, trucks flip. Luckily, nobody's died, but we've lost a lot of equipment, and our insurance bill is insane. I don't even want to talk about it. Next slide. Waste. So we do so much green waste and regular waste that I can't even believe it. I don't even know how many tons. There's more chaff than there is seed with a lot of the wildland stuff. So if we're doing hundreds of thousands of pounds of seed, then we might be doing 7 figures on the waste, potentially. They come pick it up a couple times a week, maybe every other day during harvest, and even during slow times of the year, like right now, they still come and pick it up once a week, every 2 weeks maybe. And it costs us nearly $40,000 a year just to get rid of all the dirt that comes out of the stuff from the harvest. Next slide. All right. All that being said, with wildland collected seed, the price of it since the 1980s has stayed almost flat. Probably has done better than inflation. I don't even think that it's inflated up like with inflation. And there's cost drivers that I've explained that have brought it up, but the big cost drivers that have driven the price back down to a reasonable level is competition, scale, and mechanizing and using equipment to do the cleaning. This used to be something that two guys in a screen could do in their backyard. Now it's you got a big warehouse and hundreds of thousands of dollars worth of cleaning equipment that can clean what 100 men could do in a day in an hour. And so the prices have stayed almost flat, which is really crazy. I don't think very many industries could say that about products that are remaining. And I would say quality has gone up too. Testing does have its variability, but it's probably better than it was back then. And when it comes to doing things by provisional zone and ecoregion and having to stay on top of that versus just it came from Iron County, Utah. So go ahead and throw it in Utah County. They're close enough. Like that's not how we do it anymore. So I would say that the product has gotten better while somehow remaining at the same price, even through all that inflation and even with all those added cost drivers. Next slide. So there's some species that are both native collection and commercially grown. This is an example of squirrel tail. Squirrel tail. I said before that a lot of the time that there's native collection in the wildland collection, it's to make up for a gap between supply and demand for the cultivated seed. But there's also some other things too. We found that with our native collection squirrel tail, you get more seeds per pound than the commercially grown stuff. Next slide. Generally, there's just PLS pounds that we do stuff by, but people don't regularly factor in seed count on when considering what they're purchasing and how much they're getting. In this specific example, the wildland seed had about 20,000 more seeds, 22,000 more seeds per pound than the commercially grown. And it that made it so that it had about 30,000 pounds more viable seed than the commercially grown. Whereas if you were to just look at it from a PLS perspective, they would seem a lot more similarly matched. But you're getting almost 50% more product by having purchased seed with a higher seed count. Next slide. So yeah, if you look at it from just PLS only, it's 1.07 PLS pounds to every pound for the wildland versus the native in this scenario. But if you factor in the seed count, then it's 25% more than the commercial seed. Next slide. So why buy wildland collected seed? One of the biggest reasons is that it's easy to get from a specific provisional zone and ecoregion. And if all of a sudden you realize that you need that at the end of this year and nobody's growing it over there, we can go get it for you. And so we're fast, we can move quickly. It doesn't take as long as somebody who's gonna take a field and cultivate it over the next 5 to 10 years in order to get you a consistent product. The prices have remained the same for over the decades, and I would say the quality as well as quantity from our section of the industry has gone up. There's more species that you can get wildland now than ever before, and the quality of it's going to be better now than it's ever been before. Regulations have gone up. People like the Nevada Department of Agriculture and Utah Crop Improvement keep us in check, as well as seed testing services are scrutinize the product better than they have in the past. And seed can be custom collected. So if you have a very specific job, this is like especially applies to the private sector or specific BLM field offices, but if you have a very specific job that you have that you need to have planted in the next 3 to 5 years, a wildland seed collecting company can go out and they can custom build a mix for you by going out to your specific area, collecting things from your district or from around the area that your project is in, and then you can put it right back in the ground right there. Next slide. Lastly, I just wanted to mention that it's important to purchase wildland seed from established companies. There's lots of companies, big and small, and I think that it's important to purchase from wildland seed producers who have a lot of experience in the industry, who are able to use the machinery to get the product as clean as possible, who are able to test and retest for you, and who are able to have the mobility power to go out and move and do a custom collection for you and actually go and get everything from the very specific spot that you want it. A lot of these companies, there's a lot of them, it's not just us, but we've established trustworthy relationships between the regulators as well as the BLM and the state and the Department of Transportation industries. And for anybody who's looking to purchase wildland collected seed, you can reach out to any of these companies and get their accreditations and their past performance histories. I think that's all I got. So yeah, thank you guys for listening to me. Hope you guys understand more about the wildland collecting, wildland seed collection industry and our contribution to the market.
Thanks, Jackson. Really appreciate it. All right, so we are a little ahead of schedule, which is okay. I think that'll leave more time for discussion before we break for lunch. So we're going to take a 15-minute break right now, and then come back and we'll hear from Shannon Swim about seed storage and cleaning, and then from Ed Kleiner on some of the secondary benefits. So we'll take a 15-minute break and we'll see you all back. So thanks again, Jackson and Scott, for their presentations. And if you want to put your questions in the chat that we can get answered during the panel discussion, and those in the room, if you want to write those down. Thank you. All right, everybody, if we could get back to our seats and attention in the Zoom world, we're gonna get started with our last two presenters for this morning. So appreciate everybody being patient with this sort of hybrid. So just to reiterate, during the panel discussion, we're gonna do in-person questions with a microphone or moving around the room. So those that are here during that time, you can just raise your hand. And then we'll move to the chat questions. I appreciate that some of the answers have been answered in the chat already from the virtual side, but I do want to reiterate them. So in case people here in person can't read those chats. And so I'm going to review those as well as moving forward with some of the questions that haven't been answered. So speakers that are in Zoom, if you want to answer in the chat, that's fine, but we can also just save the answers for the panel discussion. So our next speaker is Shannon Swim. Shannon received her Master's of Science in Natural Resources and Environmental Sciences from the University of Nevada, Reno in 2014. After graduating, she worked as a research technician in Dr. Ledger's plant ecology lab, setting up experiments, collecting data, and assisting with native annual forb increase, as well as constructing, maintaining, a native seed cleaning and storage facility. She has also been the Sagebrush in the Prisons project coordinator for Nevada and California, growing hundreds of thousands of sagebrush plugs with incarcerated individuals for the past 6 years. She will soon be starting a new adventure at the Walker Basin Conservancy as the native plant nursery manager, which is exciting news. And so I'm going to turn the floor over to Shannon.
Thank you, Megan. So as Megan said, I am currently the UNR Native Seed Bank manager, but I will be very soon transitioning into a new position at the Walker Basin Conservancy. But today I'm going to wear my UNR hat and I'm going to tell you a little bit about this seed cleaning and storage facility that we have set up. Next, please. Thank you. So today we're going to talk about this native seed processing center. I'm gonna give you a little bit of background about it and some of the history. We're going to talk about what seed cleaning is. We're going to talk about some of the equipment we have, some testing capabilities that we have, our storage facility, and then also some lessons learned along the way. Next, please. So kind of full circle, this project was actually part of a larger collaboration with Walker Basin Conservancy in an effort to get them plant material that was locally adapted to their area. The Walker Basin gets around 5 inches of precip a year, so you can imagine having genetically appropriate native plant material is kind of a big deal and needed to increase the success of their restoration projects. It was also a way for all of us to just kind of wrap our head around what this process actually looks like. What does it look like to figure out what seed you're going to collect, what species, where they are, how much do you need? How do you get a permit through the BLM? How do you get seed certification through NDA? And then once you have all this seed, what do you do with it? How do you clean it? How do you store it? And then how do you use it later? So it was a really, really nice project for all of us to kind of get our head wrapped around about how does this actually work? Next, please. So we were able to collect 17 different species that first year. As you can see from that list, the vast majority of those are shrubs, but we were also able to collect some grasses and some forbs as well. Most of this was done through hand stripping, but we did also use tennis rackets with tarps and buckets and whacking the shrubs, especially for some of those pokey shrubs on there. And then we did also use scissors and sometimes cut some of these things. Next. So now we had all of these different seed collections, we needed to dry it. I think that Jackson had a really nice picture up there earlier of all of the seed laid out on the warehouse and all of these different tarps and drying all of these things. I did not get an awesome picture like that. But we were able to acquire some space at UNR, and it had these shelves in it. And we lined those shelves with tarps, and then we put the seed on there, and then we would go in every so often and we would turn that seed to make sure that it was drying properly. And during this process, we learned that some species are more prone to molding than others and needed a little bit more attention. So once we got it all dried, then we had to figure out what we needed to do with it after that. Next, please. So we really had no idea what we were doing when we started this venture. We had no idea what it took to clean seed and what type of things that we would need to do this. So we decided to contact the experts on this. So we reached out to the U.S. Forest Service Bend Seed Extractory up in Oregon. We also reached out to Lucky Peak Nursery in Idaho, as well as the National Seed Lab, and through many communications with them via phone calls, emails. Bend even had us come up and do a site visit. Through all of their expertise, we were able to put together this sort of boutique-y cleaning center. And this center wasn't designed to do mass quantities. What we were really looking for was the ability to be able to do a lot of different species. So a lot of versatility to accommodate a lot of variation, not only between species, but also within species. And so through all of these fabulous people, we were able to put the center together. I do want to give a special shout out to all of these folks because we really could not have done any of it without them. Next, please. Okay, so along this process, through communications with these people, we learned that there are actually two different steps to cleaning seed. So the first step is seed conditioning, and that is where you're actually breaking up the plant material. And then the second step is actually the cleaning part, where you are separating that plant material from the seed. We also learned through this that all of these pieces of equipment that are commercially available are designed for agricultural uses. So they are not made for little teeny tiny native collected seed. And so most of them require some type of modification. We also learned that there is a reason that in agriculture, they breed uniformity into everything because it simplifies things, which I didn't really understand until I started trying to clean all of these different species. And it's complicated and it takes a lot of different methods and a lot of different ways. And sometimes there's so much variation even within species that you have to modify and adjust how you're cleaning these things. And these were things that we really had no idea when we first set out on this adventure. Next. So for seed conditioning, one of the pieces of equipment that we ended up getting was a Westruck brush machine. And one of the reasons we went with this is because it is incredibly versatile. So I don't have a fabulous picture of the mantles in there, but inside that white drum, there are these different mantles. And these mantles can be either perforated metal or they can be wire mesh, and they slide right in there. And then there's a brush in there where you can control the speed, and that brush pushes the material up against the side of that mantle and breaks apart all of your plant material. This piece of equipment is really versatile because you can use these different mantle sizes. It does, however, still have some limitations. One of those limitations is that it requires a certain amount of material to function optimally. So you need at least a paper grocery size bag of material for it to process things optimally. If you don't have that much material, you can try to run things through twice. And sometimes that works, and sometimes it just doesn't work and you don't have enough material and you really can't use this piece of equipment. So that is kind of a pretty big limitation when you are thinking about, wildland seed collections that aren't huge and, there's 2 ounces of seed or of plant material that you're starting with. Another limitation that this piece of equipment has is that it doesn't really handle the long, slender papacy asters so well. So that's like a whole bunch of our native species, right? It will process them, but typically you end up with a lot of seed breakage, and then all of the seed gets stuck inside of those mantles and you kind of hate life afterwards. And so it's just don't put them on there. You'll just be sad about things later. So the amount of material that you need, and then also the long slender papacy asters are a bit problematic with this machine. But it also processes a lot of things really, really well. So you can see on that picture up there on the right. Sorry, Megan's looking at me. I'm really terrible with my right and left. And so, and there's no pointer for you online people. So bear with me. But you can see this is Atroplex canescens. So the ones on the right is the seed before it has gone through the brush machine. And it's got all of those bracts and all those wings on there. And then you can see the one on the left, that is after it has gone through the brush machine. That's just one run. And it's taken all of those wings off of there, and you're just left with its adorable little alien seed. Next. So we also have two other pieces of equipment for conditioning. We have a thresher. So the thresher is a little bit different from the brush machine in that you don't have these mantles in there. You just have basically these pretty rigid rubber paddles that spin around at one speed. Only one speed, that's all you get. It's a little bit rough on most species. So I actually only ever use this for rabbit brush. I call it the rabbit brush boss, because there it's one of those long, slender papacy asters that the brush machine can't really handle, and the thresher handles it like a dream. But again, it does have it has limitations in that it is pretty rough. So some of those more sensitive, fragile species, it really can't handle. So it's maybe not the piece of equipment if you're really going for versatility. If you have something where you have to process a lot of rabbit brush, it's definitely the one for you. So in order to try to handle some of these papacy asters, problematic species, we had a Missoula de-winger built. So as the name suggests, the de-winger was originally designed by the Forest Service as a way to de-wing conifer seed. But the Bend Seed Extractory has discovered that it is fabulous at removing the pappus off of those asters. One thing about it though is it's not commercially available like the other two. So the thresher and the brush machine, both of those, you can go to a company, you can say, yes, I want to purchase that, and you can buy it. The Missoula de-winger, you can only get the plans online, and then you have to commission somebody to build it for you. And luckily for us, there was this fabulous department at UNR who they do just that, where you take them the plans and you say, I want this, and they build it for you. So, so they did that. And this has been a pretty recent addition to our facility. And so it has some kinks that need to be worked out. One of them being that drum that you see right there on the de-winger on the bottom. I cannot currently open that. So when it comes time to clean this piece of equipment, it's very, very, very difficult. And seed gets stuck in there. And so it needs some modifications to make it a fully functioning, awesome papacy aster seed conditioner, but we will get there. Next. So sieves. Don't forget about the sieves. So sieves are obviously nothing fancy, but sometimes you need sieves to condition your seed. Sieves, they come in all sorts of different shapes and sizes. They have wire mesh, they have perforated metal. We use all of them to condition some of our seed. Sometimes you need the sieves because your lot is too small and it won't go on any of the equipment. Sometimes you need the sieves because the seed is too fragile and it can't go on any of that equipment. So don't discount the simple things, and sometimes sieves is the ticket. Next. So after you have conditioned your seed, then you can clean it. And for cleaning the seed, which again is separating the seed away from the chaff, we have three pieces of equipment. We have an office clipper, a vibratory separator, and a continuous seed blower. Next, please. So how many of you have heard of, seen, or used an office clipper? I think, yeah, most of us have, right? Like it's a pretty common one, probably the most common piece of seed cleaning equipment out there. It can be really, really handy because it has these different screen sizes. So again, like our whole goal was to create this versatile seed cleaning area where we could clean lots and lots of different types of species. And the clipper is great for that because you have these different screens that you can use to accommodate that. And we have over 100 different screen sizes. And essentially you just put the screens, you can either stack them or use one. I don't have a pointer. You guys can see it. Screen's right there. You stack the screens and the machine vibrates, and it will vibrate the material down. The material will hit an air column, and then it will get separated into 3 different locations, with the heaviest material falling down towards the bottom, which is hopefully your seed. Not always does it happen that way, but ideally that is what's supposed to happen. So again, this one is great because it has all these different screen sizes, so it makes it really versatile. However, again, designed for ag. So it's designed for big, fat, nice, uniform seed. Most of our seeds are not that. They are teeny tiny little seeds, and they get stuck in every nook and cranny on this thing, and there are many. So I never put anything that is a precious seed lot on the clipper because my seed ends up everywhere, and that makes me sad. So its limitation is that it doesn't handle precious seed very well. Next. So we also have this vibratory separator, and it's been what? About 5 years that we've had this vibratory separator, and I have not figured out its claim to fame yet. It drives me nuts. I'm pretty sure it's small round seed, and if you just get the settings just right, it will separate things perfectly. But I can't make it do that. And we have other pieces of equipment that seem to handle that small round seed just fine too. So maybe one day somebody will find this one's claim to fame, but I have not. So its limitation is that it's too complicated to use, it's too small, and there are other things that are better. Next. So we also have this continuous seed blower. So this is absolutely one of my favorite pieces of equipment that we have. And it's really, really simple. It's basically just this machine that sends an airflow column out where you can control the total air velocity. And it's really, really great because you can't mess anything up. So you put the seed in, and seed either comes out the top or the bottom. All of your material either comes out the top or the bottom. So if you don't nail it the first time, it doesn't matter. You can just start all over. There's no seed loss. Which makes it really, really, really appealing and easy to use because you're not losing seed every mistake you make. This one is absolutely fabulous. And it also, if you get these metal tins for the seed to go into, when the seed actually does fall down, it will make this little tinging noise when the seed lands in there. And it's like music to a seed cleaner's ears. It sounds so fabulous. And I apologize for not having a recording of it because I'm sure you would all totally appreciate it. Next. Sieves again. So again, don't discredit the sieves. They're super simple, but they are incredibly, incredibly handy, highly versatile piece of seed cleaning and conditioning equipment. You can see in this picture right there, though, that we have all of this black tape around the sides of our sieves. So again, everything is developed for agricultural purposes. These perforated metal sieves, they have this crazy lip around the inside of them where all of our native seed likes to get, not all of it, but a large majority of our native seed likes to get stuck in those little lips. So we had this big project we were working on, and our quick and dirty fix was to cover that lip with electrical tape. And it worked pretty well. So always having to modify things to make it work for native seed. Next. So I did want to talk a little bit about busting some common misconceptions about seed cleaning. And I know that these misconceptions are out there because I had them before I started cleaning seed. So this whole idea that if you collect undesired seed in your seed collection, that it can be easily removed later. No, that's false. It cannot be easily removed later. It's actually quite difficult to remove that undesired seed after it has been collected and is with that seed lot, especially if it is a similar shape and size. It is very difficult, if not impossible. Think Medusa head and squirrel tail. That is a nightmare. Total nightmare. Like, you're not getting all of that Medusa head out of that squirrel tail collection. And in the process of trying to get it all off of there, it's extremely costly. It takes a lot of time to try to remove all of that stuff off of there. You're also going to have a huge loss of your desired seed in trying to remove that unwanted seed out of there. And you also risk spreading noxious or invasive species. So, you know, when you are out there and you're making these collections, it's really important to try to make sure that you are just collecting the targeted species, because it is in fact difficult to remove those unwanted species later. Next. The next misconception is that it's really easy to just remove that chaff and extraneous material during the seed cleaning process. Well, that's not true. It's actually quite difficult to remove all of that extra material. So when you go out there and you're collecting seed and you get all this extra stem because you're like, well, I'm not really sure what's mature, I'm just going to grab everything. And I know, because I've done this. And this is how I learned that it was not actually easy to remove these things. And again, it's costly. So it takes a lot of extra time to try to remove that extra material out from your seed. And then again, you have a loss of the desired seed, your target seed. You will lose a whole bunch of that, your precious seed that you spent so much time collecting, when you collect all of this extra material. This picture down here on the left has been dubbed the fuzzy chicken. So that, for some reason, a stem was made in this seed collection. I'm not really sure why the stem was in there, but all of the seeds stuck to that stem. And it took us forever and a day to remove that seed from the fuzzy chicken stem. So just to drive home the point that it takes a long time to try to separate those things out. Next. Okay, so that is kind of seed conditioning and cleaning. We're going to talk a little bit about testing. We have a very, very, very rudimentary testing situation set up. So we will do cut tests. And cut tests are really, really simple. Essentially, you take your seed, you take a razor blade, you cut your seed in half, and look, is there endosperm in there or is there not? Does the endosperm fill the entire seed coat or does it not? You know, when Jackson was up here and he was talking about these like wildly different results from these different seed testing areas, and I'm thinking, yeah, because there is some like subjectivity to this where you're like, yeah, I think that one's filled. So I get it. It can be very, very difficult to try to see these things sometimes. But this has a great picture of the cut test. We're on Cercocarpus ledifolius, mountain mahogany, where you could see the one that's filled. It's got the really nice endosperm totally filled. That little buddy was viable. And then the one that was absolutely not filled, you can see it's totally hollow. There's nothing in there. That one obviously is not filled. And these were, you never would have known this just by holding those two in your hand. You really had to do the cut test to know. We will also do purity tests by weight, where we essentially just separate the chaff out from the seed and get a total weight and figure out how much of the material we have is actually seed. We also do some germination tests. So we have this fun piece of equipment. It's from Seedborough. It's literally called the germinator. And it's great because it allows you to control temperature, light, and moisture. And so you can give the seeds the conditions that they prefer to try to force germination, so you can kind of get an idea of viability. And then we also have an X-ray machine. So our X-ray machine is not the best. I don't know how many of you have ever had X-ray photos come back from the Ben seed extractory, but their X-ray machine is fabulous. Ours is not that. However, it does give us an idea on some of these larger seeds to be able to see what is viable and what is not, and be able to do this by sampling non-destructively. So with all of these other tests, either the germination or the cut test, to get an idea of viability, you have to destroy that seed. And that can be incredibly painful sometimes, especially if you have this really, really, really precious seed lot. But with the X-ray machine, you can put it on the X-ray machine, you can get an idea of viability, and you don't have to destroy that seed. It doesn't work for all of the species, but as you can see on the super bright picture right down there on the right, that is Persia tridentata, and you can really get an idea of what seed is viable and what seed is not from being compromised by insects. So the X-ray machine can be very, very handy. Next. Okay, so we also have the ability to store seed. So when you're thinking of seed storage, you're really kind of thinking of two different things potentially. There's short-term, which is typically less than 18 months, and there's also long-term storage, which is anything greater than 18 months. And long-term storage can even be up to 20 years, you know, so sometimes this long-term storage is very, very long, like bank, think seed bank. Next, please. So short-term storage, historically, it was thought that you should be storing the seed between 33 and 41 degrees Fahrenheit. There has been some new recommendations that have come out in the last few years, and we'll talk about that a little bit in a minute. And for this, we have two commercial reach-in refrigerators. They're about 70 cubic foot each. And these temps are pretty variable because they are just commercial refrigerators. But they usually range anywhere between 34 and 45 degrees Fahrenheit. Some of the things we learned about storing seed in a commercial refrigerator is that on some of your very, very dusty species like Atriplex canescens, you're going to want to double bag it because otherwise it makes your unit upset. Ours was leaking green goo. So double bagging can be very, very helpful. Next. Long-term storage is a little bit different. It requires an extra step. So for long-term storage, you need to make sure that the moisture content of your seed is 15% or lower, because you're going to be sticking the seed into 0 degrees Fahrenheit. And if the moisture content of that seed is too high, when you stick it in there, you're going to burst your seed. And then everything you work so hard to do is all for naught. So prior to sticking your seed into 0 degrees Fahrenheit, you need to test the moisture levels. And for that, we use a Rotronic moisture analyzer. It is essentially just this fancy piece of equipment that you put your seed in this little chamber. It has a sensor and it spits you out a reading and lets you know the moisture content of your seed. This one is pretty fancy. You really don't need anything that fancy to accomplish this though. You can accomplish this by making a little moisture chamber and using a hygrometer to get an idea of the moisture level of your seed before you stick it into the freezer. Next. Okay, so in 2020, some new guidance came out. There was this paper titled The International Principles and Standards for Native Seeds and Ecological Restoration that came out by Simone Pedrini and Kingsley Dixon. I believe they are both out of Australia. And this paper is kind of a big deal because it was the first in its kind where it really offered this international standard of how do we deal with native seed throughout the entire process. And it recognized that we can't have the same standards as agriculture because we're actually like trying to do totally opposite things, right? Like agriculture breed uniformity into everything. We are trying to preserve that variation. So this paper is kind of a big deal. It did kind of mess with some things though a little bit. So for storing seed, they recommend that you store seed at 15 degrees Celsius for the short term. For those of us that are metrically challenged, that's about 59 degrees Fahrenheit. That is a lot warmer than the previous recommendation. And commercial refrigerators do not go that high. I don't know if most cold storage units actually go that high, short of just having a conditioned room. So we thought we would try something else out to see if we could find a space where we could actually store our short-term seed in 59 degrees Fahrenheit. And it just so happens UNR has this fabulous wine storage area. UNR used to have a really big wine program that's no longer up and running, but there was this wine storage area where you had total control of the temperature and the humidity was quite lovely in there. And so we were like, yes, let's put some racks in there. Let's stick the seed in there. Let's see how this goes. And it went great for a little while. It was a communal space. We all know how communal spaces go. And somebody decided they were going to grow potatoes in there. And so the humidity, I walked in one day and the humidity was through the roof. There had actually been a leak. It was leaking right onto one of our bags of seed. And then if somebody could click. Next, there it is. So we had some seed that actually got ruined because right where the leak was and the humidity up, these little buddies decided it was time for them to go, and a bunch of them sprouted. Needless to say, we have removed the seed from that area, and we'll try to find different places that can accommodate our temperature and humidity needs a little bit better. I guess at least we know that seed is still viable. Another important lesson is to check your stuff frequently. So if I had been checking this area a little bit more often, it would not have ended up like this. But it's easy to set and forget, right? Next. Okay, so when you're cleaning seed and when you're storing seed, it's very, very, very important to have labels and an inventory system. So on these labels, you really need to have what species it is, who owns it is incredibly important, when it was collected, what seed lot it is, the tests that were performed, the estimated PLS in there. Honestly, as much information as you can get on those seed tags, the better. As we all know, there's turnover in places, names change, contacts change, and it can be incredibly difficult to track these things down later. And just the more information you have with that seed that's just going to live with that seed and go with that seed wherever it goes, the better. We also learned the hard way that you really, really need an inventory system so that the seed does not end up in the wrong person's hands. So any kind of inventory system you can get, whether it's just a spreadsheet or if you're using, you know, barcodes, whatever, just some way to make sure that you're keeping track of that is important. Next. Okay, so something else you definitely want to think about when you are cleaning or storing seed is pests. So rodents, insect, and disease can all be an extreme headache. Fortunately, we have not had huge problems with any of these things. The most we've had trouble with is rodents. My biggest thing is always keep it clean. The cleaner you keep it, the less likely you are going to have any type of pest problem at all. With the seed, not only does that mean keeping the area clean and swept up and stray seeds off the ground, but also keeping everything up off the ground when you're storing it. The one time we did actually have a problem, it was because it was, again, that communal space, and somebody else decided to leave their very tasty experiment out, and a bunch of rodents moved in because they thought, you know, it was the buffet. And then they kind of migrated over into our area, and we kept trying to set traps and keep them out. But honestly, the best thing was to just clean up that area, which of course was easier said than done, but we did finally get it clean and we no longer have a rodent problem. I think Ed Kleiner with Comstock, you probably have more insight into dealing with rodents. Is there something you would like to add?
You know, we don't have too many problems, but one thing we did was build a rodent-proof shed for storage of items that are targeted a lot.
Okay.
And that's been really handy, but it's got to be ventilated and insulated.
Okay.
And a lot of species, you have to think whether they're going to eat it or live in it. And so that's a seasonal issue. Suddenly run it.
Okay. For those of you online, Ed Kleiner suggested building a rodent-proof shed and thinking of seasonality and whether they're going to eat it or live in it. Thank you, Ed. For insects, so insects can be a problem, but typically insects are a problem when they come in on the seed. So if you use a pest strip when you collect the seed and then after it gets cleaned and processed, typically insects aren't too much of a problem if measures have been taken to deal with the insects before they actually come in. And then disease, the biggest thing with disease is to make sure that you are keeping and storing your seed in the right conditions. So make sure the humidity is at a lower level, make sure that the temperature is the way that it's supposed to be, and just make sure you're not creating conditions that are conducive for disease to proliferate. Next. Okay, so some lessons learned. We learned the hard way that your cold storage units really, really, really need a monitoring system. So we had this very unfortunate event where we were storing seed in one of our long-term freezers for this project that was like a $2 million grant, and one of our pieces of equipment malfunctioned, and I don't know how it happens, but our freezer went from being 0 degrees Fahrenheit to 115 degrees. And we really had no idea how long it had been there. We don't know if it was there for a few hours, if it was there for a few days. This was a part-time gig for me, and I was not in here every day checking everything. And it was quite scary. It was a very large grant that seed was being stored for, and we were very worried that we killed the seed. Luckily for us, it was conifer seed, and we sent it off for TZ testing, and it was okay. But had it been anything other than conifer seed, it probably would not have been okay. And so from that, we actually installed these monitoring systems that will alert us remotely if the temperature goes out of range. So if any one of my units right now goes out of range, I'm going to get a text. I'm going to get an email. I'm going to get a phone call. And if I don't answer, it's going to start texting somebody else. So I don't think anything quite that fancy is needed. That was just our kind of reactionary, oh my gosh, this very terrible thing just happened to us. But something that can alert you, especially remotely, if your system is out of range. We also learned that an inventory system is needed, especially indicating who owns the seed, so you don't accidentally put the seed in the wrong person's hands. It's actually quite easy to do. It sounds like it wouldn't be, but it can be. You know, you got a lot of seeds stored in there. Somebody asks for the seed and you're like, oh yeah, let me get you your seed. Oh, well, that person actually didn't own that seed. So it's a good idea to have something in place to prevent those types of things. We have also learned that when you're shipping seed, you should always send a tracking number. Even if that person on the other end doesn't want that tracking number, always send the tracking number. And I don't have it up here, but also very, very important is to make sure that you're checking on your seed frequently, especially that stuff in storage, whether it's cold storage or long-term storage. Just make sure you're keeping an eye on it because all sorts of things can go wrong. Next. Okay, so since 2017, we have cleaned 69 different species, which was totally what we were going for, right? We wanted to be this boutique-y kind of seed cleaning area where we could clean all the different species. We've cleaned grasses, shrubs, and forbs, and we've cleaned over 280 different lots to date.
Hi, Nick. Hold on a second. I'm in a conference, so I'm trying to mute that. No, you're fine.
Thank you for muting. So with cleaning all of these 280 lots, sometimes you have to get pretty creative. Like I said, everything's created for ag. So you have to modify things. And sometimes you really, really have to think outside the box. Don't discount common household items like rolling pins and paint pans. We've used all of these things to clean the seed. Sometimes the pieces of equipment just don't do the trick. And so you really, really do have to get creative. And also don't forget your PPE. Sometimes you have to be in a full Tyvek suit and a mask and go through several dust masks, and you still go home sneezing and eyes running and incredibly itchy. This fun little getup was for cleaning Amsynchia, which is probably about the worst thing I've ever cleaned. Next. Okay, so I would again like to give a special shout out to the Ben Seed Extractory. Could not have done this without them. They were wonderful throughout the whole process. In fact, they're still wonderful when we contact them with questions. And then also the National Seed Lab and Lucky Peak Nursery. If you have questions about the UNR Processing Center, please reach out to Beth Ledger at UNR. Her office number is up there as well as her email. And I guess we're holding questions. Yeah, Leanna. Perfect. Thank you.
All right. So we have one more presenter, and then we're going to come back to the panel questions and answers. So our next presenter is Ed Kleiner, and he's going to be speaking to us about secondary benefits of seed. So do you want to have your presentation up here, or is it just being able to see?
Sure.
Okay. Do you have notes with yours, or do you want to?
No.
Okay. I'll just have it so that the presentation is up. So I have to share my screen. Do I need to share my screen, or Jess has it? Okay. All right, perfect. So does that work for you?
That's
Okay. All right. So Ed is, or I'll give you a brief introduction, sorry. Following an arts degree in economics at UNR, Ed opened his seed company 40 years ago. As a company president, he has worked in most sectors of the reclamation industry, from traditional mining venues to new age solar projects, focusing on supplying local genetic source seed for seed projects in the Western Great Basin. He has supplied seed from or for the Carson City Freeway, the I-580 corridor, Southwest Intertie project, the Tuscarora pipeline, as well as multiple projects in the Lake Tahoe Basin and Truckee Meadows. For the last 21 years, he's been applying much of his experience to developing his 43-acre seed farm in Gardnerville. So thanks, Ed, appreciate it. And you'll be able to see your presentations and then just signal to Jess. So she
All
can
right,
change
we'll
it.
do. It's been 2 years, folks, since I stood in front of a crowd. Holy cow, I've missed you all. For a while, it was fun being in your pajamas and talking to an anonymous 100 people. But when I had a chance today to either do that or jump in my truck, thank you, Stephen, I jumped in my truck. So here we are. Stephen has asked me to put on a different hat and talk about the support structures on our farm. Our farm has a reputation for doing all sorts of progressive behavior. But before I talk to you about all of that, it's very important to say that the farm and the seed company feed off of each other. One doesn't exist in a void without the other, and the synergy is what makes our operation so smooth over the years. And one other point on this synergy or this connectivity is that our relationship to the market sectors that we work in, the local farmers, the mines, the highways, the agencies, all of these groups, we know people on a first name basis. We are in their face. Somebody retires, somebody's replaced, we are in their face. We attend conferences such as these. We're going to have a booth at Earth Day. We go to kids shows, farm shows, but it's that constant activity that keeps us in all of our market sectors and keeps the operation going. So between all three forms of connectivity, that's what's most important. But today, the bulk of what we're going to talk about is on our farm. Next slide, please. There you have it in a nutshell. Humbly, the next slide is going to be about who pays the bills, how the bills are paid. But you can see down the line there that we're very involved with some NRCS programming on the farm. We embrace any opportunity to try something new, to open up to everybody in the area to see how it works and what happens. And we all get to learn about soil quality, improving pollinator habitat, the need for hedgerows, everything on a farm that will help push the program forward. Down below that, you can see the portion on infrastructure improvements. This is where we really shined. We have some very progressive constructed wetland blackwater sewer systems on the farm. That means 2 of them. We have a hoop house that came with an NRCS program that we've got to experiment with extending our growing season. Earlier on, Justin had mentioned the extreme temperature fluctuations we have when we're working with seed, and it's the same with growing farm crops. So from the data gained from that hoop house, we know that we have a little extended growing season where we get a lot of our food off, but that's not on a commercial basis. That's just on our farm for sustaining ourselves. And a lot of food in surplus will go to places like the food banks or our employees. Everybody gets to share in the wealth there. And last, the solar system. Back in 2009, we were a fairly unique and early operation in establishing 9 kW of solar on our farm. We married a USDA rural development grant for Douglas County with a rebate program through NV Energy that allowed us to afford putting a solar system on the farm that costs less than half as much today. And we plan on adding a little more even yet. So with that, let's move on to the next slide. It's a little messy looking, but at this same form in 2018, the white font, I showed this Google slide of our farm and what at that time we were producing on the farm and the money gained from it. For this presentation, I added the red overlay, which shows you the NRCS programs that are active on the farm right now. But there's something wrong with this picture. If you add up the numbers, the dollars on those seeds, it's a pittance. The hay on 28 acres, we only do 1 cutting a year because we are not active in cattle, hay, and rotation. And so we have a lean nutrient supply on that land. So a whopping $6,000 to $7,000 a year in hay off of 23 acres. And then down below, the other significant contribution to our financial had been a 3-acre experimental row in Great Basin wild rye, sulfur buckwheat, a few penstemons. And so you're looking at a whopping total of $15,000 in money coming off the farm. That's the gross. Our mortgage, 43 acres, $500,000. Think 30 years, $3,000 a month just for the mortgage. Obviously, the farm is not paying that bill, but the farm seed company synergy is. So we form a seed company corporation. We pay rental income to the farm. That is where the bulk of the mortgage money has come from over the years, as well as having a lot of seed sales, which in some years would be very lean. And in those lean years, when there isn't as much as cash flow through most of the year, it was essential to have these infrastructure developments on our farm. Once we had solar on the farm, we went from $300 a month in electrical utilities to $15 a month, $20 a month. And as our farm has grown, our need for more electrical consumption has gone up. So thus we're also adding more solar to the farm soon. So there you have it in a nutshell. It's the relationship that counts. Next. So there's a summary. You can see their average of $15,000 a year for all those years. And some years were much leaner than others. Next. This is a list of the NRCS programs that if I pulled out of a manual. The trouble in the manual is that you get the number and then you get this long description. So I abbreviated the descriptions of the kinds of programs that were available through our office that I felt like working with. Lots of habitat, lots of pollinator stuff, lots of soil related stuff. And the red asterisks is on the left side are the 5 programs that we're engaged in. But these folks are worth spending your time with because they will help you develop all sorts of activities that are very productive for the farm. And the showcase ability of that, we get a lot of visiting on the farm. And so that's marketing PR for the farm. And whenever we go out and do anything related to the farm and have activities and conferences at the farm, it all promotes marketing towards the sales of the seed. Next. So let's jump into a few of these. Early on, we did a 3-year green manure farm here. This was a fallow area between my Great Basin wild rye and my vineyard. I have 300 wine grapes on the left, Great Basin on the right. It was an area that we had never used on the farm, heavily compacted clay soils. Our farm for 80 or 90 years had been cattle walking and machines driving. And so once when we tried to put a drill onto the surface of the ground, it could not penetrate the surface of the farm between the organic production in the soil and the compaction of the heavy clay to even put seed in the ground. We had to call in a Great Plains drill and add a couple thousand pounds of the weight to penetrate the soil. So here was a perfect case in point. Our connectivity in the valley, also the NRCS knows we're there. We're willing victims, and I will call them. And they said, well, let's put in a green manure experiment. So they came in and we took this area, we did a light tilling in the spring, and we added the seed blend above is all annuals, a couple grains or ryegrass and a couple annual legumes. And we broadcast that onto the surface of a lightly tilled soil. Then we dropped our rototiller barely touching the surface, a penetration of about 1 inch, and spread the seed into the soil that way. And then this area got its normal flood irrigation starting April 1st, coming up here, which every other week we could flood it if we wanted to. And so fairly early on, we grew this nice crop. Next slide, please. So by fall of that year, this is what you're looking at. We had lean production of the 2 legumes. We had a lot of grain and annual rye come up. And the blonde areas on the upper left of the photograph are some invading cheatgrass. We had a little mallow coming in. And to the green manure program, the NRCS says, well, we don't want you disturbing the place much, but we were allowed to go weed eat annual weeds that we didn't want to go to seed. We have a 3-year program here. But what we also were told to do was each year to add more seed in the fall. The idea is to grow roots into that soil, create organic material, nodulate nitrogen with the legumes. And so we put another 9 pounds of seed onto this farm late fall, right on top of this residual plant growth. And that included the same blend as before, but we took out the oats and we added some wild flowers. And then we dropped a rototiller to the brush hog to the ground and just mowed this flat and let winter take over. All right, next slide. And there you have it. The next spring, all the legumes took over. You're looking at all-cycle clover and hairy vetch. And the lower right slide, a little later in the season, up came the grain and the annual right through it. but look at the massive amount of production on that poor soil that this created. We love this slide, but one of the things we already knew, we've done a lot of restoration work. A lot of our species don't respond in first year. It's nice to have nurse crops, annuals and grains, and anything native that you can put into a seed blend. It's going to grow easier under harsh conditions year 1, year 2. But it's here we have a second year case where all the legumes took over. Even on some of our farm jobs, the clients will call and say, we aren't seeing much of the white Dutch clover, whatever the legume or the broadleaf might be that's in a seed blend. The next year they call back and say, we have way too much clover. All right. And so we tweak our seed blends for that reason, and we lighten up on the clover, even though we know first year we may not see the clover much at all. And here is a case where we could demonstrate that's what happens. You can see the irrigation down on the lower left. We were quite able to flood irrigate once that we have our surface rights running in April. Next. And of course, with anything NRCS, they want to study the soil in this case. So on the left, we had James Comar, Cornell University, soil sampling before we put in the green manure. And then the test on the right, I pulled out of the soil 3 years later, all right? And my one dilemma with this is we have 2 different formatted seed tests. One is a quality analysis that was done by Cornell, which is lovely. It's 6-7 pages of descriptions of soil. The reds were the low quality areas, the hard pan, the less nutrient flow and an unproductive soil, basically. On the right side, you got more of an analysis viewpoint. The only 2 things in common here, pH started out in the first one a little low, 5.3, ended up on the second one, 7.3. So we bounced between on that one. The organic material did not change. It stayed constant between the 2. But if you go out in the field and you stood there, and James sort of confirmed this, it was a beautiful soil, second year or third year, because we put another project on top of this. Next slide. Here is the beginning of a pollinator experiment. Once we had done the green manure treatment, we went right back in there. The field on the left shows what the field looked like after laying fallow for 2 seasons after the green manure, right up until December of last year. And the close-up down below on the left shows you what you're looking at that's green. It's all weeds. And we had fall rains that germinated annual weeds. Solid, fillery, mallow, cheatgrass came up. And when you have these young annuals in the soil, it is so easy to remove them by a chisel harrow, drag it. And a lot of farmers will do that even in the spring on new crops. Once they get a farm crop up and running, they'll notice the amount of broadleaf weeds that are in there and the annuals, and they'll just drag the field lightly, not tearing up the perennial root systems that are in and not doing this kind of treatment too early in the game. But in the fall last year, we get a lot of rain early, and then we might have Indian summer fall, and you get all the stuff germinated. You can go out and nuke this chemically if you want, but it was so easy to drag it and create the field on the right, just uprooted all the sprouts. And so we eliminated them and we seeded into that field the next slide. In this case, it's not cheap grain, it's expensive flower seed. This is a pollinator habitat. That means we seeded in thin leaf milkweed, showy milkweed, and several other flower species, penstemons, lupins, buckwheats. It's your dream come true for a bunch of pollinators, all right? And so we got all of that seed. After we had dragged that field, we got it broadcast out there. We re-dragged it once more, and there it sat. A day after we got it all in, the snows hit last December. It was the perfect timing. And we tell a lot of folks to take advantage of that fall window for seeding. Even with agricultural crops, for some of we put as much pasture down late fall as we do in early spring, because it's already up and running now instead of waiting for the first April surface right irrigation to be coming in. But we get these things in just before winter starts in that window of opportunity, and preferably with the weed abatement that we were allowed to do as well. But so this field, we let it sit there, and came this spring. Well, guess what? Record dry January, February, no precip. We had record February sales this year that we've never seen before, because half the clients were thinking, a late window in the fall they missed. Here they are in February putting it down. And the other half were thinking, it's early spring. Let's get all the seed in with it while the weather's mild and it's still cool, expecting spring precip to come back. we haven't had snow and rain that was worth a hill of beans since. And so all these disturbed fields with seeds sitting down, and do I sleep at night? Not much. I'm hoping for the best here. But so come March, I said, well, phooey, we didn't have our surface rights yet, but I turned on my ag well. I got 300 gallons a minute. I ran it down the fields and flooded that puppy and cemented it all down too. A lot of these loose soils with seeds in them are quite vulnerable to high winds. And so we've irrigated it once. Stay tuned. Irrigation is coming on April 1st, and I'm hoping to see something in that field sprouting. It's all broadleaf flowers though. So what do you do if a bunch of broadleaf weeds come up? Not much. A few years ago, we put in an experimental legumes, native dahlias and astragalus. And we were so worried about the broadleaf weed competition in the spring that we put a pre-emergent down over those experimental transplants. And we proceeded to kill every single one of them. Shallow rooted transplanted supercells, hundreds of them. The time we spent putting them in, it was a program with the ARS. And sure, we have crash and burns, and this was a learning curve. Don't use pre-emergence until the roots are deep enough to survive the chemicals you're putting on top. Even better, try to use less chemicals anyway. Next slide. All right, let's move on. Our solar program started in 2009. Back to the idea of connectivity. I knew a guy at the USDA Rural Development Agency up in Carson City, Herb Shedd, good friend of mine. And he said to me back in 2009, why don't you guys go for a solar grant? Douglas County qualifies for rural development grants. And we said, sure. And so we put in an application to put in solar, and we were approved for 25% of the cost of the system from the USDA. Right behind it, we went to NV Energy, who had a rebate program back then. They also approved 25% of the cost of the system to go in. And so here we had 50% of the cost of the system up front to be paid for by these two programs. We were required to hire a contractor approved by the agencies. And so we called the fella up, and he came along and said, yeah, I can put the system in for you. He gave us a price, and he said he could get through the grant programs and the paperwork. And a month later, we hadn't heard from him. He called back and he said, well, I can put the system in, but I can't help you with the paperwork. Well, thanks anyway. So at that moment, we had to cancel putting in the solar system that year and wait till the following year. Well, once again, connectivity came along. We're out doing a booth over at Bentley Ag for a spring forum. And along comes Leslie Ames from Tahoe Solar Design, walks up to our booth. And I'm telling her the sob story that we missed putting in solar last year because we couldn't get the paperwork complete. And she says, well, silly, Ed, that's simple. And she says, I got all the software in the computer. We put the numbers in, they spit out the applications, and we're done. I said, give me a quote. Next slide, please. Oh, that was that last slide. Leslie was busy putting that system in. Here's some nuts and bolts on how the system came out. There's Tahoe's quote. It was $82,000 for 9 kilowatts of power. It's less than half that now. You can see the numbers down the top. Rural development grant, 20 grand. The rebate, 21 grand. We augured all the holes for the contractors. We had to fork out 38,000 cash towards the solar system. But mind you, we're putting in a 25-year warranted system that's going to pay all our electricity for the farm and the home, the office. Right? So this was a worthy investment to us. Down below there, then it gets a little more exciting. The annual depreciation deduction for an $80,000 system was $16,000 a year. We got our application through the business instead of a personal. The USDA recommended we go for a business grant instead of a personal because they were much less competitive. Thus, it was easier to get the award. Well, so the business, if it had profits, here's a $16,000 annual deduction for 5 years. For a few of those years, we did actually have some profit, so we couldn't take the deduction, but there wasn't a time element on that. Right below that, one-time federal deduction of $9,000, bingo. And then it was a surprise in the end. We had surplus solar. Well, we started getting rid of propane appliances and putting in electrical appliances. We saved $1,000 a year in propane bills. All right. What this did was push the payback period forward for us. So we get the system paid off in 9 or 10 years on a 25-year warranted system. In the end, all said and done, we leveraged $18,000 for an $80,000 system. All right. It would be nice if you could buy this system on a free market basis and pay for it. But this was back then. It's gotten much better, but at the same time, the lower the cost of the system, the lower the rebates, but the percentages were the same today. There's not as much available on the rebate programs anymore though, but solar in general is good. We want to reduce our carbon footprint and be part of the future of saving this planet. Next slide, please. Oh, excuse me, hold on, sorry. Down there on the right was our first month of performance records. We keep our own records. We watch NV Energy's billing to make sure there's some comparison between the two. We averaged 45 kilowatt hours per day in November. Our farm consumption all was about 45 kilowatt hours for the month. So it was producing exactly what we needed. So we knew we were on the way, but there's one cloud on the horizon. As you get down there towards the bottom, you notice clouds, cloudy days. Our performance plunged during cloudy days. And during the winter, our performance goes way down. We get much better performance in the winter because the panels are being cleaned by stormy weather, freezing, dew. They were cleansing the panels all the time, which is our only maintenance bill, is keeping the bird dew off the panels and the dust, right? But at the same time, spring is really clean air. Usually there's not as much dust. Winters are slower, but we get a surplus production during the sunny months, spring and fall in particular, and we get reduced deficit performance in the winter. But when you average those two together over a year, the system was perfectly matched for us. We've seen a lot of people that overbuilt systems that they didn't need, and they couldn't necessarily get refunded on the power. You can do better at that today with net metering systems, and you can do better in groups of cooperatives, small housing developments, groups of people pooling. There's a lot of new ways to go about using solar and getting credits for it without even owning the systems. A lot of creative activity going on with financing these systems. Next. And here's another a connectivity side of things. We got all done, and Dan Caffer from the Nevada Resource Conservation Development said, we're going to have a party. You are the first people to manage these grant programs. We are going to have a party on your farm. And we had a local vor feast. And all these folks that produce food locally came to our farm for a massive party. We had over 100 people in attendance. We had representatives from the governor's office, the senator's office. We had people speaking, handing out fake big checks. It was a marketing bonanza for a seed company. It painted a picture for us locally as the go-to people. We are part of the future. This was such a touchy-feely, fun event that it sort of addicted us to being part of the community in that respect. Even though it's more of a local affair, our sales are more regional in nature. We're Great Basin Sierras and Mojaves in what we sell, yet we are rock solid locally. And the energy from what we've been doing locally branches out and helps everybody. It was fun, all the people that showed up. Next. All right, our sewer systems. This is still progressive today, still not being activated on by more people. But being in the reclamation business, I always knew there was repair and restoration going on. I always knew that there was constructed wetlands being used for acid mine mitigation, shoreline structural support, riparian developments where you have to armor banks, lots of constructed wetland work. And we knew that the constructed wetlands we found through the literature were active for human sewage. And one of the earlier pioneers was the Tennessee Valley Authority. Where you don't have a high density of humans, you have a very high cost of sewer treatment through the piping and the infrastructure to bring people together. So there's lots of isolated players, all these rural people living out there. And when the TVA developed a system and built several thousand of them in the Tennessee Valley watershed and fine-tuned them for later operations, we solved the schematics and the design of that system that you could have black water sewer treatment on your farm and a subterranean fed system, hydroponic system within a lined membrane that doesn't contaminate a groundwater, and you can grow pretty plants that uptake your sewer effluent. I said, wow, that sounds too good to be true. And so we took those plans, gave them to a local stamped engineer in the state of Nevada, Dan Jenkins, recently deceased, sorry to say. And we took those to the state, the health department, and they said, well, we've never done one of these for residents before, but what the hell? And so they gave us a permit to go ahead. We took that permit from the state health department, went down to the county to get our building permit, and they looked at it and said, We don't know what this is, but if the state health department says it's okay, stamp. And so we got to build the first residential black water sewer treatment plant in Nevada. And here we set it on our farm. There's a schematic down on the lower right. It's a 2-cell system. One's lined, it's gravity fed from the septic tank in our yard. We live on flat ground in a 100-year floodplain where you usually have to build a pressurized mound system, and you have to use a sump pump to pump your effluent up into this mound to keep it out of the water table. Over time, leach fields and these unlined systems slowly leach down. Within 25 years, fecal coliform bacteria is showing up in your well water. How do you fix that? Okay, think dollars and cents and changes in your lifestyle. For us, the county commission, who knew that we were building this, invited themselves over for a brown bag lunch on my porch deck, and they could sit here and look at this system. And I swear they showed up expecting to see a black cesspool with lots of bugs and things going on. And while they're eating lunch, I walk out in the middle of the thing and stand there and jump up and down and say, look at me. You know, it was subterranean fed. We had control over the level within that system. It never had a vector for an odor. It'll never contaminate the groundwater. And one of the ladies, bless her heart, Nancy McDermott, said, what do you do if Junior pours something down the toilet and kills the wetland? I said, well, it's only 18 inches deep and it's a bunch of gravel. And if that ever happened, we'd scoop it out. But building a system like this, you educate your family or the families that you're sharing these with, that you don't do these things in the soil. You wear dark socks instead of white socks. You get rid of bleach. You do sorts of things that respond to keeping that system alive. And then I looked at her and said, what do you do when the fecal coliform shows up 25 down there? And that opens their eyes. And unfortunately, it allows developers to open their eyes and build in our 100-year floodplains. And so that's an issue there. But on the other hand, the benefits over the cons. So let's see the next slide. There's my farm, Google overhead. The 2 orange red circles are our 2 constructed wetland sewers. The one on the left is the double white house that we've lived in for many years. The one on the right is the solar barn house with a brand new system. The one on the left has been there now 22 years. The state required that the water coming out of the system be tested only in year 1. It turned out to be amazingly good. The plants were uptaking all the nutrients. The biological oxygen demand had fallen off, and all was well, and everybody forgot about the system. It's worked good ever since. The one on the right is brand new. And so people come to the farm, engineering firms have come. They've taken this same design, they've gone back to Reno. You don't have to have a cube of square on the ground that you have this system in. You can build a meandering creek. It's the square footage of the treatment of the system that's required for human flushing toilet in-house. And so that's the design bottom line. You can also see from this photograph our historic dairy farm buildings down there on the lower left that now are our seed company. You can see the experimental farm rows to the left of that. Just north of the houses, you can see the vineyard, which is, I'm going to show you pictures of that. And you can see the 9 acres of wetlands that flow down through the center of our property that we've been enhancing for many years now. Without cattle and farm machinery on the ground, the wetlands have proven to be a bird haven. Ground nesting birds, nothing's getting driven on. The pollinating habitat within the ground that the bees cooperate. We aren't out there doing much of anything. But those are the systems that we are now showcasing. There's only been a handful built, including the Witt Nature Center that the Nature Conservancy put in our valley. They built an identical system to ours, and they showcase the same idea. If I had a lifetime in another career, I would be proselytizing these things. And we should be decentralizing sewage, decentralizing stormwater, getting rid of the cost of concentrating all this stuff. Lake Tahoe's classic example, moving the stormwater drainage up into the watershed, because putting it right down on the shoreline, well, that's where the beach is, the tourism is, everybody's there. And they've done a good job of making some of those sites look pretty as constructed wetland sediment control. Next, please. So here's how you build a system. It's pretty simple. You get out a level, a laser level, you set it on the septic tank, which is the lower right, left picture. You get a qualified contractor to go dig these 2 trenches in. That's the liner there, a 60 mil vinyl liner that's going to be buried underground. You toe in the edges. On the right photo, you can see we're putting the gravel base in. This is a hydroponic garden that's going to be super saturated with a rich sewer effluent. The septic tank is a 2-cell. Sludge in the first, the effluent that's liquid form is what makes it into the system. Next slide. And that's what the finished system looks like, brand new from the roof of the house. We had to run water in it to supercharge it, but we immediately moved in the house, and two parents and 2 kids start taking showers, washing clothes, flushing toilets, doing everything you do. But you can see there's a pipe in the middle of the first cell. That was a monitoring tube for the first year of water quality control. And we planted 200 super cells in there that were grown by NDF, and we transplanted a bunch of cattails in there. And the lower cell in the background is unlined, and it's a lower elevation, so effluent drops even further if it ever makes it out of the first cell. That's the one that also fills up with water during the irrigation season, because our water table is only 18 inches underground. So this is a tight little system in a floodplain, okay? And this system, mind you, 2001 photograph. Next slide. Welcome to 2012, the prettiest sewer in the state. Okay, and that our hoop house on the left, I got to show you that too pretty shortly. And all of there's only one line coming out of our house that flows into that cell. And you can see the cattails essentially took over. When we built the second one, we saved the money and the trouble of going through super cells. We have a private farm. We took our tractor and went out and grabbed chunks of wetland out of our wetlands, had all the microbials, the soil, the plant community, the bugs. We took those over and transplanted those instead into the new system. Next picture. Even prettier, 2014, all right? There's a home sewer system. 400 feet of treatment cell, 4,000 square feet for 2,000 square feet for the whole system, all right? And so these would fit on relatively small parcels of earth, and there's much larger systems around the US. Go to Arcata, California, the estuaries into the ocean coming out of the city, our step-down wetlands that treat sewer treatment. Go to South Lake Tahoe, the Edgewood Golf Course is a stormwater treatment system. People don't realize the ponds right down through that golf course come from a underground, high-tech oil separator under one of the casino parking lots. And all that stormwater from South Lake Tahoe is funneled over to the wetlands in the Edgewood Golf Course before the waters re-enter Lake Tahoe. This stuff is happening before our eyes. Mind you, big difference between stormwater and sewer, okay? Different treatments. Next. All right, our high tunnel, this was an NRCS project. You buy a kit, you put it on the ground, you set it up, and for 3 years, you keep production records. So the food you produce, the pounds you produce, the species, and the dates. And you give all that information to the NRCS in trade for a $1,500 subsidy to build this hoop house. Now, mind you, we live in a place where we've had winds as high as 90 miles per hour. All right, I'm running out of time. Let's see, I'll leave this slide, go to the next one. The system they were allowing us to build would fail. Nothing would hold it to the ground. And our hoop house has been there 13 years now. A lot of them blew away like tumbleweeds because they weren't mounted to the ground. That's what this picture was all about. We knew better than to let that system not be anchored in concrete, more knee braces, all right? But it's been operating good ever since. Next slide. Good food production all season. Netting to keep starlings out. Next. And there's the rewards. We got a whole lot of tomatoes year one, and a whole lot of those tomatoes came after the first frost kills your outdoor gardens. Exactly what the NRCS wanted to know about. Next. Hurry along. Hugelculture. I just saw one of these not long ago when we built one. If you got compacted soils, you go dig a hole, you dump in a whole lot of organic matter, like a lot of Justin's byproduct, bury it, put the soil back over the top of it. And we saw these in some permaculture demonstrations, and we've just tried one out on our own farm. It's already settling because of the decomposition under. But the compacted soils around our constructed wetland sewer was made by those machines that made that system. So we've been having a hard time getting rid of the clay. This did it. Next. I'm running out here quick. Yeah, I won't spend much time here, but we're building a hetero, even though we don't have the program at the NRCS. We aren't waiting for them. We're building it anyway. So we put a pump into our irrigation system, ran water out to our highway because we wanted to keep plants alive out on the highway and get them going. Native plants won't need water for long, but hedgerows are really important. We're building 1,000 feet of it now on Highway 88. Next. Have big parties. This is what connectivity and synergy is all about. We invite everybody to the farm. Next. Again, Eagles and Ag, we have these groups that come to the farm, release hawks, show us eagle owls and tortoises. You know, it's a big happy family. Next. We do hay, but only once a year. We don't feed it, so it's lean, but we sell organic grass grown hay. A lot of it native now, but only once a year and doesn't make much money. Next. Experiments with growing Great Basin wild rye, including a burn. It worked. Our Great Basin wild rye, 6-7 feet tall, just before harvest, lower right. Next. Soils, make sure you study your soil maps. I got a weak area out in my wild rye. I didn't even know it until I looked at the soils map, and it corresponded to the 401 soil there that reaches down into my rows. It's a heavy clay area, and that's what made that area grow less. Roots are having a harder time to get down. Next. We leave the grass up all winter for the wildlife habitat. And then we mow it down a week ago, 2 weeks ago, just as it's starting to green. And it's great for gathering snow in the winter. Look at that. Next. We're about done here. Let's see. Vineyard, I better just save this for later because I could talk about wine grapes a lot. It's been a failure for the most part because the vines have been dying back to the ground. But that's another day. Next slide. This is an NRCS ground cover experiment we're doing with clump fescues, both fine leaf and tall fescue. We wanted to grow a ground cover, but not spread into the grapes. No sod formers. Okay, it's working. Next. And even though we don't have much grapes, we have some grapes. The important part is on the right. No income, just satisfaction. Lots of netting, very important. If they get into the netting, they eat all the grapes. They do. Next. All right, owls keep the habitat for the critters that eat rodents. You want to know what we do about mice? It's not just that rodent shed. You keep your owls happy. We have barn owls, the barn owls habitat upstairs. We have a webcam on it. The owls messed up the wiring, so I don't get to watch them this year, but they're making babies right now. Next. Same with the hawks. Hawks outdoors, owls indoors, one at night, one during the day. Same with the ponds. You keep the bats happy. They live in the wall of the barn. Our little birds. Anyway, stay safe. Next, please. We're out of time. All right. I wasn't supposed to talk too much about seed markets. I'm glad Justin put a slide up, but the fine print in this picture is dizzy. Fire is the big elephant in the room right now. It's messing with the markets in a big way, and the climate change is hurting the production of seed. The chasm between those two trends, it's Armageddon in my mind. How to keep the seed flowing. I'm not seeing as much outside anymore to collect. And the farms are being hit by the droughts in the Northwest last year, cut down on production all the way up into Canada. We're struggling with that. We're doing more with rare seed acquisitions as well for IDIQ programs, custom collections like Justin mentioned. But keep your diversification is what's keeping you stable in this business. And there's enough diversity on this screen that always somebody's busy somewhere, even when somebody's down elsewhere. Next. Are we there yet? We should be to the end. All right, diversity on the farm, same idea. I saw this 25 years ago, this fella growing so many species on 70 acres down in the Rio Grande. He taught me right off the bat, grow a wide variety of things. Next. That was the crash experiment from the ARS I told you about. Don't look, just turn the slide. Everything died, thanks to me. Natural on the farm is the creeping wild rye and the wetlands, which we harvest seed off of. On the row production, the others. So we do some of both. Just keep everything happy on the farm. We're harvesting natural as well. Creeping wild rye, great jobs. Next. All right, we're growing supercells. We're doing a wetland project up with the BlackRock people. Supercells for various projects, including transplants on our own farm. And we found one lady growing some of the native flowers that are more durable for cut flowers. And sometimes I think about that myself when I see my big rows of pretty flowers. But I'm still a seed guy. Next. And you have events like this when all's going well. Pond was going down, the fish were big, tadpoles were big, and they came in with their buddies. Next. Are we done? Oh, solar barn house, last picture. This is bringing in a crowd too, we hope. We're going to be in the Carson Valley Almanac. That means we're going to start giving tours. Those people are going to see that this is a seed farm. It's more marketing, but it's more proselytizing to me. Next. All right, that's the last slide, promise. Don't forget connectivity. All right. If you've got sandhills on your property, you win. All right. Thanks.
You bet.
All right, so we're gonna move on to the panel section. So Ed, you can stay up if you wanna come sit at the table right there. Shannon, and then I don't know, Justin and Jackson, if you both wanna come up or we can move another chair over. And then Scott, I believe I saw that you were still in the participants. So if you wanna be ready by your unmute, we're gonna do a panel section. Unfortunately, you guys are probably not gonna be able to see the panel members on Zoom, but you guys should be able to hear them. So if you guys can't, please put it in the chat, but the audience camera, if we turned it on, then you would probably see the pores in their skin. So I figured you can just get an audio and everybody in Bauer gets a visual. So we have a couple of questions. I'm gonna go through the questions that were in the chat box first, and then we'll come to people in the room. So we'll try to get through these. Again, if your question is answered, if you could throw a thumbs up in Zoom so that we know, and if not, we'll work to connect you with those. And the first two, Scott, if you could be ready with your unmute. I know you answered it in the chat, but there was a question related to, the person said, I'm curious if you do validity testing on every source for all of your pooled sessions, and if so, how much time and cost does it have? And I guess I can actually pull, no, I can't pull the chat. So Scott, if you could answer that verbally so everybody in the room can hear.
Yeah, we do try to test the cytotype of all of our different sources. You can look through the literature and find which species are likely to be problem children. Globe mallow is one that's horrendously complicated because it hybridizes a lot. Some of the other species, there aren't multiple cytotypes to even be concerned about. And this native 4 manual that Nancy Shaw is kind of leading the effort on has that information summarized in it for species that have been written up. But in areas where, or for species where it is a potential concern, we do test the cytotype of all of our different sources. And we've been doing that in-house. Bryce Richardson is a geneticist with our lab, and he's got the machinery to do that. So I don't really have a good estimate of what the pricing on that would be on a common market. But the process is relatively simple. It just requires an experienced technician and the specialized machinery.
Okay, great. Thanks, Scott. And I know that you tried to put the PDF, somebody asked about the link to the thick leaf Penstemon paper that you discussed. And if you could email that to Stephen, we'll make sure that it gets to all the participants after the meeting with all the rest of the resources. So
Okay,
just,
I think I was able to stick a link in there as well.
Okay.
That people might be able to click on, but I can get the PDF as well later on.
awesome, great, thanks. The next question is for Justin. Any reason non-viable seed is thrown out versus sent to green waste and compost? And there were several questions about why you are not composting.
Yeah, there's a lot of reasons for that. First off, we're not talking like with the sagebrush, we're talking like Jackson said, hundreds of thousands, well, I don't know, tens of thousands of pounds anyway. So much chaff. So first off, I have problems. I've had people like call the cops and the fire marshal because they're concerned about terpenes and stuff like that, you know, which isn't actually an issue. And, you know, we've got 10 acres in the middle of a bunch of alfalfa fields and stuff, but it's also on the edge of residential and commercial. So we bought the land around us to kind of have a buffer, because when we lay out all that seed and process it, that creates a lot of problems. We did send it to the green waste until they forbid us from doing it. And also we're in the middle of a super red state where our green waste facility, they had to shut down because even though it was so popular for the composting and everything, the the gentrified neighbors who moved in, who were okay with it originally, got together and sued, and they had money. So the entire green waste got shut down. So it was really outside of our control. It's not something we can go dump on public lands. I mean, I don't think any BLM official or state wildlife person is going to want to see big piles of sagebrush seed, and not to mention some of the seeds, for instance, like even around our own place, even when they used to radiate it, it doesn't kill all the seeds. So all of a sudden, like we're in the middle of the Great Basin, but I've got like desert dandelion, owl's clover and stuff like that popping up around my place. So there's a million different reasons why, but like basically because like even on my personal green waste that I do at my home, they've gotten rid of that. So it's a red state issue really. So it's out of our hands. Like if I could compost it, I would, but we've only got 10 acres. And again, like we'd fill that up in a hurry, and it's more of an alkaline soil. So it would ruin everything and so on and so forth.
Okay, great, thanks. Ed, did you want to make a quick comment?
Yes, we're so fortunate over here because we have commercial composting going on at the prison, Carson City, full circle compost, large volumes. They'll take all they can take there. Plus they're scientific about it. They do it very thoroughly and very well. We also were able to take the Bentley for the last several years, who has one of the largest landholders in our valley, and they were doing composting and keeping it all for themselves. But with Bentley closing down, we're now worried that we're about to lose a source of avenue for us to take our compost to. And early on, we were always taking it free up to Carson City, and now you pay for it a little, but I would rather pay for it than throw it away. So obviously we do that. Free is even better, but we do it. Yeah, progressive here.
All right, great. The next question is, again, for Jackson or Justin, what steps are taken to avoid over harvesting or damage to the wildland plants that can impact future yields?
Well, that's in our best interest. I mean, we've been doing this for like 30 years. So we built a relationship. We know who all the key botanists are and like permit issuers and all the different, state and federal agencies. So we're well aware of what the expectations are. We realize that the plants don't want to be over harvested, that they want to leave a certain amount. And so over the years, people have come out and seen that we don't over harvest it. We want to be able to go back to the same stands, and we don't want to like ruin that relationship. So, I mean, it might be a different deal if it's like a private land situation where we pay them, we go in for 1 year and they don't care about the crop, or they want to like plant something else. So yes, we'll over harvest something like that. And for instance, like all the areas where you see us using like the machinery for some of those flowers and stuff like that, those are areas that we were able to go into that were about to be like, they were about to be commercialized anyway. So yeah, so we try and save the resource when we can. And if we can do something like that, and we realize it's going to be concrete and we can mow it up, we do. But otherwise, yeah, we just have a long history. We know what the expectations are. We don't want to upset people. We want to be able to go back.
Okay, great, thanks. Following up on one of the other questions about over harvest, does Premier Seed communicate with landowners and other seed collectors to keep track of who else may be collecting the same species in that particular area?
That's proprietary.
Yeah, well, generally, we have private landowners where we pay them, so it's an exclusive permit. And then we have some areas where we like share it with different seed companies. We try not to do that because the truth of the matter is that like in Utah, there's a lot of different seed collectors. And so there's several times like someone might have a business card for me or something, and they'll get in trouble and they'll like present this business card, but they don't work for me. Like, I don't even know who this guy is. Somebody gave him a card, this and that. All of a sudden he's my employee. He's not. We have our own crews that we've been using. Like we have our core crew that we've been using for the last like 15 years, that they come up on a seasonal basis and then they leave. They're well, they know what all the expectations are. So we keep track of all that. So yeah, personally we do that, but if it can bleed over, not all the companies, not everybody has the same, like, I don't know, I guess you call it moral compass or realize what the rules are. And so sometimes like there's people that go in the middle of the night, for instance, and steal crops, or we've been just waiting for like, a certain stand to ripen. And a lot of these BLM people could tell you that, and they go in the middle of the night and like cut it all down. So there's only so much that we can do, but we try to do what we can to mitigate any of those kind of problems.
Okay, Thanks, Ed, you wanted to.
I was gonna say,
sorry.
the state of Utah is the only permitting agency that I know that's government owned that will allow multiple people to have the same permit for the same seed in the same area. The BLM does exclusive permitting, so if something happened in this one specific area and you had the permit, you can't blame it on that guy without some real good evidence that they came into your land and did something. State of Utah's state land it'd be much more difficult. Sometimes we get out there and there's two other companies and we're all pointing the gun at each other, like you're on our land, and we all show each other the same permit. So yeah, that's it.
Well, just to add to that though, like Sitla, the state trust lands, like their mission is to make money. So they're gonna make money no matter what. Like, so it's in their best interest to over permit it because they don't really care. Whatever the offer is, again, it's a red state, blue state thing, you know. Conservation is a lot bigger deal here, you know, it's like a blue purple state than in Utah. It just is.
Okay, great, Ed?
There's a convergence of seed companies in the Western Great Basin each year for plants like bitterbrush and sagebrush. And the BLM is pretty astute where we work along the Sierra front, and they keep those permits exclusive in areas. But there's an invisible boundary between those permits, and that's a struggle in there. And sometimes they'll even call us and say, are your guys actually working so and so? No, we aren't there. So they're calling, trying to help police their own efforts, but they're understaffed and the territory's big. And so it's a challenge for them to be managing multiple permits in the same area each year. All power to them to keep trying.
Great, thanks. So that's the list that I have here. So do we have questions in the audience here in Fallon for any of our presenters?
Hi, thanks you guys. I actually have a question for Scott, the virtual person. I was just curious, seeing that you're solarizing the beds for about 6 weeks in the heat of the summer to get rid of all the potential weeds and then planting into them. I was just curious if you're getting rid of any beneficial microbes that might be helping with the seed farms, or if that's less of a word since 60% of the time you're trying to do weed control.
Oh, that's a great question. So as it turns out, microbes have different temperature tolerances, and the beneficials tend to withstand higher temperatures than the ones that are pests. So we often mix soil, like if we're growing plants in our greenhouse, we'll often mix our own soil into different components that we want it based on the species that we're trying to grow. And as part of that, we have a steam cart, and we'll heat that soil up to 140 degrees and hold it there for 30 minutes or so. And that 140 degree threshold is where you can knock off a lot of the bad guys and still retain the good guys. Our temperature conditions in the summer, even at the hottest temperatures, rarely reach that 140 degrees when we're solarizing plants. So rather than a instant or a quick flash heat, we do more of a prolonged slow heat that has a similar effect. But it is a temperature threshold thing, and we really don't knock back many of the good guys. It's mostly just the bad guys that get knocked back in that process. Great question, though.
Thanks, Scott. Do we have other questions?
Leanna Carey, Progressive Rancher Magazine. And I'd like to ask the constant stock seed grower. I hope you had insurance coverage when you accidentally or didn't think the process through when you sprayed your plugs. Were you insured by that? And because that can happen. And
it was not an issue in the contract. There would be no liability for our experimentation, say. It was just sad because I knew they went to a lot of effort to grow those plants and get them to us and help us put them in. And it was just sad that it happened. But it's not like they were after us with any vengeance for failure. We were there in good spirit, and they acknowledged the issue, how to deal with broadleaf colonizing weeds in broadleaf small plants. And in the end, it would have been safer to let our favorable plants be overwhelmed by those weeds for 1 year and then use pre-emergence in future years to prevent that from happening. Or wait longer to put down a pre-emergent, but boy, were the weeds coming on fast. There was no question it was going to be an issue. It's our youngness at trying and noviceness, I guess.
Thank you. Thank you so much.
This question's for Jackson or Justin. I was just curious. I was a little surprised to see how much variance there was in the seed testing. And I guess I was just wondering if there is a lab that is kind of the gold standard or if it's just super variable, and if that knowledge is public information for folks that want to make sure that their seeds are quality seeds.
We deal with a lot of different species, and it definitely, the variance gets worse the lower the purities that are expected for the seed. That's when the variance on both the purity and the TZ get much higher. And also we found that with the majority of species, it's not that there's a gold standard lab, there's like the opposite of that, where like 5 out of 6 labs test pretty similarly, where it doesn't affect us too much. But this one lab over here doesn't know how to handle this product. And that's fine, we can avoid that. But if the customer comes and says, we want to use this lab for this, we're like, well, they don't even know what that is. You want to add on that?
Yeah, I just wanted to add, like, this has been ongoing ever since I've been in the seed business. And I'm sure ever since Ed's been in the seed business, like, for instance, like with the BLM, they have a list of like approved labs, right? So all this seed gets retested before it goes to the regional seed warehouse. But there's an ongoing issue right now where there's a couple of labs that we consider to be good labs, you know, that of course you pick the lab that gives you the favorable results, right? That's the one you're going to go with. But like, there's no rhyme or reason to what goes on. It's like, it really is more than an art than a science. And like some of these labs, for instance, you can take the same sample, and these aren't like when he says out of a bag or whatever, these are like, there's a bunch of protocols that have to be done with the testing. Like someone like Russ comes out and they take a lot, and then they take this probe and it's like accurately sampled. And then they'll take this to the lab and I've done this like multiple times. I'm sure Ed's done it too. You send it through a divider, right? So it should be like, though all of those samples had gone through a divider. So you would expect it to be like more homogeneous than it is. You'd expect to have like replicable results, but they just can't do it. Like there's just too much subjectivity in it, like with the TZ reading and all that, and the purity. And like some lab will be like, for instance, BLM right now is upset because some of these seeds went to different labs and no noxious weeds were found. But then they went to a different lab and a noxious weed was found. And so all of this is outside of our control, but we're held accountable for the different labs' variability. It all comes back to the seed company when it's not really something that we can actually control. And in the case of like sagebrush too, like the purity, for instance, it doesn't settle in the bag, all the sagebrush seeds. It's more like you sample the sample. It's like ice cream, right? Fudge Ripple ice cream. Sometimes you stick the spoon in, you get a whole bunch of chocolate. Other times all you get is vanilla, you know? So it's like that with some of these more impure seeds. I don't know if that answers the question.
Just to add a little bit more on top of it, it's not just necessarily variability between seed labs, but like I was saying in the presentation, you can send the same seed from the same lot to the same lab on a different day, and whether or not you give it a different name or not determines how much the results are going to vary. So there's just all these things where like on our side, we can play with it and see like how much do these different things make a difference. And sometimes better than cleaning out a lot is it's better to give it a new name. You'll get a more favorable result by giving it a new name as opposed to giving it the same name and repassing it through the machine, which is, in my opinion, a really big problem, because it's if they can't tell if I'm getting a more favorable result by not even re-cleaning the seed and just renaming it and sending it back versus passing it through the machine and giving it the same lot name because there's some type of lazy, and this isn't just with any one particular lab, this is true with a lot of different labs. So like that's been the biggest problem that I've seen so far is that even it's not just between labs, it's even the same labs themselves, even the same person testing it. And so I don't know.
We do have one, if they're not, oh, go ahead. Since we couldn't see you, Justin, when you got up and showed the bag, the hopper, they do want to see it on the camera.
Oh,
So
sure.
when we get to that point, but Leanna, if you want to ask your question and then we can do that.
Just throw in another plug for Valda. Anybody who wants one, she can sell them. So she's got them, custom made.
So Jess, we'll take care of that in just a minute. Go ahead, Leanna.
Hi, Leanne again, Progressive Rancher Magazine, and I've asked this question a couple times, and it's kind of a can start a fire conversation, is directly to Shannon. I have had this theory, and I've asked in different meetings. Cheatgrass was, in my opinion, included in the reseeding of rangeland fire rehab areas, because of the very point that Shannon has made with your information. As you presented this and as you stated, clean seeding is very difficult. Could this have happened? Could cheatgrass be included in the rehab of seeding the seeds that we need to reseed? And so, thank you. I know it's rare, but I think it's the truth.
Yeah, so it's not impossible, right? If this cheatgrass was collected in the seed and it wasn't cleaned, I mean, it really depends on what species was seeded. If it's a species where it's it's some forb and it looks totally different, the shape, the size, the weight, the color, all of those things are completely different, then it's not so terrible to remove that. But if it's a similar species, another grass species, it can be very, very, very difficult, if not impossible, to remove that. So if it was collected in the original collection, it is possible that it did end up out there. With that being said, I will say that cheatgrass is pretty ubiquitous across, the West. So even if it wasn't in your mix, it's probably going to end up in that restoration anyways, regardless. For sure.
Great. Do we have any more either online questions via chat or in the room?
Yeah, hey, Russ Wilhelm, Nevada Board of Ag. I manage the seed lab for the state of Nevada, and everybody's frustrations and concerns in respects to seed lab testing uniformity. This has been ongoing since the establishment of seed laboratories. Your guys' points are well heard, and at the end of the day, we talk a lot about the evolution of agriculture and where we started, and breeding and cultivating these new varieties that express these uniform characteristics that are easier to clean, easier to handle, easier to seed. And seed labs have kind of evolved with that, right? I think wildland seed or seed use for restoration is kind of a new phenomenon for a lot of seed labs out there. So, there's and I'll talk about this a lot later because I'm speaking to seed testing methodologies later on in the conference. So, AOSA, which is the Association of Official Seed Analysts, they develop and actually publish all of the seed testing guidelines for any species that they have, researched, examined, things like that. And a lot of these obscure species, we're still kind of working it out. So it's tough, guys, no offense about it. But at the end of the day, we're only working with what we have in the lab, right? And I don't know, I think it's going to be a work in progress more than anything. But I will let you know right now that your frustrations and concerns about seed labs are very well heard. Testing uniformity across seed labs nationwide is something that's of paramount interest and concerns to AOSA. And they work to put together a whole slew of proficiency testing and standards to ensure that all these labs are operating under the same guidelines and standards. But you're always going to have that guy that came in after a hearty lunch with sleepy and drowsy, and he's working through a lot of sagebrush, and it's going to be variable. because it is an art form. I think you're right there. Yeah.
I just wanted to throw out one more comment about that, like, for instance, one of these problems that we've noticed too is like recently, like the BLM's done these like spot checks and they'll send in these, that all of a sudden there's labs that have never seen a certain species, they send it there, okay? For instance, like yellow bee plant, okay? Like they sent it off to Montana, which had never seen yellow bee plant. And this is a seed that you can get very clean. It's always like a 99. The TZs should always be in the high 90s. You know what I mean? Because it just is what it is. It's an annual. It's very uniform. So they sent this, and I've had like this particular lot tested like 12 times, right? Always the same. Then they sent it off to Montana, and all of a sudden I've got a TZ that's like a 65, which is unheard of in that, you know, and all of a sudden it's like, well, you guys, blah, 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 blah. Are you cheating us on that sort of thing? When we've never sent it to Montana. And I tried to explain to them, like, they've never seen this. It's like, I ride like these timber sleds, which is like a motorcycle with a sled on it, right? So I'm going to have somebody work on that in Utah who's familiar with it. I'm not going to send it off to Tucson where they don't even know what it is to work on my timber sled, you know? So like, you might want to think about that when you choose a lab for that they're familiar with this species. And like you say, we do like hundreds of different species, and some of the time, like it's a new species, they might not have even heard of it, you know, like every year there's some new species that we add. Like I'd never done turpentine broom, for instance, until this year. So there's always something new. So anyway.
And you wanted to.
I totally agree that you have to keep with the labs that are in the area that's familiar with the species you're dealing with. We sent a Mojave shrub seed, Ambrosia dumosa, off to a lab north and west somewhere, and they quarantined the whole lot. Thought it was an annual weed, Ambrosia. Yeah.
Great, thanks. Russ, did you have anything else?
We'll talk later.
Okay, all right. I want to give one last call for those in Zoom and then in the room, and then if somebody could be Vanna White and show off the hopper so that the camera can see, that would be fantastic. You can stand right in the middle of the aisle.
That's right. Show off your creation, Balda. Both of them.
Oh, look at that.
So where should I stand? In the aisle. That'd be good. Yeah, it is. You can see it.
Jeez.
How close?
The camera's on the
Yeah.
body, right?
Yeah.
Griffin.
Right there is perfect.
Yeah, right there.
That's
Okay.
That's perfect.
So I came up with these two styles. I've been collecting seed for probably about 12 years now, due to fire issues. And I just go out there and kind of grab seed when I see it. And I work through the SOS program. So I saw a need for these hoppers for collecting bitterbrush and sagebrush. So I came up with this style. In a past life, I was a seamstress in an awning company. So I'm very familiar with canvas, and I had a bunch of scrap. Anyway, I came up with this style, and I took this out this fall, and getting it up under the shrub, the sagebrush, and just beating it in, I was able to collect probably about 7 or 8 pounds in just an afternoon with one of these. Anyway, I saw a need for it. I've talked to a couple of people who are on this panel, and nobody has seen these before. Nobody manufactures them. I have an industrial sewing machine. I'm more than happy to make these. I kind of have this style down. Shannon, I just gave her a prototype to try out, and hers is a little bit bigger than this one, but the frame is the same size. I've made some other ones. So this is one that I kind of saw something similar to this. It's kind of shaped like a funnel. And I saw it in an old Forest Service technical report that I read. And I changed this up. It's just PVC pipe, but I just put this together last night, and I just happen to have some webbing and had all this stuff. And so we're gonna take this out. It's kind of high right now, but it can be adjusted. And we're gonna take this out this year and collect some bitterbrush and some sagebrush with it. It might be a little long, but I can I can work with anybody if you have ideas on anything. Feel free to call me or email me. And I can come up with other ideas. I have smaller bags. A colleague of mine works in Arcata, and I made her just a little shoulder bag that has the, it's structured in the opening. And I sent her a picture of it. I'm like, is this what you're looking for collecting beach grasses? Because she does a lot of that. While she's on the coast, it's very windy, it gets rainy. She's asked me to make a flat for it. So I'm gonna do that after I leave here, probably this week. I'll do that and get that off to the mail, in the mail for her. Anyway, you can reach me on my email is bjlockie@gmail.com, or you can call me at 408-568-6743. I'd be happy to work with anybody who's having a problem with getting seed, and if you want any kind of bags.
Great, thank you.
Thanks, Scott.
And we have the great Vanna, I appreciate it. And I have one more question in the chat. Do the panelists have any recommendations for collecting, cleaning sticky seed or from sticky plants? Example, coyote tobacco.
Sticky.
So I think Ed has some experience with some sticky. I'm thinking of Menzelia labicalis and your Velcro in the
Definitely.
and the buy special clothes that you only wear once.
Or wear no clothes at all. I've gotten desperate with a Circocarpus lettifolius. And if you're going to itch, you might as well not have anything touching your skin. Snakes, I don't know.
As far as cleaning it goes, I know I will use like latex gloves or neoprene gloves, and pretty much you just have to toss them after that. And then usually don't put any of those sticky things on any piece of equipment and use sieves. And then if you use alcohol to clean the sieves later, it usually gets most of the gunk off. I don't know if that's helpful, but hopefully it is.
All right, and if not, I have James's information and we can connect him with you. Shannon, maybe you guys can talk specifics if there's things that he has questions about.
Sounds good.
Just one more thing, like to follow up with what
Yeah.
she said, like, yeah, and like Ed said, like with the mahogany, like just read up on the plant, because like he says, like collecting mahogany is like rolling around in fiberglass. And it's like at the hottest time of the year, they'll literally like you can only handle like a couple hours. But then some of these plants like Encilia farinosa, like brittle bush, it's got like this oil in it that'll cause weeping blisters on your skin. So you don't know that until after you've been out there sweating and it's been on your arms, and all of a sudden you just got weeping blisters. So read up on any new plant before you go after it.
Great. Well, thanks, everyone. We're actually ending on time for our lunch break. We're gonna have an hour lunch break, so we'll come back at 1:30. Jess, if you could put up the poll for the people for the first morning panel, or people here in person should have those. So thanks, everyone. We're gonna come back at 1:30 and then we'll finish off the second part of the afternoon on seed certification, seed testing, NRCS programs and collection. So look forward to seeing everybody at 1:30.
Stop after work one day, you know, showing off, and she was into the doctor before me. Oh, yeah, awful.
All right, good afternoon, everyone. We are getting a horrible echo. So what changed? Is that mic on?
No mic there, Megan.
I got it. He had turned on the speaker here. All right, is there an echo in, for those viewing online? You could put in the chat if not. Okay, perfect. All right, so we're gonna get started with the second half of our presentations today. For those that may have missed this morning's presentation, I wanted to kind of go over again a little bit of how it's going to go down. We're doing a hybrid meeting, so there are virtual and in-person presentations. We're gonna save all questions till the end. There'll be a panel this afternoon so that questions both virtually and in person can occur. So if you have questions, for those that are viewing on Zoom, if you could please type those in the chat and who the question is for. And then we'll get those answered during the panel discussion. And then the same for the folks here in the room. Please just hold your questions to the end and we'll make sure to get those answered. I also wanted to remind those that are attending that may not have been here this morning that we are going to share the contact list for those that registered for this event. And so we will be sharing your name, your organization, and your email. If you are not interested in having that information shared, please email Stephen, who you registered with, and we'll withhold that information before we get that shared. If you could do that by the 25th, that would be great, which is this Friday. So again, thanks for everyone joining us. My name is Megan Brown. I'm with the Nevada Department of Agriculture in their Plant Health and Compliance Division. And I'm going to hand over the rest of the next two presentations to other staff members who are really the technical experts that work in our seed lab and our native seed staff. So our first presenter who's going to talk about seed certification is Steve, and he received his bachelor's degree from natural resource management from Texas Tech University in 2017 and moved to Reno shortly after. He has previously worked as a range technician for the BLM and as an inspector for the entomology program for the Nevada Department of Ag with the infamous Jeff Knight, who I think anybody in Nevada is like 6 separations from Jeff Knight. Anyway, and currently he is the native seed program technician for the Nevada Department of Agriculture and assists in source identification, allium inspections, and outreach opportunities such as this. So I'm going to turn it over to Stephen. Thanks.
Thank you, Megan. Hope everyone had a great lunch today. So today I'm going to take you through the different types of seed certifications that the Nevada Department of Agriculture offers. Next slide, please. So the NDA's role in the native seed industry is known as the official seed certifying agency. We are authorized through state statutes to conduct quality assurance services related to all types of seed, not just natives. This is important because it allows us to legally perform inspections to ensure buyers are getting what they're paying for, essentially. Next slide, please. ASCA is the certifying agency or the organization that sets out all the standards by which we certify. Since it's a nationally recognized organization, we are able to perform inspections for companies outside of Nevada, and in turn, they can sell their seed to anyone that recognizes ASCA standards. The yellow book, which some of y'all may know about, is the law when it comes to certified seed standards. It's revised by ASCA every so often and contains standards by which every crop type is certified. It obviously goes into much further detail than I can get to today, but it does outline standards to overall crop quality, including off-type presence or anything other than the target variety, weed presence, infection, and lot purity / viability. Next slide, please. So with all the legal stuff out of the way, let's take a look at how the NDA supports both agricultural production and wildlands collection. And more importantly, we can see how they in fact can benefit each other. Next slide, please. The NDA offers 2 broad services related to native seed that a company can apply for: source identification and seed certification. Source identification involves a trained NDA staff member, usually me, traveling to a collection site to certify that the target species is present. Certification is a much more broad service and can include many different types of seed, not just natives. There are multiple tracks with multiple different levels of certification, which we'll touch a little bit on later. Each level has different standards that a producer has to meet that become increasingly more stringent the higher up the chain that you go. This process involves periodic inspections to ensure requirements are being met throughout the growing season, as well as periodic tests as well. Next slide, please. What you see now are the 2 tracks that I mentioned earlier. You can see that they are the cultivated and natural tracks, which reflect their origins. I will briefly touch on the cultivated track, but the main focus of today is going to be the natural track and specifically source ID. Source ID is the foundation of the natural track, and through selection and genetic testing, you can move seed into the selected, tested, or even cultivar status over several generations. The advantage to increasing the status of your seed is that extra level of assurance that comes with the extra care, and potentially your stock showing genetic traits that gives it a competitive edge in some rehabilitation situations. The cultivated track kind of works backwards to the natural track in that the further from the breeder status that you get, the less stringent the standards become. This is because of the increased chance that genetic variety is introduced. The foundation of the cultivated track being breeder seed is the original source of all classes of that particular variety of certified seed. And it is controlled usually by the original plant breeder to ensure maintenance for genetic purity and identity. Next slide, please. Now we're really going to deep dive into source identified seed. It's defined as propagating materials collected from natural stands, of course, but more importantly, it can also be collected from seed production areas, seed fields, or orchards where no selection or testing of the parent population has been conducted. It is essentially unaltered seed collected for planting purposes. And I would like to stress that while wildland seed collection sites are the bulk of our source ID applications, a producer can still qualify for source identification on agricultural land if they meet these specifications. Next slide, please. So these are the steps that are involved in source identification. And we're going to break each one down a little bit in a second, but let's take a look at some of the fees associated with the program. They're fairly self-explanatory, but it is worth mentioning that each application costs $10 per species. A site fee will also be charged, but different species in the same site can be combined to reduce some of that cost. Tags that state the seed has been certified can be purchased at 10 cents per tag. And each inspection will also include a fee for drive time, mileage, and inspection time. Rates for these are calculated using the current standard IRS mileage rates. These do change from time to time, so it might be worthwhile to research these costs beforehand, just to know what to expect. Next slide, please. So starting with step one, you will want to submit your pre-collection application by downloading the PDF via the NDA website. If you have trouble finding it, you can always get in contact with me. If you have any trouble with the application at all, feel free to get a hold of me and I can help you out. It is fairly self-explanatory, but I would like to highlight two sections. Your application should be submitted before collection takes place. Ideally, at least 3 days, but we understand that things happen and we can work around that. This is to ensure that the target species is flowering at the time of inspection, which makes it much more accurate and a lot quicker. Additionally, accurate GPS coordinates and driving directions are key. Your GPS coordinates should lead the inspector to within walking distance of the target species, and driving directions should also include access instructions if that's applicable. Inaccurate coordinates or directions may lead to a rejected application requiring resubmittal and additional charges. Like I said, though, we're willing to work with you, but it just makes my job a lot easier. Next slide, please. Right, so step two is the inspection occurs. What we're really looking for are the target species presence, weed density, if any, and we'll also perform an overall habitat assessment. The actual inspection is fairly simple. Typically, we'll take a GPS point, take a few pictures, some notes, and a small sample of the target species that we call a voucher. We'll typically hold on to this voucher for our own herbarium if it's a really good specimen, but it will also be used as a reference in case discrepancies should arise down the road. Next slide, please. Step three involves having your seed lot tested. Now, I know we've already talked a bit about that today, and Russ is going to cover that much more at length, so I'm not going to get too into it right now. But typically, these tests will tell you the germination rate, overall purity, amount of pure life seed, and weed presence in your tested lot. I will point out that lab analysis is not required for source identification certification. However, some buyers will request that proof of testing. Next slide, please. The tag is the end result of the process, and we actually have a few examples printed out on the table behind you if you're in person. You will attach these bags, you attach these to your bags of seed before sale, usually, to indicate that they have been certified. Just because you get an inspection, though, does not mean that you have to order tags, nor do you have to order tags during the same yearly inspection, so you do have a little bit of leeway. The application for tags can also be found on the NDA website alongside the pre-collection application I talked about previously. And again, it's fairly straightforward. You also do have the option of requesting the seed zone or average precipitation to be included on your tag. That's usually going to be at the buyer's request. Next slide, please. So this is what the source identified tag looks like. As you can see, they're yellow in color, which represents the certification status of the seed. Each different status is going to have a different color associated to it. I will say that the producer/collector is responsible for determining tag quantity and your lot number, but we can help you complete all other fields if you need assistance. Just reach out to me. Next slide, please. So your options after completing source identification. Obviously, you can sell what you have for restoration, hopefully, or continued agricultural use. Or you can use your seed to pursue higher levels of certification as I described previously. Like I mentioned, the production will be overseen by the NDA to verify crop purity. We will again utilize inspections and testing to confirm off-site presence, weed presence, disease density, and viability over the course of generations. Next slide. And that's going to bring me to the end of my presentation. I'd like to thank you all for your attention. I know it was a very short one compared to some of our others. But if you have any questions for me, you can ask them during the panel discussion later. My contact information, I'm sure all of you have reached out to me if you're here. So you have that. Feel free to contact me. Thanks again.
Good job, sir. All right, thanks, Stephen. Our next presenter is Russ, who manages our seed program at the department and is going to be talking about seed testing. Russ received his degree in environmental science from the University of Nevada, Reno in 2013. Following the receipt of his degree, he joined Nevada Department of Agriculture as an intern for the noxious weed program. After 1 year with the weeds program, he was hired to manage the department's seed program, including running the seed laboratory. Management of the seed program has allowed Russ to refine his skills and knowledge related to seed certification, seed testing, regulation, and the seed industry. So welcome, Russ. And again, for those that have questions, feel free to put them in the chat and address them to who they need to be, and we'll make sure that we'll get them answered during the panel.
Cool. All right. Well, thanks so much, Megan. And hats off to Stephen real quick, guys, because that was an incredible presentation. He's only been in this role for a few months now, and he's learned just an exorbitant amount of information over those last few months. So, and thank him again for putting together a wonderful forum, because this was no small task, buddy. Well, all right, guys. I guess I'm graced with presenting on seed testing. It's a hot topic today, I know, but hopefully we can get into the weeds a little bit. I only have 15 minutes. I guess Stephen wanted me to keep it brief, so we'll have about an hour in the panel discussion to really get into more seed testing methodology and kind of just the concerns overall in the industry out there later then, because I'm happy to make a quorum or a forum for that, right? So Jess, go ahead, next slide. Good deal. So there's an organization out there called AOSA. AOSA is the Association of Official Seed Analysts. Now, if you work in the seed industry, there's quite a bit of acronyms that sound similar to this. You've got AOSCA, you've got AOSA, you've got AFCO, you've got ASCO. This is AOSA. AOSA is the body that actually develops and publishes all the rules for testing seed. AOSA has been around for over 100 years. And, you know, as we mentioned before, modern agriculture kind of has been refined and been looking at more, you know, traditional crops that have been agronomically cultivated and manipulated over time to express different characteristics that actually make them easier to handle, not only in an agronomic setting, but also in an analytical or a lab setting, right? So
with kind of the introduction of native species or wildland species, it's kind of thrown a curveball at the seed laboratory community, right? And they've been working, I mean, decades upon decades now to refine the techniques to properly handle these native species. And as you can see there, methods have been established for over 1,000 different species of seed, and each method is reviewed and evaluated annually, right? So there's actually a pretty rigorous working body that operates with EASA, looking at and investigating more wildlands, more seed used for restoration. So just bear in mind that this is a work in progress. So we'll get there, but you know, we are where we are. So next slide, Jess. Okay. So how to request a test through the Nevada Department of Agriculture Seed Lab, how to take an appropriate sample, and then fees associated with any lab services that we do have. If you want to request a test with the Nevada Department of Ag, what you can do is just jump on our website, hop on down to seed certification. There's a link there that you can click on. It'll direct you right to that test request form, or you can simply just email me and I'll send it right over to you. Fill that out, attach that in its full detail to the actual sample of seed that you'll be submitting, and then send it away and I'll collect it and process it in a timely manner. So additionally, all of our fees and required sample sizes per species can be found online as well. So each individual species comes with a very unique sample size that you'll have to submit with it, right? A sample of, let's go with green peas, is going to be a lot different than a sample of Artemisia triticata, right? I mean, we really only need about 5 grams of something like mountain big sagebrush, whereas with green peas, we need probably about 5 pounds, right? So just make sure to review those before you submit that sample, or go ahead and get a hold of me and I can walk you through the process there. And then, you know, the big kicker here is to ensure that you provide a sample that is reflective of the current lot conditions, right? The sample is going to dictate really the validity of that test, right? So our test can only be as good as the sample quality that was submitted to us. Okay, Jess, next slide. All right. So types of tests performed, and Shannon kind of stole my thunder here a little earlier, but we also do purity analysis and germination analysis. We operate and actually conduct noxious weed analysis as well. Those 3 tests are kind of the Slayer standard tests that really any seed lab will perform. And the reason why that is because they're legally required per the Federal Seed Act. Every bag of seed that's entered into the commercial market needs to be tested for purity, germination, as well as noxious weed analysis, right? So any bag of seed that you buy, anytime you go to a Home Depot and you pick up a packet of seeds, it should have those 3 analytical results on there, right? Purity, germination, and noxious weed content. Other common tests that we conduct is a bulk analysis, where it's essentially just an over-glorified purity analysis, just a larger volume of seed that we're handling, right? Tetrazoleum analysis, and this is probably most important to this group because a lot of these wildland seeds or seeds used for restoration, you're going to want to have a tetrazoleum analysis performed on it, right? Because the germination tests in a lot of instances for a lot of these native products can take upwards of 6 to 8 weeks. I mean, there's certain species out there that take 6 months just to process in the lab, right? So the tetrazoleum analysis is essentially just a rapid viability analysis where you can biochemically stain the seed with tetrazoleum chloride, and it bonds to the respiratory sites of the embryo and actually stains it a nice vibrant red. Well, ideally it's a nice vibrant red. In a lot of instances, it's kind of a muddled pink and there's some paleness to it, but as seed analysts, we're tasked with discerning between, is that a nice vibrant red or is that a pale pink hue, you know? So yeah, we'll do tetrazoleum analysis and we'll get deeper into the mud there and tetrazoleum analysis as I go further into this presentation. We do moisture content and then GMO assay, but that's only on alfalfa. So if you're growing alfalfa and you want to know if it has any sort of GMO contamination, give me a call. All right, Jess, next slide. Okay, hopefully you guys can see this pretty well in the back. I'm not too sure, but this is just a synthesized version of the EASA rules that I put together that I think are easier to work with. And I just jotted down just some of the, most common or some of the most common species that we work with as far as wildland species. There's a few exotics on there too. But it'll tell you species name, for example, Indian ricegrass is there at the top. The working weight, so whenever we're handling Indian ricegrass in the lab and we're doing a purity analysis on it, we just need 7 grams of seed, right? For a noxious weed analysis, you multiply that by 10, we'll need 70 grams of seed. And then they jot out exactly how to perform that germination test for Achnathorum hymeneoides, which is Indian ricegrass. You need to put it in a petri dish under 3 pieces of filter paper. It'll tell you what germ temp it needs to run at the entire duration of the test, which is 15 degrees C. And then when you're going to conduct, for per germination requirements under EASA, 2 checks must occur, right? First check to pull out all those seedlings, so it's not just a metal mess inside that petri dish. And the final count to give your exact tally as far as how many of those seeds germinated, right? And anytime we're handling any species all across the board, regardless of what it is, we're testing a minimum of 400 individual seeds, right? So for Indian ricegrass, first test 1 week after you put that into the germinator, and then 42 days is the final count, right? So you're waiting a month and 12 days just to get your germination results on a hot bag of seed that needs to go out to the BLM last week, right? Which is why in most cases, native seed companies, they'll opt to perform that tetrazoleum analysis, right? The difference between the tetrazoleum analysis and the germination analysis at the end of the day is, yeah, maybe an art form and it may have a higher degree of subjectivity to it. But also it doesn't give you an idea as far as what number of total normal seedlings you have in that lot, right? The additional factor that you're looking for in the germination test is you're not only pulling out which radical emerged and which seed looks or appears to be a viable normal seedling, you're also conducting an analysis on each individual seed to ensure that it has all the anatomical components to produce a healthy flowering plant, right? And with tetrazoleum, you're just looking at the embryo, right? And then just a couple more on there, and I'll probably run out of time. Great Basin wild rye, 21 days total count. Siberian wheatgrass, 28 days total count. So it's a lengthy duration for these tests. Okay, Jess, next slide. Cool. So I don't really want to beat a dead horse by any means, but tetrazoleum testing procedures, those are also produced by EASA in the handbook called the Tetrazoleum Testing Handbook. I actually brought that here if anybody wants to take a gander at it and look through there, see what all the illustrations look like, and maybe try your hand at being a seed analyst for once and then see if you can dictate, okay, is that a positive or a negative or a positive or a negative? So if you want to have a little game after this, we can do that. So the Tetrazoleum Testing Handbook actually categorizes the testing methods into family units. So you've got Asteraceae, you've got Poaceae, you've got all the different families within the plant kingdom, right? And then they group those. So Poaceae might have 1 to 3 different subgroups, and then those subgroups are broken down by genus, right? So Poaceae 1 might have all your traditional cereal rides, right? And then Poaceae 2 might have all your more exotic, more native oriented species, right? And then the testing strategies vary based upon the overall physiology of each particular seed species, right? So depending upon what category, what family it exists in, that's how you're going to treat that seed differently, right? And then some seed species possess morphological distinctions that make tetrazoleum testing difficult. I'm sure a lot of folks in here have handled Atropox canescens, for example. It's got one of the hardest armored coatings I've ever dealt with in the lab in my entire existence. So, traditionally what you're doing with the tetrazoleum test is you soak the seed in a vial of water. Hopefully that loosens up the seed coat to the point where it's permeable, right? So then you go in there and you actually excise it, you cut it in half to make the embryo actually penetrable from the tetrazoleum chloride solution. when you're going through and cutting 4-wing saltbush, or you're burning through, you know, probably 15, 20 different razor blades. And you're doing this 200 times over because the tetrazoleum standards indicate that you have to test minimum of 200 individual seeds, right? So it's no small task. I can tell you that firsthand, right? Yeah, but for the most part, tetrazoleum tests can be quite reliable if done correctly. But like I say, you don't get that normal seedling readout, right? Similar to what you would get in the germination test. And most TC tests can be concluded in 3 days. So there you go. Jess, next slide. Thanks, Jess. So this is just a photocopy essentially that I threw up there of what the tetrazoleum testing handbook would illustrate, right? So there's POACA group 1, that's Avena, Hordeum, Orzae, Cicale, Triticale, and Triticum. And look at that, you know, that's Hordeum vulgare, that's barley right there. Just a nice vibrant red, right? It's stained on the embryo. That's all your endosperm on the top, and then the embryo is housed right there at the very bottom. Pretty easy to discern, you know, that's probably a viable seed, right? And the one right below it, you know, if you maybe gone to lunch a little later and you were having too many French fries and you're feeling a little drowsy, you might look at that second one and say, yeah, let's pass that through into the positive pile. But in all reality, that's going to be a negative seed based upon the way that the embryo was stained. It looks like the radical there, the very bottom portion of the seed embryo, was not going to be viable, right? So that radical actually protrudes from your seed coat. And that's going to be the first primary root component that forms out of that plant, right? And without that radical, you're not going to have a plant. So, and with that, we can take a closer look at this later if we'd like to. Yeah, next slide, Jess. This is just what our test reporting looks like. You know, on there, we always put a warranty, just like any other lab would. And we warn that the test results reported on this form have been carried out per the OWASA rules. And the results reflect the condition of the submitted sample, blah, 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 you know. And pretty much, you know, all seed lab report of analyses look very similar to this because AOSA also, you know, dictates what your lab report form can look like. So that's why, I mean, the ones that Jackson had pulled up earlier look pretty close to this one. Okay, Jess, next slide. So conclusions, guys. You know, the NDA lab can test most species. We went over all the tests that we can perform. I think that providing a representative sample is key, one that's proportionate to the lot, and then always provide enough seed to perform all tests desired. And like I mentioned before, seed entering the commercial marketplace must comply with both federal and state requirements. So FSA, what tests need to be done in order to make this seed legal, right? So that's that, guys.
Thanks.
There you go.
All right, thanks, Russ. We're going to move on to our next presenters, which are going to be virtual. We have Jim Gifford and Mustafa. I'm going to, now that I just practiced it, I'm going to mess it up. So I apologize, Mustafa. I'm just going to let you introduce your full name when you join and present with Jim. Jim is the state resource conservationist with the Natural Resource Conservation Service in Reno, Nevada. He's worked for NRCS for 28 years, all of which have been in Nevada, including Reno, Lovelock, and now back in Reno. His background is in range management. Mustafa is the assistant state conservationist for programs at the NRCS in Reno. Throughout his 8 years as an NRCS employee, he has worked in many different areas and states, including managing the Environmental Quality Incentive Program, which is EQIP for short, for NRCS in Iowa, as well as the soil conservationist in multiple areas of Texas and a district conservationist in Indiana. Mustafa and Jim have worked directly with farmers and ranchers and other land managers to develop conservation plans, manage multiple land uses, implemented conservation practices, and provided conservation technical assistance to growers. And so I see Jim up on the screen, so I'm going to hand it over to you, Jim. Thanks so much. And again, we're going to be hearing about NRCS programs.
Okay, yeah, thank you. And, yeah, good afternoon, everybody. This is a great forum. I've been learning a lot so far here. And I got to say thank you to Ed Kleiner for talking about NRCS. I was lucky enough to get to work with Ed and Linda on a couple of those early projects. Yeah, that's the best kind of endorsement that NRCS could hope for. I probably shouldn't even talk. I probably shouldn't even do this presentation because it could only go downhill from Ed talking about us there. But hey, we got 30 minutes to kill. So what else are we gonna do, right? Yeah, next slide, please. So these are topics that we'll cover today. I'll cover the NRCS kind of history and mission, resource concerns and conservation practices, and then Mustafa will jump in and cover NRCS programs. Then I'll cover the Great Basin Plant Materials Center, but our PMC manager, Chris Burnau, is on the call too, so he can help bail me out when there's questions from the panel. So, okay, next slide, please. So we'll start off with NRCS history and mission, because this is sort of a NRCS 101 presentation with kind of a small native seed twist to it. So next slide, please. So I'm going to talk a little bit about how we got started, because I think to understand what NRCS does now, it really helps to understand how we got started, and that's back in the Dust Bowl is the short answer. But I've included several pictures of it just because it's just kind of some fascinating history. So between 1925 and 1930, land under cultivation, in the Midwest more than tripled. And there was little to no conservation practices, in place to prevent erosion. So, next slide, please. And then you get this, they got this drought. 1934 drought was the worst that we'd had at that point in US history. Soil of the Great Plains, is drying up and blowing away. And by December 1934, about 100 million acres of cropland had lost all or most of its topsoil. The next slide, please. So that brings us to April 14th, 1935, which is the Black Sunday, the worst, what they call Black Blizzard of the Dust Bowl. You see some of the photos there of that cloud coming, the blizzard coming. Okay, next slide, please. So this is that day when it hits downtown Garden City, Kansas. So the photo on the left is just before the dust storm hits. Photo on the right is just 15 minutes later. You can pick out a couple of those lamps in the picture on the left, and that you can see in the picture on the right. It's a little bit difficult, but yeah. Okay, next slide, please. And so I'm sure there was many people, but Hugh Hammond Bennett is the father of soil conservation. He was the one that was really driving and speaking about how we need to find a solution to this national problem. And despite the formation of, there was a soil erosion service and the Civilians Conservation Corps that got started a little bit earlier, like in the early 1930s, around 1933 and that. But there need to be more of a nationwide effort. Next slide, please. And so what he does, this is I really wanted to include this because it's just a great story and proof that timing is everything. And Hugh Hammond Bennett, he definitely had the timing. So March 11th, 1935, he addresses Congress on the need for the federal agency in charge of soil conservation. He times it out so that during this address to Congress, the dust storm from the Midwest, from the Dust Bowl, hits Washington, D.C. So Congress decides actions must be taken immediately. Wasn't a single dissenting vote in Congress to establish the Soil Conservation Service. And 1 month later, in April 1935, the Soil Conservation Service is established in USDA. The next slide, please. So that's how we got started. Then in 1994, the Soil Conservation Service decides it changes its name to the Natural Resources Conservation Service to better reflect that we were really conserving all natural resources and not just soil. So our mission, we deliver conservation solutions so agricultural producers can protect natural resources and feed a growing world. Next slide, please. So next we'll talk about some of these resource concerns and our conservation practices. Next slide. So NRCS has, what we call resource concerns, which is basically just a natural resource problem. We've got 47 of these resource concerns. A few that might be addressed like with native seed would be like soil erosion, nutrients transported to surface water, sediment and surface water, plant productivity and health, plant structure and composition. And then for animals, for livestock, on the livestock side, it'd be like for feed and forage imbalance, basically not having enough feed. And then inadequate wildlife habitat as well as a great place for the native seed to fit in. Next slide. What I did want to say, like on the resource concerns, like for us, you notice they deal with soil, water, air, plants, and animals. So like purchasing, like you need a piece of like equipment, like a harvester or something to harvest native seed, that's not an NRCS resource concern, or buying, helping out with purchasing seed cleaning equipment or something like that. That's also not an NRCS resource concern, things like that. So. Okay, so now I'll move on to our conservation practices. Oh, you can back up, sorry. So our conservation practices is how we solve resource concerns. And conservation practice is just a specific treatment, like a structural or vegetative or even a management practice that's commonly used to meet specific needs, for planning and implementing conservation. So it's things like fences and irrigation structures, irrigation pipelines, water troughs, range plantings, all kinds of stuff. And all these conservation practice, each practice has a standard, and most of them also have a specification. Those can all be found in our Field Office technical guide and in section 4 of that technical guide. And you can find that off of our website, or even if you just Google NRCS Field Office technical guide, you'll be able to find that. We've got about 170 conservation practices available. And the reason it says, about is because different states will have different numbers of practices. Some practices don't apply in Nevada like they might in Massachusetts or something. So I can leave some practices out of our technical guide that we don't need in Nevada, that they might need in someplace like Massachusetts or Florida. And then conservation practices often work in systems. For example, if we do a fence, we're very likely going to do prescribed grazing as another practice to go with that fence. Okay, next slide, please. So let's look at a couple of conservation practices that are most likely to utilize native seed in Nevada. One of those would be range planting, which is code 550. That's an internal NRCS code for us. But so range planting is basically the establishment of adapted perennial or self-sustaining vegetation, such as grasses, forbs, legumes, shrubs, trees. Purposes include restoring plant communities, close to their ecological site description, livestock forage, wildlife food and cover, reducing soil erosion, et cetera. It's a pretty broad practice. And there you can see in that table early, hopefully you can see, I realize it's a little bit small, that we do recognize that the native seed likely costs more than non-native seed. And so that is built into our conservation practice, conservation financial assistance and into our components there. So you can see like the native, if we do native seed, we can help cost share $123 on that, whereas the non-native seed is only $61. So native seed's almost double on that particular practice. Okay, next slide, please. Another good one that would likely use, actually very likely to use native seeds is our wildlife habitat planting, which is, as you could guess, for establishing wildlife habitat by planting herbaceous vegetation or shrubs. Purposes include improving degraded wildlife habitat and establishing habitat that resembles the historic desired reference native plant community. And so for that table and the previous table, this happens to be for our environmental quality incentives program. And then you can see the components there, like these are just a couple of examples. The low species diversity on non-crop land, it's $194 per acre is what we would provide for something like that. And then just to give you another example, high species diversity on cropland with foregone income is clear up to like $692 an acre that we would help out if someone were to do a practice like that. Okay, next slide, please. All right, I will turn it over to Mustafa for our programs.
Can you hear me, Jen?
I can hear you now, Mustafa.
Okay, perfect. So hi, everyone. My name is Mustafa Abouari, and everyone calls me Mustafa, I guess. I will be doing a presentation about our NRCS programs that the ones I manage, and some of them are managed by Jim and our state engineer and our soil scientist also. Next slide, please. Okay, so originally when NRCS was created, this is the only program we have, a technical assistance. And we still provide free technical assistance to producers, land managers, or anyone that needs access to technical assistance can go to our field offices. Example of technical assistance that we provide: land use maps, soil informations, imageries, if there is a need for economic costs and benefits for managing a farm or a ranch. Also like engineering designs and engineering notes. And this, like just when Jim was talking about the history of NRCS, this is the original goal of what NRCS was created before we get into money and funding. So next slide, please. Okay, this is one of our small programs that's only available on certain states, including Nevada. This is usually being used in Nevada to install seasonal high channels. There is a payment limitation on this. Agriculture management assistance, the payment limitation is $50,000. So it's usually targeted towards small producers or small management practices, including seasonal high channels and other small projects. This is managed by, so some of the funding comes from risk management agencies. So it's like a program that's managed by multiple agencies or multiple USDA agencies. This is not like the Food Security Act does not apply to this. So this is one of them where if you don't have a history of cropping, where you can go in and request assistance under this program. And on Nevada, last year, on 2021 fiscal year, close to $400,000 funding that was spent on it, on about 866 acres. Next slide. I did see a presentation about some of our enhancements. So all those enhancements that were presented, including monarch butterfly enhancement and other mixed species enhancements, go under this conservation stewardship program. We usually advise our producers that have already done EQIP or EMA. This is like the next step for them to take the conservation to the next step or the next level. In Nevada, this program is just getting, like, I guess, picked up. It's not been, historically, it's not been used as much. So close to $550,000 that was spent last year. Next slide, please. Okay, environmental quality incentive program. This is the program that's mostly used, not just in Nevada, it's almost in all states. And I believe this is the program that has the most funding on the Farm Bill. I think it's over $1 billion a year. In Nevada, close to $8 million that was spent. And so this is, compared to CSP, you address the entire resource concerns on your land. On EQIP, you can address only one resource concern. That's why it's preferred by producers that are not willing to, I guess, invest time and money and management. They usually prefer to just do one practice or address one resource concern. That's why EQIP is more popular than CSP. The next slide. Okay. This is another program that's not really been used as much on Nevada, where it requires a partner to provide one-to-one funding to invest in conservation. And there are two types of RCPP. There is conventional one that mimics. It's called mimicking a program. It's either mimicking EQIP or mimicking CSP usually, or mimicking the easement program. However, there is also a funding available for projects that are not like what it could be like a enhancing a watershed or a project that's not really mimicking a program, even like if it's sometimes like something that's not similar to what was done on other programs. And it could be any entity, non-governmental organization, usually non-profit organizations, Indian tribes, state or local government. Usually it's state or local government, state Department of Agriculture that get into these partnerships, or sometimes the conservation districts also. The next slide. Okay, so these are other programs that we did not have a slide for all of them. A common one is the easement program. There are two types of easement programs. There is wetlands programs, which is for retiring land. And this could be one-to-one wetland, so to uplands. So if you apply for an easement under the wetland reserve easement, it requires a one-to-one wetland where you will put the wetland to retire. It's not like a working lands program. But however, there is the other part of it, which is more common here in Nevada, the ag land easement, which is for working lands where a partner brings in 50% of the cost of the fair market value where we put the easement on that land or ranch that's working. However, if it's a grassland of special significance, NRCS can bring up to 75% of the fair market value of the land. Then there is the other one, also conservation innovation grants. This is if an organization, an entity has some innovation that they sometimes either they want to test or they want to see if they can run it through producers or work with producers to implement some of their innovations. They can apply for this conservation innovation grants. The other one is managed by our soil survey. This is one of those technical assistance that NRCS provide also. The fourth one is snow survey. It's a technical assistance that's managed with our soils also in Nevada here. Then there is the watershed programs. If you heard about the infrastructure bill, this is where that money is going. Watershed programs, it's managed by our engineering, our state engineer. Then there's the NRI, National Resource Inventory. This is one of those technical assistance that going on. It's also managed by our soil scientists. Next slide. Okay, turning it back to you, Jim.
Okay, I'm unmuted. Yeah, thank you, Mustafa. And you could see there's quite a list of things that we work on. It's pretty diverse. So I'll talk a little bit about our NRCS Plant Materials Center. Just a couple of slides here. Next slide, please. So here's a map of all the NRCS Plant Materials Centers across the country. Excuse me. And of course, ours is here in Fallon, Nevada, called the Great Basin Plant Materials Center. Next slide, please. And so what they do, I mean, it's quite a bit, but they evaluate plants for natural resource conservation. One of their main customer is the NRCS field offices and helping our planners, our soil conservationists and range conservationists do a good job in choosing the right plants for the right situation. They develop technology and tools for our conservation practices, provide plant materials training, presentations, produce a lot of technical documents for field staff and farmers, ranchers, the public, release promising new plant selections to commercial growers. And then they maintain that breeder and foundation seed as well. Next slide, please. And so just a quick snapshot of what the PMC has done just over all the PMCs in the country have done over the last 3 years. They've written over 450 documents about like plant fact sheets and planting guides and study reports and all that. Then over 4,500 participants in their trainings, 290 presentations, including hosting 110 tours of these plant material centers, 30 field days. So yeah, there's been thousands of NRCS participants and partner agencies, and farmers and ranchers. And they've done 10 new conservation plants released to commercial growers. And just want to say, so in Fallon at the Great Basin Plant Materials Center, Chris Burnell, our manager there, he's been working on several things. One species he's been working on recently is Indian rice grass. And it was interesting to watch, the presentation that Scott Jensen did this morning. And he showed that map, and you could see where Magnar and Trailhead and Basin Wild Rye were from, clear up in Canada and Montana. Well, for Indian ricegrass, Nespar and Paloma have been developed a long ways away from Nevada as well. So Chris is trying to develop that Indian ricegrass that's a little bit closer to home for us. So. Okay, next slide, please. Okay, NRCS contacts and more information. So next slide. So for Nevada NRCS, we have field offices scattered around the state. Elko, Ely, Fallon, Las Vegas, Lovelock, Minden, Winnemucca, Yerington. And then we also have the Great Basin Plant Materials Center in Fallon as well. This map also contains a lot of the tribal entities that we work with around the state. We do have this USDA Service Center locator that's on our website, and a couple of different websites where you can go and find your service center, because that's definitely the place to start, is going into one of our field offices and talking to them about what you might be interested on your farm or ranch. Next slide, please. Here's a list of all the contact information for the district conservationists, which are basically the boss, the supervisor in those field offices around the state. And then, of course, we also included Chris Burnell's information at the bottom there, since he's the PMC manager there at the Great Basin PMC. Next slide, please. So for some more information, we've got our national NRCS website. There we've got the Nevada NRCS website link there. And just wanted to remind everyone that yeah, we're a conservation agency. And those websites can tell you a lot more about that. We've got a really great website is farmers.gov. And just throw out some reminders there. It's not farmers.com. That will take you to the Farmers Insurance Company. And it's definitely not farmerdates.com or farmersonly.com. Spouse might be a little worried if you're going on to a farmer dating website. So be careful of that. Remember, it's farmers.gov, which reminds me I need to tell my wife, remind her that I was looking up farmers only stuff, dating websites because of this presentation, and not because I wanted to be a farmer.
Thanks, Jim. Is that the end? Sorry, I didn't mean to.
Yes, that is the end. Thank you very much.
Thank you, Jim and Mustafa. Appreciate your participation virtually and giving us some good background information related to programs that might be related to native seed. And I think Ed also did a good job of talking about how he incorporated some of the benefits of using those programs and getting native seed as sort of a side benefit. So and harvesting that seed. So really appreciate that and your participation at the meeting today, both of you. We're going to move on to our next virtual presentation. Corinne Francis is going to talk to us about seed collection permits on BLM land. Corinne Francis Clark is a state forester for California and Nevada BLM. She became the forestry program lead for Nevada BLM in 2012, and then combined this position with the California BLM state forester position in 2017. All commercial and personal use permits for seed collections are issued by the forester program leads in each BLM office that manages the lands for which those collections occur. And I'm sure we'll get more details moving forward. Oh, there you are. I can see you on my screen. I can see you up on the big wall. We have two big projectors. Hopefully maybe we'll post a picture of what it looks like from those sitting at the seats here in Fallon. But again, Corinne, appreciate you, and I'll turn it over.
Yeah, thank you. How's my sound coming over? Good. Okay. Thank you. Welcome to this topic. I gave this same presentation last year at the previous forum, so it's kind of a repeat. But thank you for inviting me. I'm honored to be part of the seed problems we are facing in Nevada. I think that the permits that we issue out of forestry are kind of pivotal to getting your seed and restoration needs. So go ahead and go on to the next slide. So I just want to talk about the type of permits we have, some of the policy, how we price the seed that we have under the permit, kind of give an overview of the typical permit form, who to contact for seed collection, and some of the time frames. And then I definitely encourage questions and discussion at the end. Next slide, please. So we approach our permits into different categories, and it depends upon what the seed will be used for. So a commercial permit allows for companies to resell the seed, clean and sell it. A lot of it goes back to our seed warehouse in Boise. And so we have commercial permits, we have personal use. It's basically no resale, trading, or bartering. And then sometimes we issue free use permits when it's in the best interest of the public. And we have some regs with free use where, permits to state agencies when they're using the seed for restoration projects might have more seed and value allowable versus to say nonprofits where our free use amounts are capped at $100 per calendar year of value. So, we have a pretty robust seed program in Nevada. This is the pounds we issue each year for 18, 19, and 20. So it varies, as you can see. And it does bring in some values, some money that goes back to the Treasury. So that's kind of just an overview. I'll go on to the next slide. So, for our veg permitting, it definitely kind of lands in the forestry regulations, the CFR 5400 regulations. And so in that, we, have to appraise for fair market value and, open it up to competition so that we're not favoring one person over another. We also have a Washington office IM that allowed for more consistency across states in how we issue the permits and how we price them. So go on the next slide. So in this policy, it requires us to look at competitive measures such as bidding same areas and getting the highest priced bidder when it's practical, when somebody's interested in the same area. We only deal with 10-pound increments by species. And then the permit always has to be in possession of all collectors and harvesters. So if law enforcement stops someone who's collecting seed, they have to have that original permit in their possession. So it also encourages folks to source seed it and provide GPS locations so that we can always know where that seed came from. So we require bonding at the national level for all sales of $2,500 or more. And that's a 20% bond that when you fulfill the requirements of the permit without any stipulations being violated, you get that bond back. And in Nevada, we require it on all sales of $200 or more. You know, part of it is just encouraging collectors to notify us so that we can track everything, follow the conditions of the permit, and then they get their bond back. So we always include stipulations attached to each permit. Next slide. So we also require the use of a minimum price, and this is set at 10% of market value. Price can be set higher if there's you know, market trends to indicate it's worth more for some reason. We have yet to set prices higher at the start, you know, when we're offering a minimum price. But we have had it kind of bid up by people, you know, wanting to get the permit. So sometimes it goes for higher than minimum price. So if there's no species listed, the minimum price is 10%, 10 cents per pound. The other requirement of our program is, you know, all collectors use roads, and we take 20% of the permit value to be put into a road maintenance fund so that we can do some road work. Next slide. So each year we get a updated pricing table from the seed warehouse, and this is set by basically what the last couple seed buys happened, you know, like, so how much the seed buys brought in for each species. That's set at 10% of the clean seed. So, you know, this is what we use to set our minimum prices. Next slide. So this is just a screenshot of, you know, we have a database that all these permits go into. I have all the forestry folks in Nevada put it into one form so that we're able to kind of look up this 5450-1 and see all of our seed permits in one place instead of mixing it in with, say, firewood or Christmas tree permits. So that's just our interface. Next slide. And so this is kind of how it goes. We talk about, you know, we put in what seed, pounds, the quantity sold. And what you'll see in this is there's, you know, most of the values going into this public domain fund, but then 20% is that public domain road maintenance fund. So that's just kind of how we divvy out the revenue that's collected on each permit. Next slide. And this is what the form you'll see if you ever get a seed permit. It's a contract for the sale of vegetative resources. And so it's pretty straightforward. It's probably one of our more streamlined permitting processes in the Bureau. So Not a super confusing kind of gauntlet to run on these. Next slide. So, if you're wanting to collect, it's best to contact us 30 days prior to needing to collect, but it's definitely required 10 business days ahead of time, as long as you have the full and complete information for the permit. So part of what we need is exact locations in any form, whether it be lat and long or township range and section. You need price, pounds of the seed and the species. And then, who's going to be collecting and when. So keep in mind, if you want to collect from multiple district boundaries, you're going to have to, contact each and every district you collect from. And so we've had a lot of turnover in the forestry ranks this last year. And previously I did give out the contact names, but we have probably 80% of our positions are vacant right now. So I just took that slide out. So feel free to connect with me, and I can connect you with whoever's acting at the district level. Next slide. And that's it. That's the quick and dirty permits. I have to put in a funny forestry joke, of course. So questions, comments?
Thanks, Kareem. We're actually, if you can stick around, we're gonna hold questions until the rest of our panel or the rest of our speakers have finished, and then we're gonna do a panel to end out the day. So if that works for you, those people that have questions for that are in Zoom, again, put those in the chat, and those in the audience hold those, and then we will get to those at the end. We're actually gonna take a 15-minute break. We're actually back on time. So thanks for being punctual, Corinne, appreciate that. So we're gonna take a 15-minute break. Jess, can you put up the next set of poll questions? And if those in the audience have filled out their poll questions, can you just give them to Amber in the back, and we'll make sure to collect those and synthesize them, and appreciate that. So we'll take a 15-minute break, and then we'll come back and we'll hear from Dash about some restoration, and then we'll have our last panel for the afternoon. Thank you. All right, we're gonna work to get started back on time. Thanks to everyone for filling out the poll questions. Again, those really help us learn about the audience and future meetings and ways that the partnership and the department can be engaging with those that are participating. So thanks again for that. So we're gonna start back again, and Jess, I have the presentation pulled up here in case you don't have it, so don't worry if you don't have anything to pull up for Dash. So our next presenter is Dash Hibbard with the Walker Basin Conservancy. Dash is a stewardship manager for the Conservancy, where he's been leading restoration efforts on old fields for the last 5 years. Oh, look at that, she does have it. He received his MS from UNR, working under Dr. Beth Lodger on local adaptation and restoration potential of several Mojave plant species, and has worked in ecological restoration and plant ecology reclamation in the arid West for the past 15 years. I will turn it over.
Thank you, Megan. It's nice to be here. Thanks for putting this on. Yeah, so I'm going to talk a little bit about our restoration projects, and it's going to be a little bit different from what I think most of the larger scale restoration in Nevada is going to would look like. So it's going to be maybe a little bit weirder. And if you want to get a better idea of some of what has been done on larger scale with BLM and Forest Service and NDOW and those folks, I would encourage you to look back at some of the past seed forums, because I think there are some good resources on that. All right, next slide. All right, so I'm going to start here with a little bit about who are we? What is the Walker Basin Conservancy? So we are an environmental nonprofit that is leading the effort to restore and maintain Walker Lake while protecting agricultural, environmental, and recreational interests in the Walker Basin. And so what that looks like is we purchase water rights from willing sellers within the basin. We work to protect those rights in stream to Walker Lake, which has had a massive decrease in its volume over the past 150 years or so. So we're trying to get that water back into the lake so it can once again support the food web that it wants to, get fish back in there, get loons back in there, get everything that lake once supported that is now too salty to. And along with that, we do revegetation, restoration of lands that are associated with those water rights. And we are also working to advance recreational opportunities in the basin. So on the lake, being able to get a fishery back in there is an important thing for us, being able to work on trails in the basin, those sorts of things, access to the Walker River. We're working on those sorts of issues. All right, next slide. So where do we do that? Restoration is in the Walker Basin, which is, makes sense, right? Which is in that blue boundary on straddling the California-Nevada border. We are responsible for stewardship on about 16,000 acres with our partners. So we don't own all that land. A lot of that we're working with partners on to share those stewardship obligations on. And about 5,000 of those acres are old fields. So formerly had some type of agriculture on them, were irrigated, and our restoration is focused on those old fields. And we do this on public land. We have purchased land that we have then reconveyed to the state in the form of the Walker River State Recreation Area, which is on the East Walker River. There are 3 larger ranches that you can see there. At the northern end of Mason Valley, we've reconveyed land to the Mason Valley Wildlife Management and area and NDOW up there. We also do work on some of our own private land, and we work with some private landowners. If they have water that they are willing to sell to the program, we will work with them on their with their needs and try to do some kind of revegetation to accommodate that decreased water use on that land in a way that is congruent with their goals for that land. All right, next slide. Some things to know about the area we work. This is one of the driest areas of the Great Basin. This is going to be really important. We are at under 5 inches of precipitation annually, so we're not quite Mojave, but it's getting closer. I think we're at 4.8 is the last average I saw for us, and we're Fallon's just beating us out at 4.9. So pretty dry. These would naturally be low elevation salt desert plant communities. They are, most of what we work on is former floodplains. A lot of that has been disconnected from the river, so they're not functionally floodplains in that sense currently. And we get into the transitions into those low slopes into the salt desert shrublands as well. These are naturally really low resistance and resilience environments, so it's really hard for them to bounce back after disturbance or resist any kind of major disturbance. Unfortunately, from our perspective, there has been a long history of intense disturbance on this land. From our perspective as restorationists, as there has been all the agricultural use that we were trying to reestablish native plant communities on is difficult with that that history of disturbance. Go to the next slide, please. So on these old fields, we get them in some various states based on their past usage. So if we look at that top right-hand corner, that's the kind of field that's about the best condition that we're going to get something in. That has native shrub establishment. A lot of that's going to be rabbit brushes. That's the first thing that's going to come in, but we're also going to get greasewood and some basin big sage coming in those areas. And those maybe not have the same kind of soil disturbance, but maybe if only had some kind of clearing done on them and have been, it's been a little bit less of an intense disturbance on those fields. If we're looking at the bottom right picture, we go a little bit up in elevation away from the river. We're looking at a lot of fields that look like this, and they're pretty barren. There's not a whole lot out there, good or bad, a lot of times. So that's, if you don't have anything good or bad, that's probably going to be bad. These areas were pretty marginal farmland to begin with, and were often had annual crops in them once every like 10 years or so. When you had a really good water year, you could put something in it, you had the water to do that. And then there are other fields like in the lower left that have a very recent history in production. This is an alfalfa field. And that has its own challenges to deal with. But the alfalfa can be, we're looking at that as something that will aid a transition into something. We don't need to get rid of that. It's doing some good things for us. And we just want to ease a transition as we decrease water use on these fields. So like I mentioned up front, this is a really atypical restoration scenario for Nevada. This is not what most of our large scale fire fire restoration looks like. But there are a lot of similar challenges that I'm going to try and tie into on the seed and plant materials front. So next slide. Our goals on these are dependent on our partner needs. Some of this, so we'll work with our partners, like I said, to whether that's a state partner with NDOW or with Nevada State Parks to work with what they want to see on that landscape and try and meet their goals on those properties, or a private landowner doing the same thing. At the very minimum, we're hitting things that aren't really going to meet that restoration definition. We're looking more at revegetation or rehabilitation and looking at soil stabilization, fugitive dust abatement, those things that go hand in hand, and then noxious weed control as well. Luckily, on most of our projects, we can work with partners like Nevada State Parks, like NDOW, that have conservation goals that match pretty closely with establishing appropriate native plant communities that are more like that restoration goal. So what we try to do on these properties is initiate a restoration trajectory, and we do that by seeding and outplanting native species on these properties. And our goal there is, I mean, it's not really feasible for us to plant anything at a density that's approaching pristine. So what we're looking to do is plant at a lower density, sort of baby those plants, get good survival off of them, and get them to a point where they can serve as seed sources for the rest of those fields. And it's a process that's going to take a long time. I think we got to realize that, but it's about the best that we can do on a lot of these, just to get those natural processes started and get what the building blocks going there. Next slide. And just throw in a few of the species that we work with quite a bit here, some of our workhorses. We've got our basin big sage in the top left. We got creeping wild rye that we use in a lot of our floodplain seedings. We have four-winged saltbush that we use quite a bit in our floodplain areas, trying to get some of those shrublands established. We use a fair bit of rabbit brush. I like the early successional plants on that, just because you don't have to plant a whole lot of it, and it will do some of the job for us to get those processes started. And what we probably plant the most of, that I don't think is something that nearly anybody else is going to plant the most of, is black greasewood. So that's something that is characteristic of our restoration in the salt desert. Next slide, please. So I'll go through a few of our challenges here. Soils is a big one. These have been highly disturbed due to agricultural activity over the years. So we don't have the same sort of structure in these soils. We've had our landforms are a little bit different. Things have been flattened out. That changes things. So our horizons in those soils are different. And we also have often hard pans in these soils as well, that when we do plantings, we try and break through that on our plants to give them a better shot of survival up front. We also have high levels of nitrogen in these soils, mostly due to past alfalfa crops. That's going to be nitrogen is usually going to be good for plants in isolation, but not necessarily for ours. Ours are adapted to low nitrogen and low nutrient environments. And when we have the high nitrogen environments, that's going to be comparatively much better for the weeds that are definitely a problem out here. So when we have those high nitrogen levels, our plants are going to do relatively worse, especially when we put water on them. We have irrigation histories that can change the salt profiles in these soils, which are naturally pretty salty anyway. And after 150 years of intensive agriculture in some cases, our native seed bank is going to either be very depleted or non-existent. Some of these fields, like this picture, there is some good stuff that hangs on. So this would be like one of those fields that had a little bit less of an intense disturbance history. And you see some of our annuals that are maintaining a seed bank in these. So that's pretty encouraging. I like to see that when I'm out there. I think this is a Mencelia. White stem blazing star. And next slide. Other restoration challenges. Water. Our restoration challenges with water are going to be very different from larger scale seeding and planting projects in the Great Basin, namely because we are lucky enough to have the ability to put some irrigation on our sites. So it changes how we do things quite a bit. And it's lucky that we do have that ability, because if we didn't, I don't know if we really could reasonably expect to have any kind of success, or the very smallest amount of success that would not count as success. We also are working on these old ranches, so there is infrastructure to bring to bear on these problems as well. So that's going to also come to us in different stages or different conditions too. Yeah, our ditches might work and might need a lot of work. When we do get water on things, we end up with a lot of weeds too. So next slide. Yeah, water plus nitrogen plus massive weed seed bank equals we have a lot to deal with that, or to think about when we're either seeding or putting in plugs for restoration, and having to go into those with the plan to deal with those, because they will get out of hand quickly. We deal with both noxious and agricultural weeds. The noxious weeds, we do control on them, but they're less of a threat to our restoration projects in the levels that we have them on the properties that we work on. But the agricultural weeds, like annual kochias, maybe the bane of my existence. When you get the water and the nitrogen, they can overcrowd things, and we can sometimes grow like a 6 foot tall kochia and just get some rainforests going if we don't do things right. We're trying not to do that. So having as many tools in our arsenal to combat that is good. So mechanical, like mowing or herbicide, or if we have something that it makes sense on the property, some disking or something else to prep for that, and try and decrease our weed seed bank is going to help us out. All right, next slide. So I'm going to talk a little bit about how we try to overcome these, and a lot of these are going to have some aspect of the plant materials side of things, and working with better plant materials can help us overcome a lot of these challenges. So first of all, we have this question of seeds versus plugs. I mean, we're talking about seeds mostly here, but plugs also come from seeds. So it's kind of a similar thing, and you need the right seed to put into a plug as well. So this is a related side of this. So can we go to the next slide? We often, our strategy right now is trying to establish anything that is broadleafed, either woody or a forb from plugs, because I mean, as in our conditions, we just have not had any kind of luck, even with irrigation, to establish these from seed. So currently, we're working with this from plugs. Hopefully, that's not going to be the case forever. Hopefully, we will work on this and find better ways to do this with less intensive methods to be able to do this from seed directly. We're not there yet on our sites. So one of the advantages to this for us are they're pretty dependable. We can get some pretty good survival rates for these out in the field, and we overcome a lot in the nursery, a lot of those early establishment barriers to germination, germination establishment, things like that. In the early stages of life, that when we have weeds and really harsh temperature and maybe not the right climatic conditions or germination stimuli to get going in the field, we can do that in the greenhouse and then put something in when that matters less. So that's big reason for doing that. Downside of this, it's not very scalable. This is expensive. This is putting a ton of effort into each single plant that you get out of it. Especially when we're putting irrigation systems out on there to support them. It's still a better outcome than we would get with direct seeding, but if we could not do this, that would be great. So seeding, we do a fair bit of seeding as well, but that has, for the most part, been limited to grasses for us. It's something that it's a little bit less of a nut to crack in terms of those early establishment barriers, and we can make some things happen. So they do have a higher chance of failure for us. I don't think that's just our story, but sometimes we put stuff in the ground and it hasn't worked. As we get more better seed and plant materials, I think that's something that is going to be lessened, that chance of failure, but it'll always be there because of the climatic conditions that we're depending on. If we have the right starting material, we can get some pretty good results on this. So those two pictures that there are a floodplain seeding that we did with creeping wild rye and basin wild rye and western wheatgrass that we got from Ed at Comstock. And so that's from mostly that Carson Valley. So that's about as close as we can get to where we are in terms of that climatic envelope that was talked about earlier today. Yeah, so when we're working with something like that, this is a floodplain grass and we have flood irrigation to put onto it. So we've gotten decent results. We can establish that sort of a floodplain grassland that we see in the lower picture. And I say currently on all of this because I think there's a lot of room for improvement as we go forward, do some science, do some research, test a lot of things, and work on our process in all phases to make it better. Next slide. There are also some practices we can do to our restoration that will allow us to give each of the components that we're trying to establish in a restoration project what they want a little bit more. So we do some separation of grasses from our shrubs and our wildflowers in our projects. So can we go to the next slide? So what this looks like is we will plant our do our outplanting in rows that are separated from any kind of seeding. So on this is a seeding and planting project that we did at the Flying M Ranch, and we're trying to get a sagebrush dominant community going there with some Indian rice grass primarily as a grass component, but some of the other grasses as well. So like Elamis and Sandberg's bluegrass, those types. And we've kind of, sorry, could we stay there for a second? No. Back one more, there, thank you. Yeah, so we've, these got 1 year of irrigation on them. And we have our shrub rows are doing pretty well. They got pretty good survival on these, and getting them to a point where they can have enough establishment to put energy into resources, resources into seed, and then fill into the grass areas, which are on the right-hand side of the picture on the right. There's a lot of weeds in there too, but this also allows for differential weed control on what we do. So we're going to have different options in the broadleaf areas versus the grass areas, and that'll help establish both. Next slide. We can also do this temporally. On this project, we established grasses first and then tried to establish shrubs into that. I don't think this one's as successful. I think I'd rather, when we're establishing those grasses first, they become a little bit too competitive in the area that we're trying to get the plugs going, and it hurt us a little bit on that. Next slide. We can also try to minimize our irrigation of weeds. So next slide. And the way we're doing that is at this point, wherever we can, wherever we're trying to just locally establish primarily shrubs, we're putting them on drip lines, and we're getting a lot less irrigation of those weeds that otherwise would be irrigated with a larger system like a flood irrigation or a wheel line system or any kind of overhead sprayer system. Next slide. And then the last thing that I want to talk about on this was what we wanted to use for more appropriate plant materials. Next slide. So I think this has been talked about quite a bit today, but what would I want as a restorationist to be able to use? There's several different types of diversity that I would want to be able to put out on these fields. So species diversity, having these species that are appropriate for our site. We're in the salt desert, and it's maybe a little bit more overlooked. Having that sort of diversity to be able to put the appropriate plants into our sites would be awesome. And to put a wider breadth of the species that are appropriate there too. So we can sometimes be pretty good on the shrubs and grasses and then not have the annual forbs or perennial forbs that also are appropriate for those sites as well. So getting that species diversity, and that ties into the functional diversity that we would want on these sites to allow for properly functioning vegetative community that's going to do everything we want for habitat and ecosystem services that it would provide. We're also looking at genetic diversity, so within population, that diversity that's going to allow those populations to sustain and have different reactions to different climatic conditions, other kinds of conditions that will allow it to be able to weather all kinds of storms. So that drought resistance, there's something in there that will handle it. It might not be everything, but something will be good at that and stick around, or something will be really good at producing in those wet years. Things like that. We want to encompass that whole variation, and that is going to be really hard in an agronomic setting to get all of that. But it's something that, as a restorationist, I want that whole picture there. And that ties into the locally adapted traits as well. So we've got something that being able to fit into our actual climate is going to be super important. We're not like we saw earlier, having something that is coming from Montana and like 18 inches of precip, and when we're putting it into 5-inch precip zone in a good year, really, I mean, if we're hitting average, that's good. We can't expect that to do well. That's kind of insane, right? So having those adapted traits for where we are actually putting plants in the ground, having that same sort of provenance there is going to be important for us. Next slide. So we can do a lot of things, and a lot of people in this room and with the partnership are working on this from a ton of different angles. So I think there's a lot to be done. It's exciting time here in this sphere. I think there's really cool things happening all over the place. So, but I wanted to talk about a few of the things that we're doing on our side with partners to help some of these issues. The first thing we're doing is collecting seed. This is going to be a first step toward any kind of research that needs to happen into either figuring out specific germination and establishment difficulties, figuring out what is actually best adapted to certain locations, what's going to be best for restoration, and that's all going to be context specific. But we're going to have some populations that are going to have more of those desirable traits for restoration, and finding those and being able to put those into production is going to be super important. So we're working with Endow on this, with some species down in the Mojave Great Basin transition. Those seeds are going to go to Beth's lab up at UNR and get tested on for restoration suitability. And hopefully when we get to the end of that process, we'll have some better seed to use that we can put in some producers' hands to increase and have available for restoration. We also do our own restoration collections, and this is more to supply our restoration needs for shrubs and forbs that we are outplanting onto our restoration project. So since about 2017, we've been using almost all seed, not entirely, but almost all seed that we've collected from the Walker Basin and close to our project sites, growing those out in the nursery, and then putting those back out into our restoration plantings. All right, next slide. Other thing we can do is experiment in the greenhouse. I'm really excited to be bringing Shannon on for a lot of this. You'll get to be doing all this, but experimenting in the greenhouse to work on the different species and techniques that it's been difficult to get growing. So this is one of the big hurdles that we have, is some of these species are just they're evolutionarily great at at bed hedging and just getting those having the correct strategies for their climate. That natural history does not necessarily translate into our wanting to turn them into something that we want to put in the ground easily. So there are some tough nuts to crack in there. The more we can experiment in the nursery, the more information we can have, the more successful we can have, both for growing nursery stock and knowing those things, we can possibly be more successful in our actual seeding efforts as well, direct seeding efforts. Next slide. We can also do a little bit of experimentation in the field. So this is something that we're just starting, and there are many people who are way ahead of us on this sort of thing. So we'll probably be looking to pick some of your brains out there. But what we're looking to do is experiment a little bit more in the field in Nevada, and the in Nevada part is important because, like I mentioned, we're going back to the 5 inches of precip. It's different growing things here. You have changes in production fields associated with those populations. Being able to do that here instead of for Nevada, restoration is important rather than perhaps growing all of that seed in Washington State or elsewhere, where we're coming from something quite different. So we're working on some seed orchards, and these are partnerships with Bureau of Land Management, NDOW, Community Foundation of Western Nevada. And the idea here is that we're going to be, it's going to take us a while, but have some more reliable populations that we can pull seed from for some of these important species. I mean, Jackson was talking about this earlier today about some of the increasingly unreliable nature of wildland seed collection. If we have something that's we can irrigate in situ, we can kind of ease some of that unreliability in really dry years and things that are causing that. So we're working on that and field seed increase. Yeah, so we're working on putting things in from plugs, putting in things from direct seeding. This is a project that we started this last fall to test some of those things and just sort of start out with throwing a lot of things against the wall, really. Putting a lot of species out there and seeing how they do, getting a really initial idea of those things, and working from there. It's as an iterative process. All right, next slide. And so the last thing we can do is share those results. That is a big part of what we're looking to do in our partnerships. We don't want to, if we learn anything, we don't want to hold on to that. We want the whole industry to be better off for anything we can learn. And that'll help Nevada restoration in general. And sometimes this is a tough thing, so we might just get perplexed. Thank you to our nursery crew for that. And thank you to everybody here.
Thanks, Josh. You can stay up here because we're gonna move to the panel section. So those that presented in person, Russ, Stephen, and Dash, if you guys can come up here, and then Mustafa, Jim, and Corrine, if you could be on standby with your fingers over your unmute, we'll move into questions. And again, I apologize that you can't see any of the panelists while they'll be answering questions, but you can hopefully hear them if you guys have any audio issues. So we're going to start with questions from the Zoom world. So if you guys can put any of your questions in the chat box, that would be great. And then we'll come into in-person questions after that. So thank you. All right, so we have a couple of questions. The first two are for you, Corrine. The question is, how do you find BLM lands where it's appropriate to apply for seed collecting permits? And you're muted, Corrine, if you're talking.
Sorry, Karine, I had it so you couldn't unmute, so go ahead now.
Yeah, I figured that out and I was like, oh no. Repeat the question now that I've been stumped by the mute button.
Okay, well, and I apologize for that. So the question is, how do you find BLM lands where it's appropriate to apply for seed collecting permits?
So, you know, folks that are interested in collecting, they go out and they scout. And most BLM land is kind of open to seed collection. We don't issue permits in WSAs and wilderness, which I'm not sure that we even have wilderness in Nevada right now. But so most areas are open for collection. So I would just scout it out and submit a map to the district office and see, you know, if that area, you know, part of the tracking we do is to make sure the same wildland populations aren't collected from repeatedly, and that's just a trend kind of thing to do. So it's best just to scout it out and send it to the district. Hopefully I answered that.
I think so. And the next question is also for you, so you can leave yourself unmuted. Where is BLM allowing conversion of lands to solar or solar plants or similar type uses? Is it possible to do seed collection or plant salvage before these parcels are cleared?
Yeah, so we definitely make those types of areas available for seed collection. We work with the solar development companies to kind of inventory what kind of plant resources are on those development areas so that we know what's being impacted. And so we've definitely had a lot of proactive seed collection, and even the development folks sometimes do transplanting and try to save those plants. One of the cool programs we have is like in southern Nevada, we have areas around Vegas that are open for disposal. And though in those we do like I think a fall kind of plant collection, couple Saturdays kind of thing, and we issue permits out of those projects. So yeah, I mean, I would contact the district that has the development projects ongoing, and they'll know exactly the timing on any seed collection or plant collection activities for that.
Great, thanks, Corrine. So our next question is for Dash. Actually, you have two questions. That's what happens when you go last. All right, so in your restoration efforts at the Flying M Ranch, what are some of the varieties of seeds that you've planted with the sagebrush?
So seeds that we planted, that was a grass mix that was, like I mentioned, dominated by Indian rice grass, but we also had some Poa secunda in there, Elemis elamoides, some of those a little bit more upland grasses there. And then we planted plugs, which the sagebrush was dominant there, but we also had some four-winged saltbush in there. We have some greasewood in there. We have some spiny hopsage. Probably, I think we put some rubber rabbit brush in there as well. Again, not a whole ton, because if it's going to want to be there, it'll do a good job of coming in on its own.
Great, thanks. And another question for you, Dash, with respect to plugs versus seedings, isn't it true that plugs can grow to nurse plant size, in air quotes, earlier and would produce seed to the seed bank sooner. And then with respect, so you want to answer that first and then I'll go to the next part?
Yeah, awesome. Yeah, that is also a big part of our reason to go from plugs. We do, when we do put those plants out there, we do, we can get them up to that size, and some of the sagebrush wants to grow really quickly, and we can get it that size in about 2 years or so. And then it's providing baffle for the wind and changing microclimates in those fields, and allowing some of those other processes to make a little bit of more habitat heterogeneity, a little bit more suitability for other things to take hold in those fields. There's a second part, I think, that I missed.
And would they produce seed into the seed bank sooner?
Yes, we can see some of those plants starting to produce seed in their second year or so. And on some of our older fields in the 10-year-old range, that were some of the first things that were started before WBC was even WBC. We are seeing some of that secondary establishment from the nurse seed plants. And so we're starting that restoration trajectory that's going to be a really long process for these fields, but it's getting there towards filling in a little bit.
Great. And then the second part of the question is, with respect to plugs versus seeding temporal separation, is it possible that establishing forbs into established shrubs may be more successful than establishing grasses into established shrubs?
Yeah, that could be. We have not tried that. I think it's probably something that makes a lot of sense. That site is something that we were working on private land on, so it was a slightly different goals. So it was more on trying to get as much short-term soil stabilization as possible, and then we tried to establish some of those native components into it sort of secondarily. So it's maybe wouldn't be my ideal to begin with, but yeah, if we're looking at something that is going to be mostly shrub and forb dominated, I think that's going to make a lot of sense.
Great, thanks. And then we'll move to in-room questions. Anybody here in Falmouth have questions for our afternoon speakers? Ed.
Hey, Ash. Hey, lots of our jobs over the years have started out herbaceous, colonizing species, early sorrel, native grasses, and over time turned to shrubs that we either planted or did not plant. And we've had sagebrush steppe communities run over some of our urban herbaceous communities on their own. And I'm usually relatively conservative and hopeful that the shrub seeds that you're putting into the ground will someday wake up. And on the other flip side, we have this one community of native grass up in Northwest Reno on a rock slope that took 16 years to appear magically. I mean, we don't have that kind of patience or time to wait for erosion control or ground cover that will, especially in the urban context. But I can feel for you for the farmland. So that even our own farmland was a Pandora's box of weeds when we initially tore out a historic pasture. But it looks tougher there, it really does. But A for effort, man.
Thank you. I have just some quick questions. So my first question is for Russ and Stephen. Sorry if I'm not as familiar, but with seed, you guys have mentioned that some of the seeds over time, you'll be able to know which ones are more successful via like their traits, and that's attached to the different seeds that are being put into onto the seed tags. Am I misunderstanding that, or sorry?
Well, I think what you're touching on there is seed certification in general, right? The whole process into admitting a seed or a variety into the certification program is a lot of vetting and genetic manipulation and agronomic manipulation to harness in and isolate specific traits that make these varietals more suitable or sustainable in the agronomic realm, right? So yeah, they're more predictable in that light, right? So whenever we're looking into seed eligibility or varietal eligibility for seed certification, there's a board that exists through what's called AOSCA, which is the Association of Official Seed Certifying Agencies, the Variety Review Board, and they have a checklist essentially of all of these characteristics that they're looking for on the variety, right? So the breeder fills out that application and dictates out, rate of emergence and leaf color and leaf shape and planting time and area of adaptation and 25 other different characteristics. And the board evaluates all those characteristics. And in some instances, they'll even do grow outs. So they observe these in situ and they'll approve or deny based upon whether or not they comply with seed certified standards, right?
Cool, yeah, that answered my question. And then I was wondering, when you say that you need to make sure that you have enough seeds for testing for purity, germination, noxious weeds, do you like it, for your example, it was like 7 grams for germination and 70 grams for weeds. Would you then need like 77 grams, for example, like you need that much for each of the different tests, like or is it like in total?
In total, really. I mean, I always say provide more than what's recommended anyhow, because there always is some room for error, right? But we can usually, if the standard for something like Indian rice grass is 70 grams for a noxious weed analysis, we can use that full 70 grams to complete the noxious weed and the purity. Yep.
And then my last question is for Dash. So I just was wondering with the plugs. So for people that are wanting to try out planting out plugs, but don't have irrigation, would you still recommend it? Like if somebody was to go out with backpacks full of water and go out every couple of weeks to be planting out plugs, or is that like how often are you irrigating these? And I know you're in really dry conditions, but it's also like the plants should theoretically be more adapted to being in really dry conditions. So I'm just curious.
Yeah, so we kind of baby them right off the bat, and because that's when most of our mortality is going to happen, right? So we try to make sure they're not water stressed then, and then if we got something on like a drip system, we'll move them from like watering once a week to once every 2 weeks to once every month over the course of about 2 years. I think that's probably a little more than they actually need. We've seen in past where, because this is restoration, nothing ever goes according to plan exactly. We've had some things that haven't gotten water when we've wanted them to, and some things have gotten just 1 year of water, and they've mostly been okay. Like, I think this is also going to be really site dependent and how much you want to put in effort on it too. Like if you have a few plants pretty close by, that's going to make sense. If you've got 10,000 plants that are 5 mile hike in, that's probably not gonna, but yeah, you know that. So, yeah, and I think if you go, I mean, also site dependent on what your natural precipitation is. I know we can get success at higher elevations without much or any supplemental irrigation with some of those plug plantings where you have a fair bit of snow, like sagebrush. You're planting those sagebrush into snow, and you're getting a lot of initial moisture to the plant in those early stages. Yeah.
Deeper supercells, the better, right?
Yep.
Yeah, so I had Brittany Trimble come up from Endow, for those that obviously can't see, so I'm sorry, but Brittany, I know Endow has also done quite a bit of plugging, so I thought she'd come up and maybe answer some of that too. Go ahead.
Yeah, I could just reiterate what Dash ended on there. When we're doing our shrub plantings, the traditional method was to try to get them out in fall to try to capture all that winter precip. But the way our winter precip is kind of changing across the state, we've been leaning more towards spring plantings and just trying to get them out as soon as possible in the spring. This year, for example, like February would have been a great time to plant, but it's hard to anticipate that kind of stuff. So unless you've have your plugs and you're storing them in like reefer trucks or refrigerated storage somehow until you can plant them in the spring. You can't quite be as opportunistic. So recently, Wells High School out in Eastern Nevada just gave us 10,000 seedlings last week. And so I spent a whole week just driving around trying to find places that still had good soil moisture. Last year we planted on a mastication project. So there was a lot of woody debris around that could kind of help hold in that soil moisture as well. And in both cases, we've been super lucky and gotten like 2 inches of snow the night after we planted everything.
Yeah.
So it's pretty hard to anticipate that kind of stuff, but you can try to plant in fall, just kind of hedging your bets, hoping that you get some good winter precip. But we've found more success with spring plantings recently. So.
Great. Do we have any other, or go ahead.
My question is for Russ. So with TZ tests, from at least from my perspective, purities, even on low purity type wildland seeds, those seem to stay at least somewhat consistent between labs. It's the TZ tests that are the most variable. They're done with less seeds, right? 200 seeds generally. I'm sure in the process, there's a certain amount of definitely yes, definitely no on the viability, but then there's these seeds that are in between. Why wouldn't there be a confidence interval that's listed alongside the TZ?
You know, Jackson, I think you bring up a good point. And at the end of the day, we're following the credo that's essentially given to us, and there's no confidence interval built into the AOSA testing handbook, right? I think that there maybe should be, and that might be just an incredibly complex concept to pursue just based upon the variability and the overall physiology and morphology of seeds out there. Because your confidence index or, very structure is going to probably depend and directly correlate back to the species at hand that you're operating with, I would imagine, right? But yeah, at the end of the day, I think it really comes down to, for TZ testing, experience is huge, right? Utilizing a lab that has more experience with the species that you're sending to them, I think is definitely a major factor involved too. The problem is that, in these sales that you're completing back to the BLM, you're only able to utilize a list. It's finite, right? I mean, there's only a select handful of official labs out there that the BLM essentially gives you. So it's up to you to pick and choose that lab that has more experience with that species. But I think that's a good point. A confidence index that we could develop might be something worthwhile to pursue that could lead towards betterment of that type of analysis, right?
I think there'd be more crossover,
especially
since it would be the seed company is the lab's customer, and so your customer's on the hook. So I feel like if there's a certain level of I don't know, that needs to be listed in the test instead of just a definite stamped number.
Yep.
So yeah.
It's one blanket metric. All we give you is 25%, 50%. That's it. And it doesn't dictate out on the lab report how the test was performed, under what conditions it was performed, who performed that test even. Likewise, I mean, we'll perform GMO assay on alfalfa seed, and we run it through an index that actually gives us that confidence interval. And we say this is accurate up to 95% confidence.
Yeah.
Is
it the AOSA or ASOA? Which one?
AOSA.
AOSA.
Yeah.
Under what's a forum that I could get that submitted to? Because obviously me telling you this isn't going to do anything, but where can I submit that?
What I would recommend is the Nevada Department of Agriculture is an associated lab with AOSA. Send me an email.
Okay.
And then I can go ahead and forward that on to the committees that are appropriate.
Okay, it may not make a difference, but it might. So
yeah, totally.
Great, thanks. I think that's one of the points of these forums, is for us to understand some of these things. So, obviously the partnership has many different members, and so our role with the department is to help fill that sort of stuff. So we appreciate that information, and we can try to work to do that. So thank you.
And then real quick, I just wanted to give a little bit of history and insight into Nevada Department of Ag Seed Lab, right? I mean, we've really truly only been operational for about 8 years. When I was first hired on, I kind of had to pick up the pieces to resurrect our lab. Way back when, I mean, we did have a dedicated seed analyst who was committed and, up on all of his training and everything of the sort that way, and was a great asset to the industry. But after he retired, the seed lab kind of fell by the wayside. And now we're kind of trying to pick up the pieces here and knowing this type of information as we're kind of a newer lab, and as we onboard new positions with Steven and myself, and ideally at one point we'll have a full-time seed analyst that can work through these problems with the industry. And as we evolve and mature, we can take these types of concerns into consideration as we develop our standard operating procedures.
Awesome.
Right?
Thank you for answering my question so clearly.
Yeah. Yeah, you betcha, Jensen.
Leanne?
Leanne Carey, Progressive Rancher Magazine. This is a really naive question, but that's okay, because I am. And when you have, I collect seeds all the time for my garden. I love collecting my own seeds. And I have found, and I'm a novice, I really am, is that the less I mess with that seed, when I go in the springtime at the right time and put my flower seeds out, that the more natural I leave that seed, the larger germination I get. If I mess with those seeds and I shake them all out and I make sure they're all clean and the chaff that has like stripped from that little seed to go where. So my question is, when you have a natural seed and it has a chaff that protects it, why are we cleaning these seeds to death? And so, is that a fair question? Because you don't have the natural protection the seed has when it's out wandering around in the wind and connected to animals and being cracked out by animals that have, or birds and such that have eaten it and carried it over miles and miles away. That's a natural planting. That will be a natural germination and protected by some nutrients. So if I'm making any sense at all, please, it looks like we're working the seed to death.
You want to take that one, Dash?
I'm probably not the best person here to talk about that, but maybe Shannon or Brittany. Go for it, Brittany.
One big piece, especially for the larger land management agencies that seed huge parcels of land, like for instance, the BLM probably seeds 500,000 acres of land every year just in Nevada. And a lot of that has to be done kind of in a streamlined process. So the seed has to be able to go through the equipment that they use for these large scale restoration efforts. So it has to be able to be put in a fixed wing airplane or a helicopter hopper and not jam up the bottom of the hopper while it's coming out of the plane, so that the seed can have equal coverage across the whole area that they're trying to seed, or it can't get stuck up in the drill hoppers or anything like that to kind of slow down the seeding process. And a lot of time, well, especially with drill seeding, you're correct that seeds do develop evolutionary traits that kind of help protect them and dig themselves into the soil and all that kind of stuff. But with drill seeding, you're kind of going around that. So you're already dropping the seed into the soil at the depth where it needs to germinate, ideally, most of the time. So you don't really necessarily need that protection on the seed most of the time. You're kind of enhancing the process of germinating it.
May I ask one more question?
I'll dovetail on that too, and I think that, Leanne, your question has major market and economic implications as well, right? We clean that seed so the buyer knows exactly what they're getting at the end of the day, right? More than anything.
But that does reduce the viability and percentage of the germination. What percentage, and I know this, and or I wouldn't ask this question. What percentage are we getting in the public rangelands rehab, fire rehabs? What percentage of germination are we getting out there?
So I think we can reframe the question for the people that we have here to answer that type of question. So I think, and Shannon, I'll let you jump in too, but I think there's to the points that several people have made about the testing requirements that agencies have for rehab seed that's going to go out on the ground has those purity, germination, and requirements and testing that is done, and if seed stays in a warehouse for a particular amount of time, it's required to be retested to ensure some of those sorts of things. So there's some of those requirements in a nutshell. I'm not saying I'm an expert in any of those sorts of things, but I also don't want to put our panelists in a position to answer for BLM seed warehouse sort of stuff. So there's that sort of couch. Thank you, gentlemen, for coming and presenting. We really appreciate it. And Shannon, go ahead.
Yeah, so I just wanted to chime in and say that there's lots of different reasons that we clean seed. One of the biggest things being so that we actually know what you have. So when you collect all of that seed out in the field, you have all of this extra plant material. You don't really know how much seed you have. You might have collected 40 pounds of material, but the actual seed that you have is like 1 pound. It just depends on the season and the collection and all of that. So one of the reasons that we clean seed is so that we actually know what we have. And when you clean seed, you typically actually increase the purity and the viability. So you increase those numbers. So it's a higher quality seed that you're getting because you're actually just at the seed instead of all of this extra plant material. You also clean seed so that you minimize storage space. So if you're trying to store 40 pounds of plant material, when you really actually only have a pound of what you're looking at, that is a huge space commitment to try to store something that's actually not that helpful. And when you're cleaning seed, you shouldn't be messing with the seed at all. I'm not saying that never happens, because it does happen. Sometimes you clean something and it was a little bit too rough and you're like, whoa, I just totally jacked up the seed coat. It happens. But when you are cleaning seed, you are not supposed to be messing with the seed coat at all. You're just supposed to be getting rid of that chaff material and separating the seed from the chaff. And the chaff, so I get what you're saying about how when you just throw it out there, it seems to do much better than when you put each one in the ground. I definitely get what you're saying. I've experienced those things before myself, but a lot of times the chaff actually will hinder germination. Yep. So like on Atriplex canescens, the salt brush, the wings on that, there's such a high salt content on there that it will actually hinder germination. And the plant has that mechanism there so that it can kind of time things right. So when water conditions are just right and that all the salt's been washed off and the bracts have dissolved, then it will germinate. So we're trying to kind of force this. So we're trying to speed things up on a time scale that works better for us. And nature has that whole hedge your bet thing, which is great and beautiful. Dash, you talked about it a little bit, but it's not a workable scale for us. So that's why you clean seed, and it really shouldn't be messing with the viability or anything else like that. If anything, it should be helping. Does that help? Does that answer it? Okay.
We have to re-clean some seed soil with those particular machines.
Yeah, and I was just going to mention that Steve made a comment in the chat on Zoom saying that the short answer is seed cleaning is simply to facilitate mechanical sowing, which is pretty much to Brittany's point of
Bingo.
sort of making that efficient. So appreciate everybody's answer and people jumping up to do that. And speaking of others, Steve also had a question for you, Russ. He said, are most of the 1,000+ seed testing protocols established by AOSA for exotic or common garden species rather than native? And then approximately how many Great Basin species have AOSA have standards for?
Oh, let's see, Steve. So the mass majority of testing standards that are in place issued by AOSA jointly through the SCST, which is the Society for Seed Control Testing Association. I think something like that. They develop the standards largely for more agronomic, traditional agricultural crops, right? as of late, probably over the last 25 years, there's been a lot more wildland seed types that have been introduced into the manual. Of the Great Basin species, oh man, what are there? How many different species in the Great Basin?
Ask Gary team.
So maybe one way that there's obviously some, but maybe this could be a good thing for our Nevada Native Seed Partnership website is to be able to link to some of those ASOA species that have those standards so that people have ready access to those sorts of things too.
Yeah, and then I will say, Steve, I've always been pleasantly surprised when I've received a very exotic to the seed lab or to traditional ag, but native to the Great Basin. Something like phlox, slender phlox, that's in there, right? Some more obscure species are listed in there. Yeah, so a good deal of them.
All right, thanks, Gus. Go ahead.
Are you using anything like DNA barcoding to verify the identification of seeds that you're testing? Are you able to do that? Are you interested in doing that? And then maybe related to the question that was asked about like the dirtiness of seeds and maybe some wilder, dirtier seeds being more viable. Would you be able to test for, you mentioned testing against pathogens, would you be able to test for like beneficial endophytic fungi or something like that in the future?
Yeah, we absolutely could. No problem there by any means at all as far as any sort of bacterial testing. We would probably simply just send that over to our pathological lab who has the ability to test for any sort of bacterial infection, whether it be positive or negative, right? Genetically speaking, we do have the capabilities to run real-time PCR to determine exactly what species or subspecies we're dealing with, depending upon whether or not that's available in the bank, right? But yeah, at the end of the day, if we have something that we're concerned about in a lot, like amaranth, for example, which is, you've got Palmer's amaranth, and then you've got red root pigweed, right? So you've got red root pigweed, which is just really a common weed species that's found in pretty much every agricultural field here in the northern portion of the state. And then Palmer's amaranth, which is a restricted species. You're looking at those seeds, they're virtually identical. So we would use something like real-time PCR to determine exactly what species we had. Yep. It's expensive at the end of the day, but yeah.
Great. Do we have any more questions? I don't see any more in Zoom. Do we have any more questions here? Bella?
Let's see.
All right. Well, thanks to our panelists and presenters today. I really appreciate everyone attending our first ever hybrid version of a meeting. So hopefully everybody found it successful. I wanted to do a couple of shameless plugs before we end our meeting. One, at the beginning of the meeting, I talked about our new Nevada Native Seed Partnership website, which is now live. So you can Google Nevada Native Seed Partnership and it should come up, and it's hosted by Partners in the Sage. It will have resources, information about our partnership, information about our seed strategy, our business plan, how to contact our different partners. So I encourage you guys to use that as a resource, and we'll continue to update resources. I also want to take a moment and thank the Fallon Convention Center and Candy, who's standing in the back of the room. If you can raise your hand, thank you so much to the Fallon Convention Center. Really appreciate you helping us host this and such top-notch service. So thank you very much. We really appreciate it. I also want to thank, yay, Jess, thank you. So this is our website. Perfect. If you want to scroll down and around a little bit, we really want to thank the Partners in the Sage and Intermountain West Joint Venture for their help in putting this together. We have struggled to get this together, but I think it really helps have a succinct place for all of our different members, whether it's nonprofits, federal agencies, or state agencies, to post and have a central hub for our customers. So you can find, if you can scroll down just a little bit, Jess. So where's my seed zone? I was talking to somebody about that. So there's that link there that'll bring you to the provisional seed zones and those sorts of things that you were looking for. So hopefully you'll be able to find some of that information. So again, it'll link you to many resources, and we hope that you find it helpful and communicate with us on ways that we can make it more valuable for you as our partners and our constituents. I also really want to thank Jess. Her camera has been off, but she's been hitting next on the slide screen all day, which I'm sure she never wants to hear the word next ever again. But Jess, I really appreciate you being our Zoom guru to be able to make this hybrid thing work. So I'm going to give you a hand for that. Sophie, oh yeah, there's Jess. Oh, you guys can't see her. Hold on, let me, I don't know how to unpin it so you can see her. Keep your camera on for just a second.
Go to view in the top.
No, it's on this.
It's on my computer. So thanks, Jess, really appreciate it. And Sophie from US Fish and Wildlife Service has also been copying and pasting everybody's questions and all of that on my sheet. So, oh, Jess, turn your camera back on. Now everybody in Fallon can see you, maybe. It's coming. All right. There's Jess Kindred, so thank you. And thank you to everybody for participating today. I hope we found it valuable. If you guys have any feedback, we definitely are interested in learning how we can make this as accessible and informative as possible. So any feedback, please send to Stephen, and we'll make sure that that gets passed around to those that need it. Again, we're going to be sharing the contact information of everybody participating. If you'd like to opt out, please email Stephen and let him know if you do not want your contact information shared. And again, the recordings of this forum will be on the Nevada Department of Agriculture's website or YouTube, excuse me, and as well as the Nevada Native Seed Partnership website. And
Real quick, Megan.
Oh.
So Steve, he asked the question, is it possible to host the presentations from this and previous Nevada Native Seed forums on the new NNSP website?
Yeah, so we're working, that is one of the things that we're working on. Thanks, Russ, for saying that. I didn't see that in the chat. So the Jess is highlighting on the screen, Nevada Native Seed forums. If you click that, you should be able to find the recording of last year's presentation, as well as all the other PowerPoints from the previous presentations. So hopefully that answers your question, and we'll update it with this one as well. And we'll do this, we'll have the same format for this for 2022s. So I know we're ending a little bit early, which I'm sure probably nobody is, we're all zoomed out and conferenced out, but again, really appreciate the opportunity. Thanks to Stephen for all the great work that he did in putting this together. The first couple of years, I think it was the Russ and Megan show, and I think this year was basically the Stephen show. So it's kind of nice to pass the torch. So I'm sure you'll feel better now that this is behind you, but you did a fantastic job. And thanks to all of our participants for joining us. And if there's no other questions or needs, again, thank you for joining us, and we look forward to seeing you next year at our next forum. Woo.